Ching, ching, ching. Hello, ching, ching, ching. Hello, ching, ching. Hello, ching, ching. Hello, ching, ching. Hello, ching. Hello, ching, 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 ching. Hello, sí, 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 hello, hello, sí, hello, sí.
You'll go into 5 different websites to buy your proteins, multivitamins, collagen, and BCAA. Haven't you heard of Yoga Life? Find 300 plus brands and 9,000 plus health products in one place.
Download the Huga Life app today.
Hello? We flew over the cloud, hit the sunrise. Then
Hello?
Nanny took us into the future.
All right, good morning, folks. Welcome to the third day of the Jellyfish Bloom Symposium. So it's my great pleasure to introduce this morning's chair, Delphine Thibault. Our co-chairs will be Esram Kumar and Rizman Idid, and our rapporteur will be Pramod Kiran. Thank you all. And I'll hand over to Delphine to introduce our speaker for this morning, our plenary, Alan Collins.
Morning, everybody. Welcome. It's my great pleasure to introduce Alan Collins as our plenary speaker in this session. Alan is actually my twin brother from another mother and another dad, as we were born on the same day, but a few years apart. We are both well preserved, maybe something to do with using formalin or other preserves, but some traits are maybe less well preserved. Alan Gilbert is a director and a research zoologist at the National Systematics Lab of the NOAA's Fisheries. He's based at the Smithsonian Institution at the National Museum of Natural Science in Washington, D.C. Alan's main research interests are on the evolutionary history of cnidarians, jellyfishes, among others, placozoans, and sponges. So he's looking at evolutionary trees, developing phylogenetic hypotheses to better understand the biodiversity of his groups. Young Alan sat on the bench of the Amherst College in Massachusetts during his BA, where he graduated in 1987. At that time, he wanted to major in something that will make him a lot of money. Don't know if he chose wisely. Lucky for us, he then moved to the University of California at Berkeley, where he completed his PhD in 1999. He did collect some DNA in the field, but curiously enough, he did it naked. So Alan is really into the field and giving his whole body to the science. Alan, as all proper plankton drifter for a while, moved around from the University of Sao Paulo to the University of California, and then the current making drift a bit westward towards the University of Kansas, the University of Maryland, and then Washington, D.C., where he finally settled down, as any good planula finally does in 2004. Without further ado, please welcome Alan Collins, who will tell us all about pace and scope in characterizing the diversity and distribution of jellyfishes. How are we doing as a community? Please, Alan.
Thank you very much. Really appreciate that introduction. And thank you all. Happy Thanksgiving back in the United States. It is a day of gratitude. That's my personal favorite holiday. And I'm very grateful and honored to have been invited to speak to you all. And I'm very grateful to our hosts who've been so kind and generous and welcoming to us all. And I'm very grateful to all of you who have come all this way to share all of your knowledge on jellies. I am a curator of what I think is the largest collection of jellies in the world, and I'm often introduced as a jellyfish expert or referred to as a jellyfish expert. And these last 2 days is very humbling, because it's a nice reminder of how much I don't know. And so thank you for all sharing. So I am going to talk today about the pace and scope of characterizing jelly diversity. And the first little part is a bit of a point of view. And then the end is some more current research. So by jellies, I'm talking not just about scyphozoan jellies, but the cubozoans, the hydrozoans, and in addition, the staurozoans, as well as the comb jellies. And I should have added salps and pteropods and other gelatinous organisms to this as well. had I been thinking, but I've been convinced for sure by some of the comments that have been made during the conference that we should not neglect the other jellies. So by characterizing, what am I talking about? So documenting basically all the information that could be useful about a particular species. Of course, there's morphology, macromorphology, micromorphology. Ideally, we know something about the variations of these morphologies, and we want to know the morphologies for all the different life stages. And then linking that together, information about how they develop, how the morphology changes during ontogeny. And underlying all that, of course, is information that's encoded in the genomes, the nuclear genome, the mitochondrial genomes, the genomes of symbionts, so associates are incredibly important. And then likewise, we want to understand how those genes are used by the organism dealing with stress or any sort of the functions. So we have all of these things have been illustrated by the talks that have come before this. Of course, ecology, habitat to prey, predator, parasite, and other associate relationships are incredibly important. The behaviors matter to us. We're studying jellyfish blooms in this, or focusing on jellyfish blooms in this conference, and so behaviors are incredibly important for that. You must be interested in phylogenetic position, so you would want to know this about every species, is how are they related to other species? That way, in the absence of information from that particular species, you can make predictions about characteristics. Geographic ranges, of course, are incredibly important to know, and then population structures. And we've seen some nice examples. I'm thinking of Connie's genomic-based work with Nemiopsis, which not only shows us population structures as they currently exist, but gives us information about recent time and recent events, even invasion events. And of course, we can all think of a whole lot more. I think our grand goal is to know pretty much everything about all jelly species, everything that could possibly of interest, of impact. And the key step to a key step in the process of understanding this for all jellies is being able to link the information together and the knowledge tied to something. So one way that we know species is by describing them and naming them. This is the species accumulation curve for the comb jellies, and at present we have about just under 200 currently accepted species, and 19 of those have been described since 2000. So in the last 23 years, about 10% of those. I just quickly looked up the number for salps and for the pteropods, and those numbers are 6% and 5% respectively. So in the last 23 years, we've added that. So there are hindrances to our progress in knowing diversity in this way, naming and describing things. For comb jellies are notoriously bad preservers, so they don't wind up in museums very much. They can break down very quickly after you collect them, so you might recognize something as new, but then not be able to document it fast enough because many of the diversity is in the deep sea. It's relatively inaccessible. And of course, for all of these groups, we have a relative lack of taxonomic effort. We don't have enough taxonomists. We know there's a lot of diversity out there to still describe. But is progress in characterizing ctenophores really that bad, that we've only make incremental progress? I'll argue that we're getting there faster. And the numbers are pretty similar for the medusozoan groups. Also, 10% of storozoans have been described in the last 10 years. The numbers for cubozoa are the exception, where fully half the species have been described in the last 23 years. And I talked about this way back in Barcelona, that there was this big uptick in cubozoan systematic work, and it has continued. So it's really nice to see that that pace is continuing. There's a lot of focus on this group because of obvious reasons of their biology. And then there are the numbers for scyphozoans and hydrozoans. 14% and 21% of species in hydrozoa have been described in the last 23 years. So that's a relative anomaly. But then what about this problem? Sometimes we get described this problem of known unknowns. So we tend to accumulate in the molecular genetic literature a lot of species that have informal names. So you think of all the Aurelia species or genus species X. So those are accumulating in the literature. So we are accumulating knowledge about species, but we don't have a formal species name for them. And then there are a bunch of things that we know by what they look like. So these examples from ctenophores, and these get sort of pet names. Ctenoceros, this is the cydippid comb jelly that has these horns. Benthotina is a name you sometimes see. Liver lips is another informal name. And these names can also be used to link up information. So we know that the first Aurelia genome was published as Aurelia species 1, Cerulean. And, but still, those, if we're careful, those names can be used to link up diverse information. And there we've even seen in the literature Aurelia species X described from some bay or whatnot, and its behavior. So that can be used, but they're a little, because they're informal, they're a little more vague, and they tend to be a little less certain. But they're still an important thing. So in the context of this whole question about, how are we doing, I have been thinking about what is the ideal species description. And it's just a list of actually knowing all those things that I've already ticked off. So I have a jellyfish, Scolionema. I discovered it in my lab, or actually Rebecca, my friend Rebecca, discovered the polyp. I want to learn the life cycle, so we've been able to raise it in the lab. I've been working on the genome and sequencing it. And of course, we would want to know, send it out for transcriptome studies. So what genes are doing, and there's gene cascades are being worked on in this group. I know that I can raise it all the way through its life cycle on Artemia alone, unenriched Artemia. So that's important information, but I don't have information on its natural diet. But this is something that would be that we would want to know for this organism, its natural diet and its natural predators. We have information on its phylogenetic position based on the genetic data, and I know that it comes from my little 55-gallon aquarium that I keep in my lab, but I want to know where it comes from in the world. And so I'd like to include that information if I were to ever describe the species. And then ultimately, it would be nice to know its population structure across this whole thing. So every species is this multitude of stories that we've been telling through this whole conference. And of course, they don't just happen when we collect them. They have these histories to them, near histories. A species might arise in one part of the world, but spread to others, and also deep in time. So I wanted to share a couple of examples of less than ideal species descriptions that I've been a part of. This is the story of the two-handed ctenophore from Puerto Rico. And back in 2015, this was observed. There were 3 separate observations. The lead scientist on this trip was my colleague, Andrea Quattrini, and she gave this the very first descriptor. She called it beautiful. And because this was seen with NOAA Okeanos, they have a very rich telepresence process where there's lots of scientists back at home that can watch live and comment. It was also seen by my colleague Mike Ford, who called in and then was on the live broadcast, and he also said, wow, that's beautiful. But then he went on to describe its morphology, noting the general sort of body shape, the 2 tentacles, and referred to it as a cydippid ctenophore, very general. But we decided we wanted to write a paper about this observation, even though it was only recorded on video. But at this time, I had a very talented scientific illustrator working with me in the lab, Nick Bizzaio, and he started sketching this out, and I realized we were getting way more information out of the videos than I expected. And I thought, well, maybe we can actually name this species. And so we did. We got the name Duobrachium sparksei. And instead of a physical voucher, we have high-definition videos that are also, they serve as the holotype. So these videos and our description are what equals this species. Less than ideal species descriptions, but I think which move the field forward. My good friend Jonathan led this study on Aurelia, and we all know that Aurelia, some of the species have definitive morphology that's easy to tell apart, but many of them have accumulated in the literature as species X because they are difficult to tell apart. And we can do morphometric work, and indeed, Jonathan was really interested in trying to connect morphology to these species descriptions, but in the end, it was more relatively easy to differentiate these lineages based on the genetic data than it was on morphological data. And so that's the idea that maybe we could describe them using these characters as the diagnostic characters. And so many of these species that we created in this paper are based just on genetic data. And that's a less than ideal solution, but it, I think, moved the field forward, solidifying names that can then be used to join up information that are gathered by others. Now I want to just turn to some work that's going on in my lab with Nick Bazzaio. I'm going to focus on ctenophores and ctenophore biodiversity characterization. So we all know about the phylum. There's about 200 species. Comes in a lot of different body forms, the lobate forms, the burrowid forms, the tentaculate forms, and the benthic ones. So one of the groups that Nick is focusing on are these burrowies, and they're basically living stomachs. The majority of the body has a vast gastric chamber. It's pictured in gray on the right. And the system that converges at the apical end of the animal branches into 8 canals. 8 canals that go under the team rows, and from those canals we have these diverticuli that form. So burrowy literature, Nick's been going into the literature and looking at all the different names that have been suggested for different burrowy species, and he's found accumulated about 300 different species descriptions. And then his assessment, after looking at sort of obvious synonyms or taxa for which the description is too low quality to make anything with, so species incorrenda, leaves about 16 different burrowy names. But people are collecting and sequencing burrowy around the world, lots of different groups, and they tend to always get identified by these 6 names. Burrowy abyssacola, for instance, most burrowies that have deep red pigmentation will be called Burrowy abyssacola, Burrowy cucumis, gracilis, etc. You know these things. But what are the genetics telling us? So basically, they are far more diverse than 6 and more diverse than 16. At present, we're doing species delineation methods that indicate that there's about 20 different of the burrowy lineages, distinct lineages. Now, because Nick is has a great eye and is a keen morphologist and is able to transfer that morphology to images that people can understand, he's been drawing the specimens that we've been using to collect data, or that other people have collected data. So they come in a variety of sort of shapes, right? But these animals are very squishy, and they can change these shapes in very short periods of time. And so he's looking at other characters, and in particular, he's been focusing on the canals and the diverticuli. And so you can see these are cross sections of the canals with how the diverticuli come out from the canals. And he's able to find unique patterns that associate with some of the genetic lineages. And so this is a possible character tool. He's looking at other characters, of course, like the macrocilial teeth at the front ends of the burrowies. And this is allowing us to make first inferences of how these characters have evolved within the group of burrowi. So you can see with a simple tree like this, we might infer that the type 1 teeth are the ancestral condition within the group, and that these type 2, 3, and 4 have evolved independently. So the other group that Nick is spending a lot of time focusing on are the platytenids, the benthic ctenophores. So these animals, they do have a pelagic part of their life cycle. They do have the combs and when they're juveniles, but they settle down onto the bottom. All but one species lose those combs, and they settle down on their mouth, and then they have the 2 tentacles that they deploy up into the water column in order to catch organisms. So He's roughly dividing these into sort of shallow water groups and focusing on them, and as well as the deep water groups. And this is an example of several pictures of Coeloplana, which is a quite diverse genus found in many shallow water environments. And these species have been differentiated in the literature and described based on basically 3 different characters: the arrangement of some papillae that are on their aboral surface, the host or substrate that they're found on, and then also the coloration and the patterns of the pigmentation across the body. So, but how do these characters relate to the genetic data that's coming out on them? Well, it turns out that these 3 features do a good job of sorting out the different species. So unlike the Burroes, there's quite a good correspondence between known and described species, not perfect, and these characters. So these genetic data reinforce the morphological delineations. He's also working a lot on deep water platytenids. So in present, there are only 2 species of platytines that are described from greater than 200 meters, and that's Lyracteus imperatoris and Gelfiella tristoma, described from very different parts of the world. But we know from lots of ocean exploration that is associated with high definition video that there are quite a number of species that are down there living on glass sponges and octocorals typically. So we know that there's a lot of known unknowns. There are species there to be described. And as we've been accumulating the genetic data, in fact, it's true that almost every different one that we've been sequencing is a distinct lineage, one from the next. Now, for this work, Nick has been doing computed microtomography, so micro-CT, and unlike most comb jellies, the platytines are quite good at preservation. So these are preserved, and then they're amenable to being put in the micro-CT and studied. And then Nick, because he's such a good illustrator, is able to color code the different parts of the ctenophores, these specimens here. And then he's making drawings of these new species based on the micro-CT data. So we expect to describe a lot of these species in the near future, and they will come with sort of morphological descriptions coupled with the genetic data that differentiates them. Now I want to turn a little bit to the method that we've been using to generate genetic data in our lab lately. It's not a particularly novel technique, but it's called genome skimming, and we've been doing it at a large scale. We've been doing this for thousands of fish species. The US wants to have voucher-based genetic data for every species that lives in US waters. We've been doing it for sea stars, and I've been doing it for hundreds of sponges. So it's really simple. You just start with a DNA extract, and this does not have to be a really high quality DNA extract, because one of the very first steps you do is you shear this DNA down to about 400 base pairs, so an average of 400 base pairs. Then we pool those, we tag them, pool them, send them out to sequencing on very new technology sequencers, NovaSeq sequencers, Illumina data, paired 150s, and what we wind up is, then we get 10 to 20 million reads per sample out of these. And then what we do is we just basically assemble them. We create kind of a poor quality whole genome assembly. And you see that in the bottom right there. That's basically just seeing where do these sequences line up, right? Where do they go together? And that gives you a list of contigs of various lengths and various coverage amounts. So that's the amount of data that's sort of behind them that lines up underneath them. And then with that list of the contigs, we're able to search through that bioinformatically to look for, so where is the cytochrome oxidase 1 gene? And that almost always comes out on a long contig that corresponds to the mitochondrial genome. And we can look for other data in there. So we've been mining it for the ribosomal repeat units, which is 18S, ITS1, 5.8S, ITS2, and 28S. And then these data contain a lot of other information. There's microbial data in there, so we've been mining the sponge ones for microbial data. The ctenophores are full of photoproteins. So evidently, photoproteins have a lot of tandem repeats, and the types of data that are easy to get out of this are data that are abundant in the genome. So the mitochondria, we have lots of mitochondria, the ribosomal repeats. Oh, advance. So here's what we've been getting for, we've tried about 107 ctenophore samples, and a couple of those were non-ctenophores. 5 of those failed, but for 90 of the 107, 84%, we're able to retrieve the complete or the very near complete mitochondrial genome. And we have 10 partial mitochondrial genomes, and they all include the commonly used COX-1 barcode. And of those 107, more than 90%, we can get the complete ribosomal repeat unit. These numbers are very similar for what's our success rate in sponges, sea stars, and we're doing Medusae as well. So this technique is relatively inexpensive too. So we can do it for about $40 per sample, which is not that much more than trying to Sanger sequence 2 or 3 markers. So of course, there's lots of other information that we can get out of that. This is a backbone phylogeny from these new data across the ctenophores. And in addition to sort of supporting the taxonomic work that Nick's doing in the baroids and platytenes, we know that there are people who are working on some of these taxa that have these known unknowns like Tenoceros or liver lips, and we're able to place them in a phylogenetic context and provide this data to the people who are describing these taxa, so which we will do. Also, I want to mention that through this genetic characterization of species, these make them accessible to being found in metabarcode libraries. So these are cytochrome oxidase 1 metabarcodes that you can see directly link a sequence to this in this case, a ctenophore Euramphia vexillogira, I think. And this is the ctenophore that inks. And we know more about its biology, and so now we know the places that this can occur. These metabarcode data and metabarcode occurrences are going to be put into GBIF, and so GBIF is working in a way, the Global Biodiversity Information, and this will rapidly expand our knowledge of what organisms are found in what parts of the world. There are other one of the reasons we did the genome skimming is that many different groups are targeting metabarcodes of the fish people like 12S or some part of 12S, cytochrome oxidase 1. This is a case where another guy, Luke McCartan, who's a PhD student at Lehigh University, he's working on corals, and he designed some coral-specific primers for 28S that he thought were coral-specific. And he told me they had a bunch of non-specific amplification. And so I asked him if I could see those primers, and they're perfect matches for the ctenophores. And so he has been generating lots of ctenophore data that he didn't know about. And then on the left here, you can see some of the ctenophore barcodes that are aligned against our voucher-based 28S sequences. And so this will tell us what diversity we're missing, where we need to collect more stuff, and pretty excited about where this is going to take us. So we've heard a little bit about eDNA, and I'm in the museum, and the museum may become a place to voucher eDNA samples, and I would suggest that you consider vouchering at least some of your samples, putting them in a museum so that they can be used by later researchers for other technologies. And in the presence of other knowledge, you may be interested in species X to do sort of like quantitative PCRs out of that, but those samples are a snapshot in time and space, and you're never going to collect that sample again, right? You're never going to go to that place again at that time. So they have lots of stories in them because they're holding lots of species occurrence status, but they can only be identified if we have reliable voucher-based barcodes that you can go back to and look at the original specimen and say, no, 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 that was wrong, you know? So we need to have a system in here where we can fix these things. And of course, eDNA is being deployed by lots of people. I work for NOAA Fisheries, and then NOAA Fisheries is planning on collecting tens of thousands of these samples for their work just in US waters. So I know that this is going on in lots of different places in the world. And so the genetic characterization combined with the key is the museum specimen that someone can go back to, even if it's high definition video or good photos in the lab before the animal fell apart, because eDNA is being used to address all sorts of fishery management questions, and to influence decision making. And with that, I would like to thank you very much for your time and attention, and especially the bunch of different Tina4 researchers that we've been working with and will be continuing to collaborate with. And I am happy to take any questions.
Thanks, Alan, for a wonderful talk. Any question?
Yeah, great talk over here. When you're doing the morphology of the pelagic ctenophores, are you doing that mostly from live specimens, or do you have some secret preservation method you could tell us about?
I do not have a secret. There was a method published not too long ago that has to do with Rain-X, which is a commercial product that you put on your windshield to bead the water up, and that people have used. I tried that once. I did not have much success with it. I tried it on a deep sea cruise, which was a really dumb idea. I need to practice this with animals, but no, I think the very best thing, and Dougal has made this point much in his papers, is to get really good photographs and video of the animals when you bring them on board, because you're gonna lose that information nearly always.
Any other question? Connie?
You're welcome.
Fantastic talk. Thanks, Alan. So should we basically have a modification of our approaches and kind of go over to genome skimming instead of, having primer-based specific targets, that we kind of have a harmonization of methods?
Yeah, I don't like to tell people what they should do. And not everyone has access to that. It is very simple though to make the libraries and to send them out for sequencing. So I do recommend it as a good technique, but it also has limitations, of course, right? So you're not going to get genes or certain things in that assembly that you might be interested in. It's a very, relatively poor genome assembly. So as an alternative to traditional Sanger barcoding, for sure. Yeah, I think it's a really nice approach, and I would recommend it, but I certainly, wouldn't want to mandate.
With with the micro CT scans of the benthic ctenophores,
Yeah.
Was that done like in air or in a liquid, or they're still attached to a substrate?
We basically put them into a small, small Eppendorf, the smallest one that they'll fit in, with a little bit of just sort of spongy stuff to keep them from moving around. And they're in 70% ethanol stained with phosphotungstic acid. And yeah.
And so the scan is while they're in the Eppendorf?
Yeah, they sit in the Eppendorf, and then they twist around, and then you can remove the plastic and the little the spongy bits in there that hold it in place, so that, because you don't want it to move as the scan is going and it turns around on the stage.
Oh, that's cool. And for the if you want to go back in time for the genetic work for species that we've already described, you're saying you cut it into 400 base pairs and then try and assemble it. And I can imagine there's lots of bits missing when you're trying to assemble.
Yeah,
And-
it's not very good for assembling data that have lots of repeat elements in it. So parts of the genome that are repeated a lot, the assemblies will get confused if you use short pieces of DNA. You'd like to augment that with long reads. But I would argue that a lot of these are sort of like it's like a good first step, and you might discover things in these data. Some of the fish people have contacted us looking for ultra-conserved elements. They wanted fish borrowed from us, and we said, oh, we've published some genetic data. Can you find them in there? And they said, oh my God, we got everything. We don't, you know. So I don't know if it'll always be that good, but
Have you tried doing nanopore as well as the nanopore sequencing as well as your other stuff on the same animal?
I have not. But that's a good idea.
Thank you very much, Alan, for this talk.
Thanks, Professor Colin, for the excellent presentation. And on behalf of the organizing committee, we wish to honor Dr. Colin with a memento. I request the chair to honor, I think that's your sister, to honor. Thank you. So there is an announcement for you. And by 10 o'clock, we'll have a parallel session, and which will be organized in a mini auditorium, which is just above the cafeteria. So those who are willing to attend the one, so please proceed there after 10 o'clock. Thank you. Yeah, back to the chair.
Thank you very much. The next talk will be by Michael Dawson, and he will present Symposium, Systematics, Genomics, and the New Age of Reason.
Okay, thanks, Delphine. Just quickly, two announcements, which I forgot to make earlier. One is if you're a student and you would like to be evaluated for the awards, then please let the chairs know as you come up. And second is that the registration booklet that everybody's signing is now out at the registration desk. So if you didn't get a chance to do that, pop out there and you can do that. Okay. All right, so you'll see a lot of similarities, I think, between Alan's talk and what I'm going to say here. The emphases are a little bit different. We disagree on one or two things, and that's all good, right? So we're good colleagues and stuff. I took a couple of different other people's graphics over on the left side of this, but they kind of like speak to this integration of molecular and morphological or phenotypic data more broadly. The title is a play on a title of a paper that I wrote in 2005 about Renaissance taxonomy. Both allude to the periods of intellectual advancement in the Western societies through the 14th to 19th centuries. And I would argue that we're at a time when there can be a new enlightenment, a new age of reason, by bringing more data to bear in our approaches to taxonomy. Okay. All right. So for 250 years, right, we practiced systematics by looking at a relatively few morphological characteristics that were not quantified, and the simplicity and ambiguity in the characteristics were interpreted subjectively, and it led to essentially a lot of confusion in cyphozoan systematics. Right. This is not unique to cyphozoan systematics, right? It's for many of the gelatinous zooplankton. The examples I'll give are cyphozoans. And the problem with this, right, is that Alan has said, is that the species are where we put all of the other information, right? It's how we organize stuff. It's how we understand life on Earth. It's how we understand life in the plankton and how we interpret it. So we have to do a good job of making the boxes and putting the stuff in the boxes, right? So in around 2000, essentially, we started to introduce quantitative morphometric techniques and molecular data for trying to identify species of cypsones. Not the first to do this in taxa generally, right? But we try to introduce it to this. And it generates really quite remarkable results, right? And so this is an example from Aurelia. You've seen this tree already this morning in part. And because you have suddenly thousands, not a few characters in DNA versus the qualitative morphological data, and because you can have quantitative characters in the morphology and many more of them, you gain a great deal of resolution and a great deal of stability, right? And these things are repeatable because they're quantitative, they're measurable, they're not about subjective interpretation. And for the most part, you will get reliable taxon or OTUs, if you want to call them that, operational taxonomic units, that are morphologically distinguishable most of the time, right? Not all the time, most of the time. Yeah, I would like that. Okay. But not all the time, right? And the reason is that jellyfishes or gelatinous zooplankton, they are simple, right, in terms of their morphology. We can probably go measure many other things about them, other phenotypes of Alan talked about, various techniques, physiological things, behavioral things as well, right? And try to put them in these boxes. But for morphology, it is ultimately limited at some point. How limited? Well, there are some couple of like basic mathematical issues here, right? So you essentially need about this many number of characters that are informative and reliably distinguish species to have a fully dichotomous tree, right? But that's not a fully dichotomous tree that is statistically well supported, right? To do that, you need something like this number of characters, right? Reliably informative characters. So where are we with Aurelia? Well, so John Lawley's paper, right, reckons that there are maybe about 28 species currently identifiable of Aurelia. So that would mean for a fully statistically resolved tree, we would need that many characters, right? How many do we have? Well, it's probably something around this in terms of things that we measure, right? But the things that are actually reliably informative, well, there's some variation in these, so maybe you get more, but then a bunch of them are not informative, so you actually end up with fewer actual informative characteristics, right? So our shortfall in morphological data is that for generating a resolved tree, and this for generating a statistically robust resolved tree out of morphological data only. And that's for the medusa, which is relatively morphologically complex, right? If you're trying to identify from a fiery or polyps, then it's a much harder problem. And this isn't to say that we shouldn't measure these things, right? But that we should not have an expectation that we can resolve taxa from these things. But it's kind of not the point anyway, right? The point is that we can measure these things and then we can start to put them into these more reliable boxes that we have that are taxa. And by doing that, we can learn about the way that they make their living and we can compare the ways that they make their living. So we can identify characteristics that will distinguish some taxa from another, right? It might not give us all the information, but we can do that. In generating this information, we also can identify differences between members of the same species that are in different places, right? The key thing here is that we're generating information on variation, right? Variation is the substance of evolution, is the way that things make different livings. And so it starts to generate important questions, right? So simply the act of measurement generates these important questions, such as why do these species differ or not in not only their gross morphology, but also particular traits. And we can put that into its geographic context, which is also its environmental context, right? So then what are the factors that explain trait variation, whether that's genetic or morphological or some other kind of phenotype? Why is it distributed the way it is spatially, environmentally, right? These are the key questions that we're interested in. The key questions will help us answer how things are going to respond to change in the future. An example of this, I've shown this figure before. I still have not written it up, unfortunately, but we'll change that, try to change that soon. Is this information about morphology and genotype tells us something about the way things make a living, about patterns of diversification historically, about functional traits, right? And give us a macroecological, a big picture, a big picture understanding of cyphosome diversity in this case. And so the example that's up here is from Lisa Gomez Daglio's fantastic work in the tropical Eastern Pacific, where she found 25 new species. And what it shows is that rarely do you find a species within the same genus occurring in the same location, right? You almost always find species, the taxa that co-occur are in different genera, different families, right? These are the exceptions for Chrysiora. And so this suggests that that there's some kind of functional exclusion between species, right? This is niche separation, niche diversification. This is not how we used to think about the plankton, right? It's a bunch of things that drift around, they're generalists, right? It's not what plankton is, right? We learn this through this integration of phenotypic and molecular data that allows us to build these reliable structures for boxing up the information into things. All right. Let me give a totally different example of why, some of the ways of integrating phenotypic and genetic data. So about 5 years ago, this is from Carl Higgins' thesis. About 5 years ago, we happened to be in Palau in marine lakes. This was one of those periods of times when there was not many jellyfish in the lakes, right? There are usually millions. People will go as tourists to go see this phenomenon of many millions of jellyfish in the lake. There weren't that many this particular time. But the interesting thing was that there were some animals that were like normal, zooxanthellae, right? So they have this brown color from zooxanthellae. And there were others that were syntopic in the same lake at the same time, just intermingled with each other, that were white and largely azooxanthellae. Okay. And so we're like, are these things genetically different? Are they behaving differently, right? We know that these mastigious might have a diurnal horizontal migration. And so we're like, let's measure it. So we took some samples for genomics. It turns out that genomically, they're more or less indistinguishable. But if you look at the behavior, the white ones don't migrate as strongly as the golden ones. And so the golden ones, this is a Rose diagram that shows an instantaneous swimming direction. Essentially, the golden ones are oriented towards the sun in the morning and in the afternoon, whereas the white ones are randomly oriented, right? So these are genomically indistinguishable hosts in the same environment at the same time. This is the zooxanthella making the migration of the mastigious, right? That's an amazing thing. Like, how does that do that? Right? And this is just one example of the interaction of a zooxanthella and a cnidarian host. And for cnidarians, right, we know about interactions of zooxanthellae and cnidarians and zooxanthellae primarily from a two-dimensional benthic environment, right? Corals. And so what happens when you take that symbiosis and you put it into a much more complex three-dimensional environment where you can modify things with behavior, right? So this is one of the questions we're interested in asking now. But it's not just that behavior, right? There's a long history of evolution of symbioses in cyphozoans. There are at least 2 independent origins of symbiosis within cyphozoans. There are more if you go out across the other cnidarians. And some taxa seem to have single symbionts, others seem to have multiple symbionts. Why? Right? We don't know. What's the consequence of that? So there are evolutionary transitions to understand. There are also life history transitions to understand, right? Why do they not have vertical inheritance? What are the benefits of horizontal inheritance of zooxanthellae? And for mastigious, it has kind of like a traditional-ish life cycle in this context, is that you lose the zooxanthellae when you reproduce. But there are other taxa, such as Cephea, that loses the zooxanthellae essentially around here. Why? Right? What are the benefits? What are the costs of having zooxanthellae at different life history stages? So this is a project that is being partially funded in a way by the Moore Foundation and the Wellcome Sanger Institute. The portion of the project this is paying for essentially is this project we call it Symbiosis in 3D. And they're generating high quality reference genomes for 50 species of cnidarian zooxanthellae pairs, right? So 100 genomes together. And the idea is to build a reference genome so that we can go ask all of these other interesting questions about symbiosis. This is the team that's doing it, right? So Alan had a very similar graph figure like this at the end of his talk, right? It's a global collaboration. We heard, saw Rebecca Helm with a similar thing yesterday. And it's not possible without this diversity of people working in diversity places, right? And it's not so much fun without those folk either, right? So, but it's not just the symbiosis, and it's not just the fun, it's not just the reference genome, right? The interesting thing is to build the phylogeny, understand the boxes, and to put all of this other stuff into there, right? Is it symbiotic? How symbiotic is it? Where does it live? What are the environmental conditions there? What's its growth morphology? What are the characteristics of, for example, this like highly engineered, apparently, canal systems? Why does that vary between taxa? What's it doing? Which bits of this are actually adaptive and which are not adaptive, right? There is this great idea about that Alan, what's the anyway, I'll come back to it. All right. Not everything you can measure is adaptive is the basic idea. Spandrels of San Marco, right? Stephen Jay Gould, right? Got it. All right. So in summary, right, we've got like a huge amount to learn about species distributions, life history, their interactions, the morphology, the physiology. Alan named more, right? Systematics, phylogenetics, taxonomy is the way we organize that information. Molecular and phenotypic data are more powerful together, I will argue. Right? The simple observation raises questions, gathering data on it focuses those, it allows us to make hypotheses that we can test and learn stuff from. I think we're at a time of unprecedented opportunity, right, in terms of being able, we've seen lots of great talks here, all right, on new ways to collect data, but it will pass us up if we don't take it. Right? And so I think we need to be active about saying we have lots of ways to collect data. We're here as a community. Like, let's chat and let's work out what collaborations we can make so that we can grab this opportunity and learn a bunch of stuff, have a new enlightenment, blah, blah, blah. All right, so I just pulled out like a half dozen examples of some of the great talks we've seen that cover different aspects of this, right? And it's not all of them. Other people here have talked and like given great talks too about stuff, right? But there's lots going on. So what should our next steps be? And I'm being a little bit more dogmatic about this, or whatever, let's stand back with Alan, because I think we should do this, right? And we've kind of like had these conversations before, not necessarily done a huge amount about them, except individually, but as a community, I think we can do these things, right? So I think we should establish some working groups. Let's chat about that before we leave here. We should then identify some priorities, maybe some low-hanging fruit. We should build and expand our collaborative networks, building on strengths and opportunities, right? Some of us are able to do some things in some places some of the time, right? But together we can probably do a lot more, right? So let's like take up those opportunities. We should standardize some approaches. I put Mark Gibbons' name up there because Mark talked to me a couple of years ago about essentially writing a bunch of papers on standardization so we can use the same protocol so we can actually integrate the data that we all collect, right? We need to identify and create data structures that will facilitate integration, right? It's not enough just to collect the data. If you're a late stage grad student, early career postdoc, like the world is your oyster, right? Reach out to us if you want to like take a leadership role in some of this stuff. You have like the time and the opportunity to do it. And then we'll need to get some funding, et cetera, blah, blah, blah. But we should make it our goal to report back on this at the next Jellyfish Bloom Symposium meeting. Okay, all right, thank you.
Thanks, Mike. Always interesting to listen to you. Any question? There's one question over there.
This always kind of happens to me. I like give these talks and there's like questions, like no questions. Was that like so bad? Or
not, we're still thinking about what you have been saying.
You know.
Thank you for extremely interesting talk. And is there any possibility that the symbiont mediated gene expression or something like that in the case of jellyfish?
Yeah, so we don't know, right? We haven't generated any RNA expression data yet, but that would be the idea, right? There's something going on. I suspect that it's not solely energetic, right? So there was talk about energetics and differences in pinealoid reproduction, right? And so this is kind of like why I asked that question about what's going on there, what's moderating that. I think it's more complex than energetics alone.
There's one question from Rizman on the stand.
Hi, Mike. Interesting talk. Just going to go into sort of eDNA or metagenomics a little bit, particularly in terms of which areas are still needed, especially having sort of regional references. Because sometimes that's one of the issue whereby suddenly you get all these unknowns.
Yeah, and yeah, I mean, I think we can all probably guess the areas that need more research, right? And they're the more biodiverse research parts of the planet. So the question is, how do we enable that and support that? And that's like a really key part to me. So, hey, are we overrunning? I can like just volunteer to get off the stage. All right. Okay.
Okay.
Thank you.
Thank you, Mike.
Yeah. Chat with me afterwards.
Yeah, I haven't figured the clock yet, so. Next talk will be by Mehul Sangegar on the new technologies of detecting blooms of mesoplanktonic jellies.
Hello, good morning. Okay?
Yes, go ahead.
All right, so good morning to all. I am Mehul Sangekar. I'm from India, but I have been in Japan for far too long. And I'm currently working in Jamstack. I'm actually not a biologist, so this is my first any jellyfish related conference. So my work, I'm actually from robotics, underwater robotics background. But I'm working with Dr. Duga Lindsay, and we are trying to do more new imaging techniques for jellyfish. And I realized that some of the work we have been doing might be applicable to studying jellyfish blooms. So yeah, listening to all the talks in this conference has got me a lot of new insight about how the jellyfish blooms can affect fisheries. I mean, I always knew about it, but some of the presentations have been quite phenomenal. Of course, I realized that large jellyfish are more easy to see, and there's work done on using drones and aerial mapping to see them. But what about really small ones? I mean, most of the work is done by taking samples, which is great, but can we complement it by using other imaging techniques, which can also help you to get some early warnings? So if you wanted to make a monitoring platform, how would you do it? What would you need? So I guess you would need a system which can be kept for a long time, and that would have to have low power. So some imaging technique which uses low power, you should be able to look at small objects. And if you really want to identify them properly, if you can see through them, through the internal structure, it will be even better. If you can get size, very nice. And if you can count them, count their density, even better. And then combine it to say, oh, there's going to be a jellyfish plume. Or then alert people on the land saying, you should go take your samples, or you should collect more data. So the things which we were developing in the lab were shadowgraphs. So I know shadowgraph technique itself is not a very new technique. It's been there for a long time, which, unlike a normal camera, it uses collimated light. So because the light is parallel, doesn't matter how far you are from the camera, you can always get the same size. Plus, because it's a backlight imaging, you can see through the animals. So we've already been developing some shadowgraphs in the lab, low-cost ones, and we have been trying different resolutions. So this is the same masticas, I don't know, I'm not biologist. I don't know the names. Sorry. But yeah, you can see it at two different resolutions, and you can see the details are quite different. But we developed two cameras right now which can record videos at 20 frames per second. And we tried them in Japan in collaboration with Tokai University. The two shadowgraphs, we put them on a frame, and to get where we were, GPS, and of course a CTD profiler, which can give you depth and the temperature, density, salinity, etc. And we deployed it off a small boat called the Hokuto to see what we get. And we realized, oh, we saw some very interesting things with the shadowgraph. Of course, we can see large objects, including jellyfish and other planktons, etc. But we could also see particle plumes. Like you could see dense particle layers, layers with no particles. We could see mixing layers where there's a density change. And that was quite interesting. So other than just looking at big jellyfish in the shadowgraphs, you could also get some indirect information. So we saw that, okay, if you take all these shadowgraphs and put them along a depth profile, you can actually know, oh, exactly where the mixing layer is, or at which depth which animals are, etc. But this was done manually. So you went through all the frames of the shadowgraph and then find out where everything is, and you put it along a depth profile. But you can't keep doing that. And especially if you're going to do automated monitoring, that's impossible. So then we started thinking, oh, okay, maybe it's time to start working on a more smarter pipeline to go through the data. And one of the idea was to have a quick look at the data and try to see what is in the frame. Use something which is very low computation to do this. And the idea is to use something like an autoencoder. And the idea is very roughly, videos use different compression for different frames. Something very complex gets compressed differently, whereas something which is not so complicated gets compressed differently. So we trained the autoencoder to look at different frames which have low particles, high particles, mixing layers, and big objects, and then made a model. So if a frame with any of these things came, it would extract features. And then we tried it on actual shadowgraph data. So the idea was the trained features which were extracted before in the model, the autoencoder would again extract features from the current frame and then try to match them and then put them and decide, oh, whether this frame is more closer to like a high particle frame or a low object frame or a mixing layer frame, or there's a big animal inside. So we could go through the video data more quickly and find out areas of interest, and then group the data into different zones. For example, the previous depth profile, we could now split into high particle areas on the top, the mixing area, mixing layer, and then the low particle round very quickly. And then based on the different layers, we can analyze the layer further. For example, the layer which has high particle, we can do things like particle count. So for that, we also established an algorithm where we started finding out ways to remove the background from shadowgraphs and detecting objects. Not very complicated, so they can not easy to run real time, but not too bad. And I don't know if it runs, but yeah, you can see on the left side is particles being detected real time, almost real time on a video. And if you have jellyfish, you can also see and track them. And of course, if you have big objects, then all the frames all the group which had big objects, what to do with them? We decided, okay, if you want to classify them, then we wanted to do a more machine learning based approach, and which also helps in ecology, or if you have an invasive species, etc., you can find it. So to start with, we are using YOLO 8, and we started making a training set for AI. We have just begun. We don't have so many images yet, but we started making training sets for Solmaris and Doliolitz just to see whether it will work on our shadowgraphs with the background, etc. And we have our own annotation tool in the lab, which is called Squiddle, where you can load a video and you can annotate it. And if you have metadata, like your depth profiles, etc., you can load it along with the video, and it keeps a track of everything. So we tried the models on one of our videos, and yes, we could actually, it could pull out solmars and doliolids from the videos. And if instead of putting it on the depth profile, we could put it on the time profile, thinking that if this was a underwater monitoring station, then as per time, you could see when the group changes. That means, I think I've made a mistake in the index, but you could see when there's a high particle area, there's a low particle area where there are no particles. And when there are high particles, you can see it can find you automatically doliolids and solmars. We only started with 2 now, but we are now expanding it to other animals as well. Yeah, and now what could you do with such a system? So if we could set up a monitoring system having multiple shadowgraphs, we already have 2 with 2 different resolutions, but you could make them based on what target you're looking for. And based on the patterns of the group which comes in front of the shadowgraphs, like you can predict whether it's a animal, whether it's high particles, dense particles, and then use that, use the CTD, and then guess if there's going to be a plume or an event happening, and then alert the station. I think that will be quite nice. You could also use the shadowgraph data as an input for other modeling techniques. Like I think I heard some of the presentations where you're using other techniques for guessing or predicting plumes. So you could use the shadowgraph as an input for those as well. And now we are trying to make more AR data sets. So we have set up like a tank setup in our lab where we are going to try to get more samples and make our own training data with shadowgraphs. Currently, there are a lot of groups working on data sets for visual animals, but not so much for shadowgraphs. So we would really like to work with people who are shadowgraph data. And right now, the autoencoder is made to work with only 3 types, which is the low particles, dense particles, mixing layers. But we realized that we could probably train it to find even more subtle changes, so we can split the shadowgraph dive into more smaller details. And yeah, thank you.
Thank you, Mehul. Always interesting to see how we can visualize the little goodies. Any question?
Hi, Mahal. That's really neat work. I'm just wondering what the accuracy of your classifications are for the really small things like doliolids.
For the YOLO?
Yes.
Yeah, I mean, right now we have just started with it. We realized that the orientation matters a lot. I mean, one of the things is, for example, with solmars, they look so different like this and like this, right? So that is tricky. I mean, if you do see them very clearly, it works very well. There's the other technique called morphoclustering, which I don't know if you have heard, but one of the other ideas, instead of calling it solmars, you call it solmars front and solmars top, actually make 2 separate groups. That might actually work. So that's another thing to try. And the other thing we're trying is just make a massive training data. We don't have that big training data to see how it would improve over time. So we just need more shadowgraph data, not just normal images.
Rebecca?
Thank you. Yeah, I agree. It's always interesting to see new ways to image organisms because we can't be in all of these places, even though we might want to be. Thinking a little outside the box, I've been trying to imagine a system that we might use to image things that are floating on the ocean's surface. Do you think a shadowgraph might actually be a reasonable approach for something like that? Maybe something below looking up?
That will be slightly difficult. I think one thing is because the shadowgraph does pick up mix-in water densities or temperatures. The light doesn't go parallel when there is a density change. I think the surface has a lot of mixture, so we will have to try that and see. And also looking up, because we need to have light on one side and the camera on the other. So unless we do like a 45-degree, they do exist, but we haven't really tried on the surface. I'm more worried about the density.
Okay. Thank you.
There's a question in front.
Thanks, that's really cool. Just wondering if you're doing or planning to do any ground truthing with like plankton samples, just to make sure that the shadow graphs are relating to what you're identifying in the shadow graphs is actually present in the area
Well,
you're
I
sampling.
think sampling is still necessary. I mean, there's stuff like DNA and things you can extract with samples as well. The only difference is like with the shadow graphs, because you see them in situ, you get slightly better morphology, because if you're using nets, you do damage them. But yeah, I mean, we always take samples. There's no excuse. I mean, there's no alternative. So this is complementary. That would be better, yeah, I think.
Thanks for your talk, Michael. I'm here.
Yeah.
I got yeah.
I was wondering, so you're also using aquariums to train these AI models, right? Or a bit, how much different is it from using it on the field? Is there a lot of variation in detection if you use aquarium-based and field-based training data?
I think we have just started, so we don't know, but they do tend to move to the corner or the bottom or the top or float. So it's not that easy to image them in a tank. You have to keep pushing them or generating flow in the water to be able to have it in focus or, in the frame of view. So I think you do affect their natural behavior.
Do you also use different water qualities that reflects what you see in the field?
Haven't tried it, but that's a very good idea.
Thank
Yes.
I got one question.
Yes.
How do you compare your system with the ones that are already on the market? Like the UV underwater video profiler, the HoloList, the LOPC?
Actually, they're pretty good as well. Just they're very expensive.
Okay, it's a homemade,
And-
cheaper version.
And I think we want to put more effort in the pipeline, which goes after collecting data. The thing is, if it is low cost and more people use it, there has to be a supporting pipeline which goes to actually process the data. Otherwise, we are just going to have data in hard disks and do nothing with it. So that has to go hand in hand if you do make the system low cost. So right now, a lot of these commercial systems come with their software, which works, but that means not many people can use them. So
Okay, thank you very much, Mehul. Okay. Okay. Okay, just a reminder, there's a parallel event. It's in the mini conference hall above the restaurant. And the theme is the jellyfish fisheries and trade, status, trends, and impacts on livelihood. So if you want to join the other session, please do. If you want to stay, please stay.
Please stay.
Okay. Sorry, Johan. Nobody is running away yet. So Johan Soto-Angel is going to tell us about bipolar bipolar jellyfish, and evolutionary origin and diversification.
Thank you. Probably most of you, or at least some of you that have heard the term bipolarity or bipolar in the last years, it's definitely something different to what I'm gonna talk about. So apologies if you were expecting me talking about bipolar disorders in brainless animals. That's not gonna be the case. But this is a very interesting topic as well. So thanks for not going to the parallel session. I'm John Soto. I work with polar jellyfish. I'm very interested in the ones that are bipolar. And what is bipolarity? Bipolarity is basically the presence of the same species or related species in both polar regions. This phenomenon, this biogeographic phenomenon, is widely spread in the tree of life. It happens from various to whales. There we have animals like the fulmars, they are bipolar. There's one species in the northern hemisphere, another species in the southern hemisphere. The right whales, the Cleone, the theropods are bipolar as well, and we have examples of jellyfish. For example, Periphylla is an example of a bipolar jellyfish. if we track back the history of bipolarity, we have to go as early as nearly 200 years ago, where it already interested a lot of the scientific community. And James Clark Ross, he was by no means a biologist, but he was very interested in trying to figure out the connections between species that were very similar in both polar and subpolar regions. So he did many, many observations during his trips. Then Darwin, on the origin of the species, he already noted that some plants that inhabit in temperate areas, they can be also found in tropical areas, but at way higher altitudes, so up in the mountains. And related with this, but instead of altitude going to deep waters, Berg realized that some temperate species can also be found in tropical waters, but only very, very, very deep. So we have these species that are found primarily in shallow or relatively deep waters in both hemispheres, but can also be found in very deep waters. And this is another type of bipolarity. Graham, he's a biogeographer and a paleontologist at British Antarctic Survey. He has published a lot on mollusks, especially based on the fossil record, and he has been able to define divergence time based on the fossil record for many mollusk lineages. So he was actually able to track the origin of this split of this diversification using the fossil record. But still, for many groups, there is a lot of gaps in the knowledge because there is not a lot of molecular data supporting this evidence. So we have a snapshot. We know that bipolar distributions occur. We know that there is one species in the north, another in the south, but we don't know how and when these distributions appear, which is the origin of this peculiar distribution. So Alcon and Griffiths basically summarize all the literature on the topic, and they basically provide two hypotheses. Either it's through vicariance, in which we are in a glacial period, we have a cosmopolitan, widely distributed species, and then the warmer it gets, this species basically splits and goes northern and southern, and then we have a barrier that the species is not able to keep gene flow, and then it speciates, and we have basically two species, one in the northern hemisphere and another one in the southern. The other hypothesis is one species that it's distributed only in one of the hemispheres. We are in an interglacial period, so we are in a warm environment as we are now, and then the globe gets colder and colder and colder, and this northern or southern species is able to jump the equator, and then through founder effect, it's able to establish itself in the other hemisphere, and then we have the same, right? So we have two potential origins. There are more hypotheses, but broadly we have these two potential explanations. But again, it's a matter of how and when. So we don't know, and for this we need to include either fossil record, molecules, or preferably both. We are lucky enough to have had Sofia Stepan-Jans within the Cnidaria Medusozoa Hydrozoa community. She has worked a lot on bipolar. She was super interested in bipolarity in Medusozoa and Hydrozoa particularly. And she identified 32 genera and 23 species that sensu lato could be considered bipolar. There's more genera than species because some genus can be considered bipolar, even though not all the species within the genus are bipolar. So she found several species, and she also suggested that there are some genera that could also be relict of ancient times, that they have been kept in polar regions, but they were a wider group in previous geological times. Recently, we reviewed the existing information about it, and we narrow it down to only the species that are strictly bipolar, because they are only in the poles, and most of them are absent anywhere else in the world. And but we are still lacking molecular data for most of them. So the question, at least for Hydrozoa, is still how and when these distributions arose. And here's when our project Pole2Pole came into the game. It's a project funded by European Commission under the program Marie Curie within the Horizon 2020. And we want to use Hydrozoa and their wider range of life cycle strategies to answer the question whether life cycle strategies have something to do with the time of diversification. Our hypothesis is that for those species that have highly dispersal capabilities, they should have kept somehow gene flow, and they should have an earlier split. So they should have kept gene flow not so long ago. And then for those species with very limited dispersal capabilities, such as benthic hydroids without a medusa stage, this split should have happened earlier in the time history. So we are going to the polar regions, collecting many samples. We are revisiting the species delimitation. We are working with both Sanger sequencing and next generation sequencing to see how many lineages we have. It is a matter of how many species do we have in there. Is there cryptic speciation in there? Are those species the same? And then the final goal is to be able to determine how and when this split occurred using calibrated trees. These are the models we are working with. We have selected them because they represent very well the different life cycle strategies. So on one hand, we have Buyonia. It's a benthic polyp. It doesn't have medusa stage. And it has, it's very peculiar because it self-recruits. The polyps are shedding the larvae into the water, and basically they settle a few meters away from the mother polyp. This is known for the species. So in principle, it's an example of very restricted dispersal capabilities. Then we have a benthic hydromedusa, a Trachymedusa Tychogastria polaris, which has been described from both poles. Then we have a group of species that have both polyp and medusa stage, the group Tychogyna stegopoma, I will explain later. And then we have a representative of an holoplanktonic life cycle, Dimophies arctica, that, as the name says, it was reported originally from the Arctic, but we found it in the Antarctic as well. So I will briefly tell you a bit more about its case, and whenever we have results, I will show them to you. There's not much we can say about Buyonia. So far, we have some sequences, and it seems that it has a very peculiar position within the phylogenetic group it belongs. It's a tubularide, and we don't have much data about it, but it really seems it's like the sister group of all the other tubularidae. If this is the case, it could confirm the hypothesis given by Stepan Jans that it could actually be a relict group from polar regions. It is a species that you find very often on moors and moorings in very deep water. So if you ever see it, just give me a call because it's a very interesting species. Then we jump into the benthic hydromedusa. This is Tychogastria. There is 3 species of Tychogastria considered valid today. There were a bit more, but they have been synonymized with Tychogastria polaris. The species was originally described from the Arctic, but it's known in the Antarctic waters. And we don't know much about this jellyfish. We know that it can create blooms as well, which is strange for a tiny benthic hydromedusa, but they do. There is a publication in 2017 that they observed huge numbers in the Antarctica, and they were not all sitting on the bottom. They were swimming in the water column. So it seems that it's a species that it's not as it's hyperbenthic still, but it's swimming a lot. And we have already seen that we only in the northern hemisphere we have 2-3 different clades. So we are working on it at the moment, but there is definitely cryptic speciation in there. We were lucky enough to be on board of the Ocean X some months ago. You can see how happy Aino was to go on the submarine. And actually in one of the dives, that was my dive, it was meant to be, we saw dozens and dozens of Tychogastria CF polaris. We have some evidence that it's probably an undescribed species from the North Sea, and because we've seen that it's not the same as the Arctic one. So we captured a lot of them. We put them in chrysals. We took great pictures because, of course, after fixation, they are not going to look that great. And we are working on it, but it looks like a promising finding. Then Dimophies arctica. The 2 on the left are from the Arctic. The one on the right is from the Antarctic. Not many morphological difference we can ascribe so far, but it's a tricky species to barcode. There must be some issues with the primers we are currently using. So we aim to sequence the whole mitogenome supported by the Artsdatabanken project NOAA, Norwegian Arctic Hydrozoa. We recently got funding. And yeah, it's our example of holoplanktonic species. So we are crossing our fingers that they will not diverge so much. So it could be potentially the first example of a true bipolar species. We'll see what the molecules say. And last but not least, we have the case of Stegopoma and Tycochina. For those that have been working on taxonomy, this is the perfect nightmare. And I will explain you why briefly. In 2017, Schuchert and collaborators, Aino included, they found out that the polyp of Stegopoma plicatile is actually the polyp stage of the jellyfish Tycochina crocea. What happens? Stegopoma plicatile is known worldwide. It's basically everywhere in the world, from Indonesia, Antarctica, North Sea, Arctic Ocean, basically everywhere. But Tycochina crocea has a very restricted distribution. So we found the paradigm of a widely distributed polyp with a very restricted jellyfish. And because the jellyfish is not found everywhere, then there might be some cryptic speciation there. And it's a nice example of selection acting in only one of the life stages, but not in the polyp, at least with the characters we can recognize so far. Another nightmare, Stegopoma plicatile is the type species of Stegopoma, but Tycochina crocea is not the type species of Tycochina. Another nightmare is they are currently located in two different families. So it's one species located in two different families. It's like having a butterfly in one family and what do you call it? The worm of the butterfly. Yeah, a caterpillar in a complete different family, right? So that's something we have to solve. And then actually the type material of the species comes from 4 different localities, 2 in the North Sea and 2 in the Arctic Ocean. So that makes the topic even more tricky. What we know so far is that, and as I said, if you check worms nowadays, Stegopoma plicatile is considered an accepted species, and it's synonymized with Tycochina crocea, which is not completely true, because if we keep calling it Tycochina crocea, we will be citing a North Sea species everywhere in the world, and we know that that is far from true. So now we know that other Tycochina species, particularly Tycochina lactea, they have an identical polyp, or nearly identical polyp than Tycochina crocea. And other Tycochina species... Whoa, sorry. Hi? Oops. Can you hear me? Yeah. Other Tycochina species, such as Tycochina hyperborea, it has absolutely nothing to do with Tycochina. It's not a Tycochina. It doesn't even belong to the family, right? So as you can see in these results, this is for Bayesian CO1, but we found the same with maximum likelihood, and both for 16S and CO1. You can see that Tycochina lactea has a polyp that is identical to Stegopoma plicatile, but Tycochina hyperborea in the lower part, it's not a Tycochina at all. So we think we found the polyp for Tycochina hyperborea as well, but it's definitely another thing. So take-home messages is that we still have a lot to know about this jellyfish, that we still have to do a better work at studying jellyfish that are not large, they are not conspicuous, because they are also part of the diversity. They also have a key role. They also bloom. Tiny species also bloom, and we really have to do an effort on this. It looks like bipolarity at the species level is probably unlikely, but we are still seeing with Dimophias what we get in there. And there's definitely a lot of cryptic diversity, but this is our challenge. We care about the identity of the species. I don't need to justify why, because Alan Collins and Mike Dawson already explained why do we really need to make an effort into putting a name or at least a code to the evolutionary lineages that we have, because that have consequences at many levels. And life cycle wise, we have a mess there. We really have to make an effort into try to get this. And if I get 30 more seconds, just a final consideration. On the left-hand side, you have the diversity of gelatinous zooplankton. So most of the gelatinous zooplankton are basically hydrozoa, non-siphonal hydrozoa. And on the right side, we have the tox that we have in the jellyfish bloom symposium, or better called the Scyphozoan bloom symposium, right? So I think there is a lot of species we know nearly nothing about, just the fact that they exist, or for many species we still don't even know if they exist. So I think as a community, we really have to center our efforts in underrepresented groups. And with this, thanks to the funding agencies, my collaborators, and thank you for staying.
Thanks, Johan. Cool stuff. Any question?
Thank you. That was very interesting. So now that you have your results, and especially in light of Mike's talk about how if we don't have enough characters, it can be hard to distinguish species. Are you more skeptical about the idea of sort of bipolar species? Do you think a lot of this could be convergence? Or do you think it's just a matter of maybe not having enough information to resolve?
Briefly, I think a lot of Antarctic species were basically named using Arctic guides. So people go into the Antarctic for the first time, they were saying, okay, polar region cold, let's bring the Arctic guides. And they were naming them using the Arctic names that they were available, right? So for most cases, I think they are different species, definitely. And there could be some convergence as we know it happens for other groups. But the fact that they are different species doesn't make the case less interesting because they are still the closest relative. They share an ancestry, right? And that is still bipolarity. So we have to consider that this is there's no two sister species that can be farther away in the world. They are in the poles, right? So it's a very interesting phenomenon in that sense, even if it's not at species level.
Hi, sure. Can you speak a little bit about the differences between antitropicality, bipolarity, and cosmopolitanism? It seems like there's some overlap between these things, and maybe there's some information in that.
Yeah. Stepan Jens described basically two types of bipolarity. The ones that they are definitely absent from tropical regions, and the ones that they basically are still connected through very deep waters. So for some species that could be the case, but it's a matter of sampling effort, right? Like we don't really know everything. I'm still wondering, do we find Dimophias at 10 degrees north, 2,000 meters? That would be a cool finding, but yeah. So those are the, let's say, two main types of bipolarity. And the other description is what it's been called amphitropical for those species that are not in the poles, but they are in subpolar regions. So they are definitely absent again from the tropical areas, but they are present in both hemispheres. But again, we have examples within birds, whales, so it's not just a case of one particular group, but.
One last question.
With Tychogastria polaris, one of your main species, looking at comparing specimen, it has a lot of different characters compared to many hydrozoan medusae to look at, right? But comparing Antarctic specimens and caught with a core sampler, so very good condition, and Japan Sea, Northern Pacific Tychogastria caught with an ROV, could not find any morphological difference between them and very high genetic divergence. So I wonder if you have any thoughts about evolution of characters or like selection pressures or other things like this, that why?
Yeah, we have a master student working on that. He's working, focusing on the North Hemisphere specimen, because just focusing on the Northern Hemisphere, we have a lot to play with. But for now, other than color, that's the only character we can attribute so far, coloration patterns. But not much we can say at the moment. But we have very nice formalin preserved specimens from different parts of the world. So hopefully I will be able to answer that question soon.
Thanks, Johan. Next speaker is Saravanan Raju, and the talk will be about mapping and monitoring recurrent jellyfish blooms in Indian coastal waters, known and unknown.
Good morning to you all. And today I would like to present my work of our institute, mapping and monitoring recurrent jellyfish blooms in Indian coastal waters, known and unknown. And coincidentally, my topic falls in the lines of Alan Collins' known, unknown, known, unknown, and unknown, unknown style. And as a Thanksgiving mentioned, so I would like to thank my organization, Central Marine Fisheries Research Institute, first director H.S. Rao, who was working on Siphomidaceae and described 2 species, which still supports a vibrant fishery in India. And many of you may not know about my institution, so just a brief on that. And we are a national Marine Fisheries Research Institute and covering nearly 8,000 kilometer coastal line in 9 states. And it nearly involved 2 million square kilometer of EEZ and 500,000 kilometer square of continental shelf and 1.25 million hectares of brackish water. So jellyfish is everywhere in these water. So our responsibility is very high. And we started actually a brief history about it. In 2010, I was asked to work on jellyfish, and in my institute, I was working for nearly 6 years alone on jellyfish. And in 2017, a national dedicated monitoring project was started in our institute. And for the first 3 years up to 2020, we made initial capacity building, and we established nearly 74 jellyfish focal points where recurrent jellyfish swarming happens around the Indian coastline. And these places, what are all the different jellyfishes are swarming and how year after year that varies along with the abiotic factors, it is monitored. And this is the baseline which we established by 2020. And based on that, we have categorized our 9 maritime states, starting from Gujarat. Is it visible inside? Actually, here or on this side, actually the pointer is not working. Is it working? The Gujarat, where you see the left side, to my left side, 6 numbers of Cyphozoan and Cubozoan. That means including only one Cubozoan is dominant in nature in Indian waters. Is it visible to you? Is it visible? No. So, okay, I will go ahead. Maharashtra, the next 5 numbers of Cyphozoan and Cubozoan, that is only 1 out of 5, and Cubozoan. In Karnataka, 8 numbers, and down south in Kerala, where now the Symposium is going on, 10 numbers. And the other side on the east coast of India, 15 numbers. And Andhra Pradesh, it is 11 number, Odisha, 6 numbers, and West Bengal. So 9 maritime states we are monitoring. We have nearly 15 scientists are based in 9 maritime states to look after these jellyfish swarms in our Indian waters. And based on this in a pilot mode, because the Gujarat, which is located on the this side, you see in the Indian map, to my left, the Gujarat state. So that is the longest coastal line. So we made a pilot to geo-reference jellyfish blooms. So this is what the output. So similar manner for each states, we are planning to geo-reference the jellyfish blooms to create a data set in the coming years. That work is already started from 2020. It's going on. This is the pilot of Gujarat state, where an active. This is okay. No. Yeah. So this is the longest coastline in Indian province. So here there is an active jellyfish fishery of Ketostylus spiracy and Rhopalema hispidum. And this species-wise, this geo-referencing has been done for the Gujarat coast. And here in Indian, you must have seen some of our Indian presentations. Finally, definitely there will be variations and there won't be much uniformity in a swarming pattern, even to comprehend that it is a bit difficult. The reason is that, so if you look at that Indian coastline, this is Indian west coast and Indian east coast. This is from during the southwest, because this is a monsoon country influenced by 2 monsoons, southwest and northeast monsoon. And during the southwest monsoon, the West India coastal currents that comes from this northern Arabian Sea and flow through the entire Sri Lanka and pass up to this Bay of Bengal region. So the flow will be during this period, especially June to September, it will be like this. So water from the northern Arabian Sea will be going to the Bay of Bengal up to this West Bengal state. So the flow is like this. it carries a high saline water. So characteristically, the bloom species differs based on this circulations. Whereas in the northeast monsoon, the water currents reversal. The East India coastal currents flow from this Arabian Bay of Bengal towards the Arabian Sea. This is low saline in character. So because of these two reasons, highly influenced by these West India coastal currents and East India coastal currents, really the swarming pattern and the distribution, spatial distribution and all highly varies, and introduction of species from the Arabian Sea is happening in the Bay of Bengal. Mostly as per our observations, we see that the route of introduction from west to east in India. And we have a number of coastal ecosystem around 8,000 kilometer of coastal line. So I am giving a small area that is called Park Bay in the southeastern coast. So these two different ecosystem, Park Bay, a shallow bay, and Gulf of Mannar, which is a coral reef ecosystem. So here, during this 2017 to 2020, the SST and salinity variation was observed. And you can see the SST variation somewhere from 28 degree, it varies up to 33 degree during this period. So nearly 5 degree temperature variation can happen in SST. Whereas in the salinity, somewhere during in 2020 up to 28 ppt, there was a heavy monsoon during that time. From this up to 35 ppt, and salinity variation is there. And normally here above 34 ppt salinity only there is a bloom in these waters, especially in Park Bay waters. Temperature as it should be above 33. In this case only we get a huge swarm. And by comparing all the entire Indian coastline, we can summarize the swarming season in Indian coastal water something like this. Actually, the monsoon is the main thing. So before monsoon and after monsoon, like that only the major and minor swarm happens. In East Coast, the minor swarm in September, October, and major swarm during Indian summer, March, June. Whereas in the West Coast, Kerala, the pre-monsoon swarm is major and post-monsoon swarm is minor. Whereas in other states in the West Coast, Karnataka and Goa and Maharashtra and as well as in Gujarat, there is a slight variation in swarming season, September to January and November to May. And even now in Gujarat, there is an active jellyfish fishery is going on. The winter bloom is there in Gujarat coast. And by far the most prevalent jellyfish genus we can call in Indian water Chrysiora. This is prevalent in both the east and west coast. And as far as fishery supporting species is concerned, only 4 species are supporting fishery. The Crumbionella annandale restricted to only along the east coast of India. In Orissa and Andhra Pradesh, in 2 states, there is an active fishery. And arseny is restricted to Kerala waters. And Rhopalema hispidum and Ketostylus ferresi is restricted to Gujarat coast. So this is how the fishery supporting species are distributed along the Indian coastline. And here, this is the trend observed in 2022. So this is summarizing all these species found in state-wise. And here in Tamil Nadu, these 2 important ecosystem, Park Bay and Gulf of Mannar. So maybe in futures, we may have to look at the Indian coastline ecosystem-wise. That only it will give a meaningful conclusion. Even the state-wise categorization also very difficult to interpret. So by these studies since 2017, we have listed out the major bloom forming jellyfish species in India like this. All Crumbionella only there are 2 species, Annandale and arseny. And Pelagia noctiluca found only in the east coast in a major swarming condition. And Chrysiora, as I told you, is the most dominant species in Indian waters. And Ketostylus ferresi is a fishery supporting species. Acrometus flagellatus is found along the both coast. And Cyanea, I think there are two species, Nosaki and Lamaki is found in Indian waters. And Lycnorhiza malayensis, Netrostoma cirulensis is the dominant species along the west coast, especially even in the Kerala waters, swam in a big way. And Rhopalema hispidum, again an edible jellyfish. Marividia stellata has some introduction in east coast of India. Earlier, this was restricted to only the west coast of India. Now we could get some samples from the east coast also. And the Sepa is there only in the west coast. Lobonomydus robustus restricted to east coast, and the only jellyfish, box jellyfish, which is found in blooming condition is Chiropsidus buttonski, and that is restricted to the west coast of India only. So this is the major bloom forming box jellyfish in Indian water. And when we compare, actually this is a paper which we have recently brought in 2023. The Netrostoma cirulensis, which was only found in the west coast, is now available somewhere here along this coast, Odisha coast. So earlier, before 2023 and all, this was not found along this coast. So as I told you, the Western India coastal currents and Eastern India coastal currents play a major role in the distribution of species along the Indian coast, especially along the east coast of India. And here, this is the major species which you can find in the west and east in swarming nature. So there is a variations you can see. The arseny is restricted only to the west coast, and Annandale is restricted only to the east coast of India. And Ketostylus parisi yet to be found along the east coast of India. And then Acrometus flagellatus swarming in the low saline areas, especially in the estuarine and backwater areas of India. And Lycnorhiza malayensis is also very much restricted to west coast of India. And Netrostoma cirulensis has been found along the west coast. So we have brought out a work on that. And Chrysiora, as I told you, the most dominant, has pan-Indian presence. And this is the Chiropsidus buttonski is the only box jellyfish found in the blooming condition, especially in the west coast. And here along the east coast, we get Alatina during the season, here especially in the Tamil Nadu coast. This particular season, especially the last 2 months, more than 200 cases of fishermen has had to be treated for these stings, envenomation cases. So only along the, actually you must have heard in the earlier presentation yesterday, many of the west coast, they were not having much practice of managing envenomation, but whereas along the east coast, there is a severe envenomation is going on due to box jellyfish stings. So there is a need to manage this sting cases in a better manner, especially box jellyfish. And one another species which has been described here, the Acrometus rabanchatu. From this area, there is a lake, a brackish water lake, Chilka Lake, but not much work, nothing has been done, and no other work has been done. So Acrometus flagellatus only described from Indian waters, and Acrometus maculosus and rabanchatu has been very well described from Indian waters, but unknown category. And here again, this Crumbunella annandale, which is described by our the first director and is a fishery supporting species, and this has been considered by the international community as Nomen dubium for long, and we described redescribed this species again with the help of one of our associates from South Africa, and so we published it, and the Crumbunella annandale and the Crumbunella arseni is this overlapping areas like this, and here normally you have found Crumbunella arseni here in a chocolate in color. We have recently collected some bluish color arseni also in recent collections. And some molecular work also has been done during this time, and we have found in Gulf of Mannar water, especially in the east coast of India, Cassiopeia zamacana, and whereas on the west coast, Cassiopeia andromeda has been recorded. And here another unknown is that I am working from somewhere here, and these waters Lobonemades robustus is found, and my Sri Lankan counterpart tells me that Lobonemades gracilis is found here, and some of the earlier works states that Lobonemades gracilis is suspected to be the juvenile form of Lobonemades robustus. So these things has to be worked out in the future. And similarly, the third species of Lobonemades civelli, which is available very much holotype in our Zoological Survey of India, which was collected by Ray H.S. Rao, our first director from Andaman Sea. It is adjacent to Myanmar and from this area, but no description, no work, nothing has been done so far. So one another work is pending for a long in India. And as far as DNA barcoding work is concerned, 3 different color type of Crassiora has been found in east coast of India, and we tried molecular work, and only with this particular pattern, which gives good results, and it turns out to be Crassiora chinensis. These two are not successfully. We are failing in getting result, so work is on. And here from Kerala waters again, the Crassiora species molecular work has been done, but my associates tells me that the species level confirmation is a bit difficult with what is available along the Kerala coast. So as far as Crassiora is concerned, it is found to swarm both the coast, east and west coast. Still, species problem exists. Only one species, Crassiora chinensis, can only be confirmed. And another species which has been described as Crassiora calipariae from east coast of India, but many South Asian workers are conferring or using this particular species name, but no work, no molecular data, and even Morandini has tried to describe this in his monograph, but still this needs to be known. And Acrometus flagellatus work as far as Kerala water is concerned has been carried out by one of my associates, and these two color patterns, the blue and brown, both falls into this Acrometus flagellatus type only. And whereas Catostylus species, again the species level confirmation needs further analysis. And whereas recent phenomena is Noctiluca scintillans bloom comes in a big way. Earlier it was following a 10 years cycle. Now since 2019, year after year, it comes in a big way. Here this is Gulf of Mannar, this is Park Bay. I am working from somewhere here. You can see the green, and there is an interplay between jellyfish bloom and Noctiluca scintillans, like that many works are there. And we really do not know how this Noctiluca blooms, which leads to hypoxic condition in the seas, helps jellyfish blooms. So with all this unknown, actually many people now work in India also on jellyfish. This has got attention of many. And similarly, an Indian cartoonist is also attracted to jellyfish bloom, and he has come out of a series of jellyfish cartoons. With that, I conclude my presentation. Thank you.
Thank you, Saravanan. Just to keep the time, if you have questions, I will ask you to come forward after during the tea break. Okay? So the next presentation will be done by Florian Lusko. And Florian is going to speak about freshwater jellyfish research, hidden gems, knowledge gaps, and current progress.
Hello, everyone. Yes. So, first of all, I want to say it is a pleasure speaking to you today about a topic that is close and very important for me, and not only to me, but for a whole bunch of people. Some of them are actually sitting today in the audience, and I must say that this work has been the product of a year-long effort with scientists involved from 4 different continents. And before I start, I need to acknowledge that the work presented today has been conducted mostly in the ancestral, traditional, and unceded territory of the Musqueam people in British Columbia in Canada. And if anybody Googles jellyfish blooms, looks for nice pictures, of course, you would expect this very colorful, bright, shiny pictures of scyphozoan and hydrozoan blooms from the coasts, sometimes from the open ocean. But what you would never find is something from a lake or pond or river. And honestly, most people don't even know about freshwater jellyfish, and I'm sure not even all of you knew before this conference about it. But I'm actually very happy to see that 3 contributions in this symposium actually address this. It was the very nice talk by Sergio 2 days ago. It is now my talk, and there's a poster later on today also addressing freshwater jellyfish blooms. But before I go ahead, we need to kind of like set the stage what freshwater jellyfish are. And it's a very small group of hydrozoans that are medusa bodying, and they need to be separated from hydra and other non-bodying hydrozoans in limbic systems. As I said, it's a small group of organisms, 30 to 40 species about, and they occur globally and temperate to tropical habitats. And they mainly follow a typical metagenetic life cycle with a pelagic and a benthic phase. There's some shortcomings and there's some unknowns, but in general, this is what we believe is true for all of these. And when I talk about freshwater jellyfish, I concentrate on the 3 main genera, Astrohydra, Craspedacusta, and Limnocnida. There's more, which I'm not going to address today. And some of these species actually became invasive, namely Craspedacusta sovabi. And well, we have seen the symposium that we know quite a lot already about marine jellyfish, and we can't stop talking about all the limitations which doubtless are around. We know very, very little for the freshwater part. So, and I'm going to address some of the reasons why we know so little and why this is an actual problem. And so the reason is, why should we learn more about the freshwater jellyfish? Well, with climate change, we see that they are expanding their distributions. I will show you Meg in a minute. So we need to start caring soon before it's too late. And here are some more nice pictures of different species and life cycle stages from all across the world. But what is most impressive is actually the 2 bottom doesn't really work well. The 2 bottom pictures of blooms. The left one is coming from a lake in Canada, and then the right one is coming from Lake Tanganyika in Zambia. Different species, but same appearance. So one might ask, okay, when I say we have a hampered progress of knowledge gain in jellyfish, well, how does it really compare with marine jellyfish? What are the reasons for this hampered progress? And so if I say, well, it becomes an issue, where do we go next? What are the gaps we need to address? And very briefly, I want to give you an overview about the methods that we use to compile this massive data set. So we started with a literature search to see what is actually known already, classically with the Web of Science and Google Scholar, and used a couple of selection criteria, which I don't want to go into much detail now. But we very soon realized there's tons of articles that all our collaborators knew already, which weren't picked up by these search engines. So we decided to make a more exhausting by hand approach and came up with about 700 literature pieces in the end. And just to give you an impression how most of this year looked for me and several of my co-authors, so I went to many, many different libraries and looked up books that has been sitting in the shelves collecting dust for 100 years. Nobody looked them up. And even more exotic is the use of microfilms. I'm sure some of the older in the audience still know what microfilms are. I just learned to use them this year. All right, so we want to see how freshwater jellyfish research compares with marine jellies. And for that, I picked the best studied, doesn't really show up, it's too bad, the best studied freshwater jellyfish genus Craspedacusta, and compare the succession of research since 1880 to 2022 with that of Aquaria, the probably best studied marine hydrozoan genus, and then the best studied freshwater hydrozoan genus Hydra. And well, the picture is quite different. So we see this near exponential number of publication compiling for Hydra because of obvious reasons, and we see a very different picture for Aquaria, that basically until the late 90s, nothing so much really happened with the genus just after the discovery of the green fluorescent protein that we see this bump in the number of publications. But of course, it's very, very obvious that we know very little about Crosspedacusta, which is already the best known genus. All right, so what are the reasons for the hampered progress? And I just want to quickly walk you through the design of this plot in the bottom right corner. So we have the decade on the right, on the x-axis starting in the 1880s, and then the number of publications per year on the y-axis, and then color-coded in this example are the life cycle stages in question, frustules, medusae, and the polyps. And then on the right-hand side, the box plot basically compares the number of publications of the dominant life cycle stage, so the medusa, with the sum of all others. All right. But coming back to the reasons that basically hampered the progress, it is the misspelling and the change of species names. And this is not a very specific problem for freshwater jellyfish, but it occurred especially abundant in this. So it happened very often in the late 19th century that the polyp and the medusae stages were described separately, and people continued working with these different species in quotation marks, for years and decades until eventually they figured out, okay, the life cycle brings both species together and they needed to redescribe this. So there's a general impression of uncertain taxonomic entities. And of course, this is also not a problem for freshwater jellyfish only, but we see that the medusa stage is considerably better studied than everything else combined. So about 80% of the literature available on freshwater jellyfish is on the medusa stage because of obvious reasons. Well, the jellyfish is what the people usually find and has a potential harm for humans. It is also abundant in the literature that the research is very much biased towards one genus, Crosspedacusta. You can see it in the dark gray colors. And this makes very much sense because where does this species or the genus usually occur? In temperate to subtropical regions, primarily in the northern hemisphere, and then in the countries of the global north, where actually people have enough resources to make research and publish this research. And it's also happened that this is the area on the globe, same on this, in the southern hemisphere, where you have the highest human population. So there's actually people around to make reports of sightings. And of course, stuff like geopolitical complications, wars, or objectively difficult areas make research on tropical species, especially in Africa, very difficult, that hampered the Limnocnida progress. And then another aspect that is not so much obvious for the marine jellyfish literature is the diversity in languages. While marine jellyfish research is most often in international waters, so it's obvious to use English as a communication language, this is not so apparent in regional scale and often national freshwater bodies, because the audience is most often a national dimension, so you publish in a national language. And if you look over the last 150 years, it's clear that in the first few decades, until about the 1920s, the colonial languages like English, French, German, and to a smaller degree, Japanese were dominant. But we see also a very strong diversification in languages in then the first half of the 20th century, and especially after the dawn of the 21st century, two languages become dominant, it's English and Chinese. So for understanding freshwater jellyfish literature, it's important to not only read the English literature, but also to include other languages or literature published in other languages. And then there's, of course, a colonial economic crisis and global war signature in this general pattern that you see. Well, as I mentioned already, in the first 4 or 5 decades, we have a very strong colonial fingerprint basically on who conducted this kind of research. It was, of course, mostly the North Americans and the Europeans who had sufficient funding and who were the colonial nations to make this kind of research, to go on expensive long-distance travels, to collect all these data and eventually publish them in journals that are still accessible today. So publishing is not only the problem, it needs to be findable today. And so in the 1930s, we see a very strong diversification of people doing this kind of research on all continents, and then a very strong regionalization after the Second World War, which again hangs then together with the end of the golden 20s. Oh, sorry, now I mixed it up a little bit. Well, the golden 20s happened before the Second World War, apparently. Excuse me. So, but anyway, you see this in the 1930s, this very high output of papers, which then plummeted in the 1940s related to World War II activities, and then this uprising again in the 1950s, which is of course related to an increase in funding for fundamental and applied research, not for freshwater jellyfish only, but in a global perspective because of a nuclear potential threat. And one aspect that we shouldn't underestimate is the complex between science and military. In many marine sciences, we as scientists piggyback on marine military vessels like the Coast Guard or Navy, which facilitates our research as an expensive platform and waives a lot of our expenses. But nothing like this exists in smaller freshwater, often within national boundary water bodies. So this kind of research faces very strong financial challenges. And then lastly, there's a lack of theme-led journals where actually freshwater jellyfish research is published. And I summarized all these literature and found that only 15% are published in 5 primary outlets. Well, there's Science and Nature among those. Obviously, very few people can publish freshwater jellyfish research these days in these journals, but this is abundant in the early decades. And again, this strongly contrasts with marine jellyfish research, where we have well-known journals. And all of this contributes then to the impression, well, there's little progress, there's little need, so why should we care? And the reason why we should care is actually these 2 maps. On the left-hand side, you see the present day. On the case, and the right-hand side, you see the 2050 case, and it's a species distribution model for the most abundant and west-studied species, Crustbeta costasovabi. And what I can basically say as one just sentence, that over the course of climate change, this species and potentially many other freshwater species will expand the distribution. So whatever they do, we need to start caring and understanding before we actually can start managing them. And because I'm running short in time, and this is a very extensive list of things that we actually don't know, and I'm sure every one of you will find some of these aspects reflected in their own work, well, this is the open field. Feel free to join in. It's basically each of these points has 1 or 2 papers historically, and we know literally very little about where they are, what they do, how do they distribute, how can they be managed, what effects they have on fish, tourism, how they interact with other food web components, and so on and so on and so on. All of this is detailed in the associated paper, and I can't go through this all right now. But what I want to say at the end of my talk is that all this literature has been summarized in this Pangea data set that is also freely available already, so you don't need to start from scratch again. All of this information has been summarized, is accessible now. And as a very quick take-home slide, I know it's a lot of things to take in, 150 years compressed in 12 minutes. It's hard, but our present perception is governed by one genus only. There's more. All of these different genera and species do different things, which we have very little idea about. If the researcher is only reading English literature, it's apparently missing about half of the literature available, so it's important to group up in international teams. And we need especially to put forward the knowledge gain in Africa, Oceania, and South America in collaborative efforts, because these are the areas where we know the least. Thank you for your attention.
Thank you, Florian. They're sweet little bugs. I'm going to ask a question myself. And from what I understood, I thought some of the species we got in Europe, in France, for example, were coming from Asia through something dealing with the World War. Is that correct? So if the water is actually warming, is that going to increase the presence of jellyfish, or is that going to be going the other way because they come from cold water regions?
Yeah, well, most of these species, if not all, are subtropical or tropical origin, so they're actually expanding to the higher latitudes. And so eventually the actually tropical species and populations might encounter any issues with their maintenance, but for the invaded areas, I don't see any problems. They are thriving and they will continue.
Thank you. Any question?
A nice presentation. I have a small question. In scientific perspective, we think that origin of all creation happened from the marine, right? But in terms of in these closed bodies, how come these jellies, the origin?
So I just repeat the question if I understood it right.
Yeah.
So how is it possible that in these small closed water bodies we see these jellyfish?
Yeah.
Yeah, that's an excellent question. I don't have, unfortunately, a definite answer. There's lots of ideas about this, and so bird migration and then the carrying of polyps, protocysts is suggested to be the major vector for this dispersal. Could be also the restocking of like local water bodies with fish, and that there could be something in the water, aquaria trade, on growth of the boat hulls. So the list is literally endless, but there's nothing of this proven yet.
Hello. Yes. Thank you for your talk and looking forward for the review. That must be so much work. I had a question in terms of genetic connectivity, because coming from the marine world, obviously there's not as much barriers compared to the freshwater. So do you observe like a species per every lake, or is there connectivity? How?
Yeah, it's another excellent question. So there's lots of things to address in that, but the first thing is that most of the populations in invaded areas are actually of one sex only. So sexual reproduction is doomed to fail. So literally in all these lakes that are sampled in British Columbia, Canada, they are clones. They are genetically identical. And what Sergio mentioned in his talk 2 days ago is that the populations in southern Brazil are genetically identical to the ones I sampled. The interesting thing is all of his were females, all of mine are males. So again, genetic connectivity, big question mark.
So the males can reproduce by cloning as well? Interesting.
Yeah.
Okay, thank you, Florian, for your talk.
Thank you.
And the last talk before the tea break is by Mashek. He told me, sorry. Manko, about gelatinous zooplankton as indicators of the Arctic Ocean Atlantification.
Thank you for this introduction, Delphine. So Mache is the proper one, but whatever you do, it's fine. So I would like to start by saying that I'm super grateful to be among so many jelly crazy people, and this was an amazing pleasure to listen to all your amazing talks. So the common theme among many of the talks that we've heard so far was that people were looking into exploring jellyfish biology by taking account of the environment. So try to figure out how the environment shapes the jelly biology. We heard a talk on how like winds accumulate jellies, or how the temperature promotes outbreaks of or jelly blooms. My idea is quite opposite. So I would like to suggest that we could also use jellyfish biology to try to infer the state of the ecosystems. And for that, I will be using the Arctic Ocean as my model system. So first things first, climate change is real. Temperatures are rising globally, but there's a particular place in the world's ocean that's warming nearly 3 times faster than the global average, and this is the Arctic Ocean. And the reason behind that, it's called Arctic amplification. So this all comes down to the fact that the more warmer water gets to the Arctic, the faster the ice melts, the more dark ocean it uncovers, and the dark ocean can absorb more of the heat from the sun, which warms it up even faster. But where do we get warm water in the Arctic Ocean from? from the Atlantic Ocean. So there's this huge connectivity between the Atlantic Ocean that delivers the warm and more saline Atlantic water with the Gulf Stream or its northward extension directly to the Arctic. So the Gulf Stream then splits into two branches nearly at the top of the continental Norway. One branch would flow through the shallow Barents Sea, where it loses most of its heat to the atmosphere. And then there is this very important branch that goes through the deep Fram Strait, and it fuels basically the entire circulation of the Arctic Ocean. So what we've seen in recent years is that this water flow of the Atlantic waters, it's becoming more warmer, more saline, and then there's this bigger volume of Atlantic waters getting delivered higher into the Arctic. And this obviously comes with the cascade of environmental effects. So what we're seeing now is that the typically polar environments are now turning into more a boreal state in a process referred to as Atlantification. And Atlantification comes with the magnitude of ecological effects. And the most obvious are biogeographic changes. So we are seeing more boreal species appearing upwards, like in the higher Arctic. But we are also seeing some differences in the reproductive patterns. So boreal species are now able to reproduce in the Arctic, but they are also even able to reproduce 2 times in the Arctic, much like in temperate zones. But as is usually the case, we know all of that based on the studies on crustaceans, and the literature on gelatinous zooplankton is very scarce, if any. So to tackle my big question, whether jellies can be used to observe or monitor the Atlantification of the Arctic, we first needed to come up with the diversity baseline of jellyfish in the Arctic, and then study their population structure as a potential another indicator of the Atlantification, and then try to link that with environmental drivers that we could sample in the Arctic. So since going to the Arctic is rather expensive, we decided to use samples that were already collected as a part of the Polish monitoring of Arctic zooplankton. So I picked up a 12 years long time series of plankton samples collected from the area spanning nearly 300,000 square kilometers in the European Arctic. So I got the plankton samples and the CTD probe readings from this area. And then I've also added some samples collected by a friend in 2012. They were collected in the vicinity of my research area, but with the multi-net plankton sampler. So he went deeper down to 1 kilometer water deep, so to get like a better baseline of the diversity in the region. And I must note here that all those samples were previously analyzed, the papers were published, but the taxonomic resolution in jellyfish was basically jellyfish. So they counted jellyfish without going deeper into what species were present there. So this was basically useless for us, but this is a common case for regular plankton monitoring, that people don't care about jellies. So my task was basically picking all the jellies from the samples, identifying them. So we got nearly 15,000 records of jellies from the region, and then measuring each and every individual, and then identifying life cycle stages if possible, to get the proxy of the population structure of different species in the area. Since we are limited on time, I will not go into much detail, but if you want to learn more, I invite you to read some of the papers that resulted from this work. And so just to give you I try to be brief overview of what we found. So to start with the diversity baseline, we identified 26 taxa, and they were all like species that we expected to see there. So there was nothing new, nothing surprising there. We see highest diversity in shallow shelf waters, primarily due to the presence of meroplanktonic taxa. And then in the open ocean, what we see was high diversity, mainly driven by Trachymedusae. Then, given this diversity baseline, we went to look at the environmental drivers of those distribution patterns. So because of the giant spatial and vertical scope of our sampling, majority of the differences were driven by geographic factors, with the hydrological factors, the ones that we are really interested in, driving only 1% of the variance on their own. But what's very important is that the interaction of the hydrological factors and of other factors explained most of the variance in our data, which was something that we were hoping for. And this just to note that biotic factors, which were basically food abundance for different jellies, were also able to explain significant part of variance in our data. But this was not very important. What's really important is the fact that we found some species that were strongly associated with particular conditions, like this siphonophore, Physophora hydrostatica. We found it only in super shallow and super warm waters in the Arctic. And then what's even more interesting is that some species, like some developmental stages of the same species, showed proclivity to different environments. So they had different environmental preferences alongside their life cycle. And this is Aglanta digitale. So with that in mind, we went on to apply indicatory species framework proposed by Louis Legendre to test the strength of association of different species towards particular water mass, oceanic fronts, or water mass or oceanic currents. And these are a few examples. So Siphonophore rugiacovia plicata was found indicatory to Arctic water mass, juveniles of Aglanta digitale for the Atlantic water mass, and this nice Hydromedusae Bougainvillea superciliaris was indicatory for polar surface shelf waters. So the next big question was whether our indicator species could mix in between adjoining water masses. And so when two water masses meet, they usually have different temperature or salinity signatures. The oceanic front would form in between. And when the two water masses have different density, then we have something called active oceanic front, which basically means that we have strong density gradient that's impermeable for jellyfish. And we know that from literature that jellies tend to aggregate near the density discontinuities. But what we have in the Arctic is that because we have warm and more saline waters meeting cold and less saline waters, there is basically no density gradients in between them. So we are seeing what's called in literature passive oceanic fronts, not associated with density gradients. And there was no information whether this is permeable for plankton or not. So we start with the regular indicatory species analysis, pointing out to Beroe being typical for the site west of Arctic front, Aglanta for the Atlantic inflow, and then Mertensia ovum being found mostly for the zone east of polar front. And then we took advantage of the fact that heat content of the Atlantic inflow showed the interannual variability. So in extremely warm years, like in 2006, the amount of the heat delivered to the Arctic pushed aside the oceanic fronts. So this allowed us to pick particular sampling sites that jumped from one side of the oceanic front to another site while remaining at the same geographic position. So we used this case to try to see whether we would have the same jelly composition, with the hypothesis here being that if the fronts would be completely permeable, we would see the same composition irrespectively of whether jelly sampling site would be on the east or west side of either of the front. Fortunately for us, that was not the case. So we found that even those not density-driven oceanic fronts, they can structure jellyfish community. So what's very interesting and maybe slightly counterintuitive, the warmer Atlantic waters had the higher proportion of Aglanta digitale, had lower diversity of jellyfish that was basically dominated by Aglanta, and they had also higher abundance. And this could mean something for the future forecast of how the Arctic would look like, but I'll get back to that later. So the final big question that we had was, since Aglanta was emerging as an important indicator of those warm Atlantic waters, we wanted to look deeper into their population structure to see whether the population structure in itself could be an indicator of the progression of Atlantification of the Arctic. So what you see here are the 1 millimeter size classes of Aglanta that we use as a proxy of the population structure. So if we focus on extremely warm years, what you can see is that they were characterized by higher abundance of Aglanta, irrespectively of sizes. And these are the white dots that you can see. But what's very important is that in those extremely warm years, what we were seeing in the southernmost area of our sampling or investigative area was a high proportion of baby jellies. And this may seem counterintuitive. Warmer water should promote earlier onset of the reproductive cycle, so we were expecting to see tons of large jellies. But what this actually means is that our jellies are now reproducing 2 times in this area, so twice a year, which so far was only reported in the north of England. So we are like jumping with the boreal state really quickly to to the Arctic. So what also came out of this data was that there is this clear linkage between the situation or the population structure in the lower latitudes versus higher latitudes, clearly pointing towards the connectivity of the populations. So the southern population was seeding the northern population. And we don't have any molecular data. These are ancient samples kept in formalin, but I know that Charlotte Havermans is currently working on the population genetic study of Aglanta, and she's been finding no structuring across the Arctic Ocean. So basically all gets derived from the Atlantic up north. And then the final confirmation, so this was a slight exercise that we did. So we were trying to use the presence and absence data as well as abundances of Aglanta. And we built this simple model trying to delineate the differences in response or ecological response of baby jellies and large jellies. So that's why we introduced this interaction term with all the variables tested. And what this allowed us to do was to delineate how baby jellies and large jellies differentially respond to different environmental patterns. And what we saw here was that we see this very pronounced response from the baby jellies to changing spatial patterns that we are sampling or changing environmental factors, whereas larger jellies were not as responsive as the babies. So I know that I touched on many topics, so I just, as a quick conclusion of the talk, what I show is that jellyfish diversity, but also their population structure, can be used as a good indicator of Arctic Ocean Atlantification, and most likely for any other oceanic processes that we are observing. And that the passive fronts, that at least those that we see in European Arctic, can form barriers for jellyfish mixing. And then Atlantification will likely promote less diverse and more abundant Aglanta-dominated jelly community in the Arctic. So it seems that jellies are indeed coming to the Arctic en masse. So with that, I would like to thank you all for your attention.
Thank you very much.
Thank you.
Been back in the Arctic many months ago, so it's nice to be back now.
Yeah, I don't know why I decided to go to the Arctic. I hate when it's cold, but it was a fun experience, I must say.
Any question?
Hi, nice presentation here.
Thank you.
Yeah, I just would like to know what is the maximum that these ctenophores can act as indicators.
What's the maximum of, sorry, what?
Depth.
Of depth?
Yeah, to what extent? I mean, to what depth they can act as indicators?
So they were not acting as indicators of the sampling site, but they were acting as indicators of the water mass. So we had vertically stratified sampling, so we could pinpoint, like, the water mass was just like 20 meters deep, so we would see most of these ctenophores only here, or siphonophores or whatsoever. So it was not like sampling site specific, but water mass or oceanic front or oceanic current specific.
Okay. The deeper water ctenophores have not been analyzed, or
Sorry?
deeper water ctenophores.
So as many people before me already mentioned, ctenophores are a bit tricky to work with, and I'd like to remind you that I was working with samples that were 5 to 10 years old. So the best I could do was to say it's Beroi or not Beroi, or it's a Mertensia, but it might as well be Euplocamis or other cedipede. So there is some caution that has to be taken with the ctenophore data that I'm showing.
Okay, thank you. Great work.
Thank you.
Thank you. Love Aglantha, love seeing it pop up all over the world. Do you think, based on your data, there are strengths or limitations to particular depths? So like I imagine when you get really close to the surface, you have a lot more influence from wind versus slightly deeper down, and how might that all kind of play out in practice?
That's a very interesting question, and for that I think we would need some better resolved like vertical sampling that we had. So what we had for the majority of time series was basically somebody was putting CTD probe first in the water and then identifying water masses, and then we were collecting samples just within these water masses. And then for multinet, we were using like predefined depths of sampling, and that's not enough to infer like more intricate patterns. But I'm sure that like winds and other stuff, they for sure play a role there. So this is more like a big picture and like directions to further studies.
With all those glanthas that you looked at, I wonder if you have any comments on, because you put prey up as one factor, on the stomach contents of them, and if there's any change or.
No, so again, the pictures I showed, they were taken on board when I was sampling, and these samples were not included in this work. Whereas those samples that I got, they were pretty damaged. So the best I could do was identify species, which was not always possible, and then gut content that would be impossible. But we know that Aglanta, they have different dietary preferences throughout their life cycle. So babies, they tend to eat protozoans, and then as they get bigger, they tend to shift towards copepods, and they are even able to capture some ketognaths, but it's just like not a big portion of their diet. So I'm sure that the diet plays a big role in their distribution, but not enough data to say anything more.
Thank you, Maciek.
Thank you very much.
Thank you for your talk. So we are going to have a shorter break. So if you can make it 20 minutes rather than half an hour, because we are way behind time. And yes, please come back about 10 to 12, please. They what? Yes, they're not in. Okay. A couple of announcements. Martin and Doris, we're still waiting for your presentation. I don't see Martin. Doris is online. I think Martin is here. Yeah, his presentation did not, he didn't upload his presentation for now. It's the one before last in this session. Martin, did you upload your presentation? Man. That's why jellyfish are divorcing. The male are on one side and the female goes to another continent. They're smart. Okay, we're going to start with Verena Ras' presentation. Please, Verena. Verena is going to tell us all about hiding in plain sight, hidden genetic diversity of West African jellyfish.
Okay, good morning / afternoon, everyone. My name is Verena Ras. I'm from the Department of Biodiversity and Conservation Biology at the University of the Western Cape in South Africa. Just before I kick off my talk, I just wanted to say that it has been a really fantastic experience to see all of the wonderful work being done on jellyfish throughout the last couple of days. It's great to see more and more regions represented, and also to see more and more taxa being studied. But in as much as we are seeing an uptick and an increase in jellyfish research across the world, unfortunately, there are still many regions that are sorely underrepresented. Africa is one of those regions, notorious for being underrepresented in several domains, with global studies often only having one or few samples from Africa. For example, the study by Aboud, which was a truly global genetic study, but only encompassed one sample locality from Africa. The entire African continent was represented by only one sample. This is problematic. But thanks to the advocacy that Professor Mark Gibbons has been doing over the last decade, probably much longer than that, jellyfish has been added to the sailing orders of a couple of research efforts, particularly the Dr. Fridtjof Nansen, which embarked on widespread collections across the entire African coast, collecting not just jellyfish, but lots of other taxa along the way. This expedition spanned the west coast of Africa as well as the east coast of Africa, and probably encompassed the most widespread collection that we've seen in recent years. And so you can imagine how excited we were to get our hands on a lot of this data and a lot of these specimens. So I was lucky enough to be able to work with a lot of the West African samples. And of course, I couldn't look at everything. And so what I did was I randomly selected samples from each leg of the cruise and just looked at what I was seeing, essentially looking at patterns of diversity, uncovering some genetic diversity or taxonomic diversity. I looked at a lot of taxonomy, but I focused a lot more on the genetics. So I did a lot of barcoding, not just CO1, but I looked at a range of different gene regions. And I want you to brace yourselves for what comes next. I found some really, really cool things already, although I didn't have such large sample sizes. We saw some really interesting things. Lots of atypical patterns emerged in Africa, which I'm going to cherry pick a few of them. I'm not going to present all of them, but I'll present some of the things that I found really, really cool. So I'm going to start with the Otolla. So we know that the going theory is that with increasing depth, we typically have increasing gene flow. While quite interestingly, the Otolla species that we recovered, or taxa that we recovered, showed instances of sympatry actually with Otolla van Hoefene and Y Valley present on the West African coast and occurring in sympatry in a couple of the samples that we analyzed. And although this is not the best tree, this is a consensus tree. I just wanted to show you some of the diversity a little bit more visually. And you can see that from this tree alone, there is a little bit more structuring even that might exist, which we can explore further in the future. So this was really cool for me to see already that a deep ocean species is showing so much variability. Fast forward to the pelagia. We also recovered quite a number of pelagia samples from the West African coast, and we also saw some very interesting patterns. These are not just West African samples, these encompass samples from Ale, Stopa, and Miller as well, but I'm going to point out a couple of interesting things. The cluster you see over there is mostly Southern African samples, so samples collected from South Africa. And interestingly, although it's not maybe presented so well here and maybe you can't see it so well, they actually formed a cluster with a number of haplotypes of Pelagia panopyra, which is from the Indian Ocean. So that was quite interesting. I was really excited to see that. Other trends, though, were quite similar to what other studies have published. So, for example, specimens from the Bay of Bengal, Indian Ocean, was placing mostly basal to all the other taxa, and also formed a quite distinct cluster. You can see that at the bottom of the phylogenetic tree over there. And a lot of the Mediterranean and North Atlantic species obviously tended to cluster together as well. We know this from the work by Stopa and Ale from the past as well. Costa Rican samples also formed a distinct cluster. You can see that over there. Again, this is not really new. But what was really interesting was that there were some distant Mediterranean haplotypes that clustered with some of the Indian / Bay of Bengal samples, so that was quite cool. We need to explore that obviously a little bit further. And even more interestingly was that the North African pelagia that we collected formed a completely distinct cluster, and we still don't really have any idea why. We need to explore this a lot more. Maybe looking at the morphology in a lot more detail and exploring the genetics a lot more as well. But I thought that was very interesting. And this was a cluster of samples from Morocco, Angola, and just outside the Gulf of Guinea. So that was also quite interesting. So it shows that there could be higher levels of structuring that exists along the West African coast. And Pelagia is seen along the West African coast all of the time. And so this is why I've called this hidden in plain sight, because we see them all the time, but we had no idea that we had this level of structuring and diversity existing along the coast. Now I'm going to switch to talking a little bit about Chrysoora, which is the love of my life. So we managed to collect quite a number of Chrysoora from the West African coast as well. Probably one of the more widespread collections that we've managed to make over the last decade or so. And so those of you that are familiar with Chrysoora would know that along the west coast of Africa, we have 4 species that occur there: Chrysoora isosala, Chrysoora africana, Chrysoora fulgida, and Chrysoora agalensis, which is a recently erected species. Interestingly, I did not recover any Chrysoora isosala in any of the samples that we collected. This blew my mind because traditionally this has been thought to be the dominant species along the west coast of Africa. I saw no Chrysoora isosala. I did, however, recover 2 morphological specimens that was phenotypically quite similar to isosala, but when we looked at the genetics, we only saw something in the region of about 2.4 to sometimes more than 3% variation, but that was sort of where it stopped. So the variability was quite low. So just diving into this a little bit deeper, just a simple phylogenetic tree again. We obviously didn't recover a whole lot of Africana, but the Africana that we did recover also showed some very interesting trends. Again, North African samples were basal to all the other samples, particularly the Western African samples where Chrysoora africana is typically collected and recovered from. So that to me was also very interesting. There were also some differences in phenotype. And one thing I found really interesting was the further north we collected Africana, the more and more it started to resemble Chrysoora pseudosolata. So that was quite interesting, and I'd like to unpack that a little bit more. And we also saw high levels of genetic variation of up to 4% between the North African samples and the Southwest African samples, which was also quite interesting to me because it shows that there's something going on in the north of Africa that we need to explore a little bit more. Another underexplored, very underexplored region. Now, the Chrysoora fulgida gave me a headache. Every single gene I analyzed produced almost a complete star tree, and I freaked out because I thought I was doing something wrong. What I've presented here is the best resolved tree that I could recover. No matter how many runs I did, This was the best tree that I could recover, and you can see all the support values are incredibly low. But I'll come back to why I think we were seeing this in just a second. But the one thing I do want to point out is when I dove into the genetics a little bit deeper, I found that across the entire Benguela region, spreading up to Angola, every single sample that we collected, when we compared them, showed less than 1% pairwise sequence divergence, which I didn't really expect from the region. I thought there would be a lot more variability, but it was coupled with extremely high levels of structuring. So we had FST values that were incredibly high across the region, but coupled with extremely low diversity, and that was quite interesting for me to see as well. I'm going to come back to these two CI Sisala specimens if I have some time at the end. But just to say that we did also see a lot of morphological variability in the specimens. The specimen on the far right is a juvenile specimen, so obviously we know that they look quite different. But I just put a couple of images up there so you could see some of the variability. Not too variable, but there was some variability that we could detect. So here's where things get even more interesting. Chrysoora agelensis is a recently described species, and we presumed a recently diverged species. But when we started looking at more morphological specimens, more taxonomic specimens, more genetic specimens, we actually started to see kind of high levels of structuring that we didn't expect in something that was supposedly a newly diverged species. So that was very interesting for me to see. And again, large variation in phenotype. These are all coming from the exact same bay. And you can see all the variations that we can see in terms of the phenotype. There's lots of variation, and I have absolutely no idea why this is happening. But the more we sample, the more phenotypes we're starting to see. And we see lots of structuring, even within the same bay, in the genetics, but also in the morphology. So that has also blown my mind, and I'd like to really unpack that quite a lot more. I won't have a lot of time to talk to some of my next results, but what I thought I would highlight here, although I'm not presenting it in the slides, is that I did also obviously draw a bigger phylogeny that incorporated all known Chrysoora. And what was very interesting was that this species was basal to Chrysoora fulgida in every single phylogeny, which might suggest that actually this is the ancestral species or the source population. And then all the sort of star topologies that we're seeing in the Chrysoora fulgida might make sense because it might be indicative of recent population expansion or rapid population expansion. Just a theory, we'll have to unpack that quite a bit more. Now I wanted to talk, oh, my slides went a bit funky. I wanted to talk a little bit about some of the other analyses that we did, but I'm going to put a pin in this in the interest of time and maybe just highlight that because of the wonderful work that's coming out of the Dawson lab, they've been standardizing a lot of the procedures. And so because of that, I was able to duplicate a lot of Aboud's work across African LMEs, and we actually uncovered quite a large amount of structuring within the African LMEs that I'm not going to talk to right now, but you're welcome to come and chat to me about it during the lunch break. So I'm going to skip those and maybe just end off by saying ultimately what is really exciting is that we are starting to build a baseline of evidence for species diversity across Africa, which is not really there right now. And though this might not seem like anything pioneering, I think it creates the avenue for better, bigger, bolder questions in the future, and it's helping us uncover the scale at which those questions need to be asked. And I'm hoping that in the future, many more maps look a lot like this and a lot less like the first one. This is a horrible map, I know, but it was horribly distorted when I visualized it. But just to give you an inclination of what I hope to see in the future. So thank you to everybody who participated in all of these efforts. You really made my time in the lab really, really wonderful. Thank you.
Thank you, Verena. And I remember seeing some of those big jellies of Namibia. Any question? Okay.
Thanks, Verena. That's awesome. Hey, so the one pelagia that's a little wacky up in Morocco, is there any chance that that's like Mawia?
Yes.
Yeah.
So I did anticipate that it was Mawia. After looking at some of the morphological specimens, I do actually think it's Mawia, but it's not like any Mawia we've ever seen. So that's why initially I thought it was a pelagia, because it resembles pelagia tit for tat, but the genetics I would suspect is probably more along the lines of Mawia. And also, I didn't mention this here, But Maui is widespread along the West African coast. We found that in our samples. And so that was also quite interesting because it might hint at different source populations for Maui as well.
Anyway, just wanted to know in terms of the trees that you did. So which marker? Is it a single gene or concatenated?
It's a concatenated data set of CO1, 18S, 16S, and ITS.
There's a question in the back over there.
Mine is the nice work. Mine is just a suggestion because you are having a very good morphometrically difference. Why can't you construct a tree with morphometry as well as sequence with DNTs with other file? Maybe you should use other softwares. Maybe it can resolve better. But very good. Thank you.
Yeah, no, so I did look at morphology as well, but this talk was called Hidden Genetic Diversity, so I focused on the genetic diversity, but we have looked a lot at the morphology as well. We've measured tons of specimens.
Mine is, you can, what I meant to say is,
A
you
combined
can
tree, yeah.
construct both with morphometry as well as phylogeny. So
We will.
it will resolve better. Yeah.
Yeah, we will definitely. I totally agree as well. Yeah, thank you.
Thank you. John, over there.
Hi, great talk. I totally understand you are in love with that guy because it's gorgeous. But any large hydromedusae that you are working with or that you have seen?
So I haven't worked with a lot of hydromedusae, but we do know that there's Chirodropus that occurs along the West African coast. There's also Chironix, there's Tomoya. But other than that, I haven't really worked much with the hydromedusae.
Okay, thank you very much. And we're going to welcome Ramesh Shatragada. And the talk of the presentation is Underexplored Jellyfish Blooms in Some Location of India, Impacts, Research Gaps, and Future Directions in Sustainable Utilization and Management.
Yeah, thank you.
And remember, if you're a student and you want to compete in the award, just wave at us.
Good afternoon, all. I thank the organizing committee for giving me this wonderful opportunity. And I stand before all the expertises and I present just my preliminary work, although it is not the primary work, but the main work that I focus is on a coral reef survey. But while we working on these coral reefs, we come across a lot of jellyfish blooms, although we could not spend a lot of time on it, but whatever we have obtained the data that I'm going to present here. That's why we put the title Underexplored Jellyfish Blooms in some locations. So a lot of introduction has already been given for this Cnidarians and ctenophore, but I just would like to give small introduction. Mostly the jellyfishes will have these four phenomena, biooptical phenomena. One is bioluminescence and fluorescence, pigments, and iridescence. Bioluminescence can be seen only visually through our naked eyes or by giving a stimulation by splash or any mechanical or chemical stress. Or whereas in the fluorescence we can see only through by giving external light sources like high energy wavelength light like UV light or blue light. And the pigments also we can see through visually through naked eyes. Whereas iridescence also can be seen only in presence of a light, the refracted light that gives the iridescence effect. So the background of this study is like there are a lot of limitations in documenting jellyfish blooms in India. If we can see here is I have categorized three different individual and institutional and societal. When we come to the individual, there are safety concerns that we cannot reach many locations, and considering the rough seas or other local environmental conditions, we could not able to reach out some of the areas. And also there are regulations or legislations from the local governments. That's why we cannot access some of the areas, particularly the island setups. And also there is a limited funding and a lack of equipments or consumables to carry on our basic research on jellyfish blooms. And institutional wise also, certainly there is a limitation of research infrastructure. In particularly the whatever I am presenting here is I am talking mostly about Indian scenario. So the limited infrastructures in various research institutions along the coastal areas, and also the limited funding, and also whoever are working in this particular field, they are already prefixed to some of the locations which are already well studied. So that is why they could not able to explore the new locations where we find some of the rare jellyfish blooms that I'm going to present here. And societal, the lack of awareness about jellyfish blooms for particularly local fishermen and their communities, and also lack of local contacts, so that we could not able to establish the local contacts to get the seasonal data about the these jellyfish blooms, and also lack of support, financial support from government or from any particular other sources. So based on these limitations, we have studied 5 different locations in across the India. 2 are in Andaman, and 1 is in Gulf of Mannar, and 2 are from Maharashtra and Goa coast. So we have used in underwater camera. As I mentioned, we mostly primarily focused on coral reef surveys. We used underwater camera and GPS coordinates we have taken wherever we found these jellyfish blooms. And the small medusae or other small jellies are collected in this tarsal sample container or Ziploc bags using this hand net or scoop net. The samples are transported in ice box to the laboratory by fixing in the ethanol or in 4% formalin. So mostly we focused on the stranded jellyfish blooms, or jellyfish blooms that are away from the subtidal or little bit open sea side, and also some of the blooms are observed underwater in the reef regions. So I just given here only a few examples. Drift jellies, mostly these are biomass is very less, which are prone to drift away by the winds and waves, and they come on shore on the strand. And the other jellyfish that I referred here is current following jellyfish. So there are sayings like some jellyfish, they follow the current pattern, particularly the Pelagian actiluca. So that is why what we have recorded, this is from Gulf of Mannar. You can see the entire chain kind of stretch. It's about more than a kilometer. I have taken only to one particular area, but this is about almost 2 kilometer far away. And other free living jellyfish, which are in terms of biomass, they are huge, and they can be seen in a large number, but they will not form chains, neither they will not drift away. They just swim freely. And underwater blooms, this Cassiopeia particular genera that we have recorded from the Gulf of Mannar, it has the ability to form blooms. So we actually, what we have done is the quadrates we have taken, whatever we used for the reef surveys, that we have laid, and approximately we have counted what will be the number of jellies that can be documented within a transect, 1 meter square transect. And these are the results. So mostly, if you can see, the number can be ranged from 50, minimum 50 or 30s, depends on the species, from that is a minimum number, and it can goes up to 1,000 in case of Pelagian actiluca. So this is the biggest jellyfish bloom that we have ever recorded in the huge number within a 1 meter square quadrate. So the next one is Aurelia areata that can able to form up to 200 numbers per meter square. So, but based on these results, what we can interpret is like these blooms are geograph specific. If you can see in Andaman, only Aurelia areata is very frequently found in each and every year, and particularly before pre-monsoon. And the people say that this Aurelia areata blooms comes from the deeper water, so that indicates that monsoon is arriving. So, but the scientific validation of this particular saying is not validated, but people used to say that. So this is about Andaman, and when we talk about Gulf of Mannar, this Cassiopeia and Pelagica noctiluca are very commonly seen, but these particular two species. But the bloom is we have encountered for the first time in 2019, and this is every Aurelia areata blooms can be seen every year. There is no exception, unlike other species. And in terms of this Goa, Western Arabian Sea, west coast of India, sorry. So the Chrysosera species are forming, and the Cramionella are forming very frequently the blooms. As mentioned, Sarvan Raj and other two of our colleagues, they also highlighted the same. So mostly these three particular species having bloom formation in the Western Arabian Sea, whereas in the east coast of India, mostly this Pelagica noctiluca and this Aurelia areata having this particular bloom forming ability. So this is the example of Pelagia noctiluca, the close-up shot of this particular bloom. So you can see it's starting, and you can see a light color here. It goes up to several kilometers. So our idea is to, the date and times and latitude, longitude, that the details we have, and we are going to just map it through satellite imaging. So this can form. We are unsure whether this chain formation of bloom is due to a current pattern, or they have their own pattern forming, chain forming ability like this. So we are not sure, and we have to validate that also. And the negative impacts of jellyfish blooms that we have encountered throughout all our coral reef surveys are as follows. So mostly we try to collect some phytoplankton or phytoplankton samples. That time when we operate, not I have just given for example this Pelagia noctiluca, but there when we operate this plankton net also, the time that we operate plankton nets, before that time itself these jellies accumulate, and it becomes heavy, and we cannot drag it furthermore. So and we have to avoid the net operation. So in this way, it is affecting plankton research also, and fisheries assessment research. So some of my colleagues who came for catching some fishes for their particular study also, because of this blooms, jellyfish blooms, they could not able to catch some of whatever the fish they wanted. So although the negative impacts are very short term, but it is affecting. These are the some of the areas, scientific areas that are affected by these jellyfish blooms, particularly coral reef research also. So we went during this Pelagia noctiluca bloom event, and we had traveled around 12 kilometers to Gulf of Mannar, one of the island, Hare Island, and then when we tried to do the coral restoration, we got in, but unfortunately, this chain kind of bloom was not seen the other south side of the island, but the spars, whatever the jellies are there, this particular species, all those when they touched us, it is a very bad stings we got, and we could not able to do the work. So such a strong stinging effect we got. So because of these things, we have to stop some of these research aspects. And when this bloom fades away, then we again go for starting this particular research. And the social And there is a time and manpower consuming process involved in when we when fishermen they catch fish, they have to segregate all of them, and also the recreations, whoever tourists or whoever goes for beach, they also they are also not permitted to enter. And tourism also affected to some extent. We did not analyze to how much, but we observed and we have taken the local uh, uh, whoever have tourism operation people, they have mentioned all these details. Economy, fish catch is also limited by these jellyfish blooms. Because of in few seasons, wherever it depends on the type of species that occurs in the water, jellyfish blooms, that bloom comes. So for a short time period, the economy also, local economy also got impacted by these jellyfish blooms. Environment, and we do not know exactly how these jellyfish blooms ultimately contributing to the dissolved organic matter, thereby it's involving their major role in the food web and algal blooms. We do not know. Applications of jellyfish blooms. So mostly of the jellyfish species having the collagen as a main biomaterial that can be used in food and supplement and cosmetic, medicinal, and other applications. But there are special biomolecules which are associated with this particular species, Aquariobacteria and Pelagia noctiluca. This having a lot of value. 50 microgram of GFP is equal to almost $150, and 250 microgram cylindrogen is equal to almost $380. In this way, the bloom, whenever the bloom occurs, we have to focus on exploring this kind of particular molecules. It depends on the species, either it is collagen or other peptides or fatty acids, molecules, or whatever the compound. In that way, in that perspective, we have to think and utilize them. I think my time is getting over. I'll just skip this. Ongoing research. Currently, we are focused on DNA barcoding of some jellyfish, and we are eDNA identification. We are identifying from the coral reef areas, and also we are trying to identify what kind of symbiotic clades are associated with the jellyfish. Whether those are the same clades are obtained from the corals also, or this can be transferred from jellyfish to corals. It can be transferred and all that we do not know. So we are trying to understand what can be the clade patterns within the jellies and this. And we are trying to understand the kind of substratum that is being provided for this particular jellies in the reef environment. Mostly we come across with most of these species in the coral reef areas. So we are trying to understand what kind of substrate will help these jellies to settle in the reef areas. And bioprospecting of jellyfish. So whoever are interested in these particular topics, you please do let me know so that we can have a possible collaboration work. Research and research gaps. As I mentioned, I'll take 1 minute. Jellyfish larval settlement areas in coastal areas are least documented, including reefs. So we know the blooms, but we do not know where exactly these larvae settle in the coastal areas or coral reefs. That we have to focus. And the symbiotic clades also we have to explore. And main thing is fisher folk, they know better. They know a lot of things about jellyfish than scientific people, scientific reports, or than scientific sightings. So we have to make them to involve in this particular research so that we'll get better data. Awareness and involvement of fish work is very, very essential. And sustainable utilization of jellyfish blooms has to be made mandatory. We have to teach them to store the blooms, and thereby we can utilize such kind of material for industries in future. And the integrated eDNA with the remote sensing can be used for forecasting jellyfish blooms. And I think our colleagues, they mentioned jellyfish associated microbes, parasites, and fauna are to be assessed. Feeding behavior of turtles and other animals on various jellyfish need to be monitored. And bio-waste. This is one of the main important thing. The discarded jellyfish material has to be utilized for valorization and biomedical application. We have to explore the possibilities. And in 2018, for characterizing GFP, Osamu Shimomura, he got the Nobel Prize. So in this particular generation, how can we revolutionize this particular area on using jellyfish, what we can do, whether we can focus on management or sustainable utilization, what can be done, that we have to think. And I thank all my colleagues from NIO and NCCR, Pondicherry University, JDSA, and ICAR. Thank you.
Time for one quick question.
We have surveyed from Chidiya Tapu, which is not the public area where people often they go. We have covered the areas where people have not been there. Yeah, we dive there. Yes.
Thank you, Ramesh.
Thank you, ma'am.
All right. Next, I would like to welcome Delphine to present her talk on siphonophores. So just make sure you're on time as well.
Thank you. I'm going to present some work that has been done by one of our master's students when I was in Cape Town already a few years back. And he did look at the siphonophore community along the east coast of the South Africa. And the east coast is actually based by the Agulhas Current. Agulhas Current is one of the biggest, even though the largest, western boundary currents, located in the southwest Indian Ocean. The flows carried by the current is about 70 sverdrup, and much more water transported by that current than any of the other current, and is a key player in the exchange of water and heat and salt between the Indian Ocean and the Atlantic Ocean. It forms south of the Delago Bight, about here, and it flows southwards along the east coast of Africa and closely following the bathymetric slope. It retroflects at the bottom, and actually there's some bubbles of Indian Ocean moving up in the Atlantic Ocean. So the continental shelf is narrow, it goes really deep, really quickly, and so the topographic is actually controlling most of the stability of the current. So the current is very, very narrow, and it's very, very dynamic. There's a lot of ADs going down from the Mozambique channels that's actually being seen in the Agulhas Current. So due to those ADs, there's some local upwelling cells that are developing along the shore, depending on the season, and we'll show you that a bit later. So what's global change going to do into that specific region? SST might increase, and mesoscale activity might actually increase, which might increase the width of the current. What's the biodiversity of the Agulhas Current? There's about 100 different habitats, a few examples, and that goes from mangroves to kelp forest in the south. The Transkei, so the shore of the east coast of South Africa, is important as it also carries or show bight that actually important in transport of water. And there's a well-known sardine run that runs from the southeast to the north along that shore every year, and it's a huge economic value for South Africa. There's reef building on the outer part of the current, but overall, do we have enough information on the ecology and to predict the changes in those sensitive ecosystems? In 2014, South Africa put together the Operation Fakisa, and trying to improve, unlock economic potential for the coastal and offshore environment for the country and its citizens. And in that goal, they want to expand the number of MPAs and trying to make it more working MPAs than just paper MPAs. Zooplankton community Actually, it's not very well known. There's only a few data available. On copepods, there's a few papers, and most of them are actually looking at the Agulhas Bank, which is on the south part of that coast. A few from Mark Gibbons on euphausids, lobsters, chaetognats, and ichthyoplankton. There's one paper that by Mark looking at ctenophores, but basically there's very limited. There's not much project going and looking at the whole transect of that Agulhas current. And among those zooplankton, I've been focusing, which I did already when I'm doing my postdoc in the late past of the century. And siphonophores are very good indicators of water mass. And they have been already used as water mass indicators or water mass movement in the past. And it's what we did already with Mark in 91. And the goal was also to see how the population of siphonophores change or not over time. And trying to see how the population diversity is actually in link with nearshore upwelling or the offshore hot waters. So that's what I'm going to show now. So it's based on a high resolution distribution of siphonophores and how along the shore and across the shore is actually showing diversity modulation or not. So that's where we did sample in 2017, 2 cruises, one in January, February, and one in July, August, so summer and winter. 38 transects were done, and 6 to 8 stations across the transect were perpendicular to the coast, and the samples were done from the south, from Cape St. Francis to Port Shepstone in the north. And the samples were limited to the 20 meter to the 1,000 meter depth. Macrozooplankton, among other things, were collected using bango nets, 50 microns mesh size, and were preserved with formalin. We did settle biovolume, which is a non-destructive, very quick way of knowing how much plankton were there. And with Dylan, we look at the siphonophores population, and we did, mostly did, I supervised, look at 95 stations in summer, 93 stations in winter. I have to say it's an amazing piece of work. So we did some statistical treatment. It's a ongoing work, so please, I haven't done everything yet, but mostly. So if we look at the environment, we can see a very wide differences between the summer on the left and the winter on the right in temperature. And we can see those eddies in blue, the cyclonic eddies that will actually bring some cold water to the shore. So it's transport, strong eddies, and the temperature gradient is much warmer in summer than in winter. So the lowest SST was seen onshore. If we look at temperature, the variation is between 10 and 26 degrees, plus 4 degrees in summer, and lower bottom temperature offshore. Salinity, bottom salinity was high, low bottom in winter, and there were several layers of water mass on top of each other. Chlorophyllae, we can see that in link with the upwelling of cold water, there was some increase in chlorophyllae biomass over the region, and reaching about 123 microgram in integrated chlorophyllae. Zooplankton, actually, in biovolume, is showing very different distribution between summer on the top and winter at the bottom. You can't really see it on the scale, but it's about 5 times more in summertime. So the biovolume goes up to almost 400 milliliters per 100 cubic meters. And there's a large contribution of crustacean, as you can see on the pictures. And we looked, a student looked at ichthyoplankton too, and they were removed from the biovolume. And the diagram here is actually looking at vertical plankton, and we can get basically the same picture. So in terms of siphonophore, what's 56 different species, mostly calicophorans, and we observed Physalia, Cordacalma, and Lancia lupi, that were new record for the region, and we got more species in summers and in winter. So, sorry. If we look at the community, there's a clear increase in richness between the shore, the 0 to 50 meter depth, and going towards the core of the Agulhas current. And the species we see that are involved mainly in that trend is Mugaea atlantica, is actually driving most of the abundance in the region, but there's other species that are also important. If we look only for the summer season at the distribution of the main species, we can see the different, watch for the different scale bars, and we can see that actually there were some species that were very common but in low abundance, like Lancia subtilis, but others like Eudoxides spiralis was reaching 220 individuals per 100 cubic meters. And we can see that the distribution is more towards the warm water for most of them than towards the shore, like Mugaea. So there's a very dichotomic distribution between Mugaea on one sense and the other species. So I tried to do some community structure looking at absence and presence of siphonophores. And actually I was a bit surprised to see something so different, surprisingly so different. So on that cluster on the A is only driven by Mugaea atlantica abundance. And I tried to superpose that cluster over the map, and it's all those dark dots. And the rest of the population actually grouped together and was following more nicely the main core of the Agulhas current. So that was in summer. If we look in winter, we see basically the same diversification with A again leading by Mugaea atlantica and the rest grouped together. If we look at the map, it's a bit less clear how the two clusters are actually distributed. So that's, like I said, it's an ongoing work. I still have to do more statistic and looking at more in-depth information on the physics, the sea surface anomaly and things like that. But what I try to actually do is that ordination of the species presence, absence, and the different environmental parameters. We can see that the group A is actually driven by chlorophyll concentration, which was richer within the upwelling system. And for the summertime and for the wintertime, bottom temperature and oxygen seems to be partly driving the differences. So, like I said, there's a lot more to go into that. So the home take-home message is that it's one of the most exhaustive data set on siphonophore diversity. I check around, I haven't seen something so with so many stations. And the other study that was done in 91 was only 36 stations over 9 transects. Were done at different stations, were done at different period. Only February was done during the same times as us. And previously we saw 59 species, so about 5, 6 species less. Species were found now that were not found before, and vice versa. And on another work I've done in the Mozambique Channel, I only found 36 species. So the diversity and abundance of siphonophores are seasonally variable, link with the currents on the Transkai shelf, and there's a strong dominance of the Mugae atlantica in the Agilas current. And looking at the ectoplankton distribution, actually the region just up north from that high abundance of Mugae has been identified at nursery areas for several small pelagic fish. So is the potential predation by Mugae and other jellies going to affect the fish eggs and larvae in that region? That's something we need to look into. And Algoal Bay, so this southern section, is becoming also a suitable and potential nursery areas for anchovies. And those anchovies seems to be moving up east under the changes we have seen in the region. So siphonophores are good indicators for water mass. High aggregation of siphonophores have been shown during that work that they do occur at small scale, and they can impact strongly the structure and the functioning of the ecosystem. And under that FACISA, Operation FACISA, the management of the southeast coast of South Africa should take into account the siphonophore community. So that's mostly all I wanted to show today. I just want to, give an appreciation to a friend of mine, to John Wacky. He was one of the persons that helped me at the beginning. I'm a very good friend of his daughter, and he passed away in August. And thanks for Rebecca. And you see I put some babies Velela in my picture. Here we are.
Thank you, Delphine. Maybe we can have one question. Yeah.
Thank you, Delphine. Very nice presentation. Just a quick question. Are those counts of siphonophores based on polygastric stages only, or also nudoxids?
What I've shown is only polygastric of Elcophora.
Okay.
So I do have a value for eudoxid, mostly for ability, but the distribution, maybe I should speak loud. The distribution for ability, polygastric, and eudoxid is basically the same. They don't show any special variation.
Okay.
But we do have the data, so we'll work more on that part too.
Looking forward to seeing that. Thank you.
You will.
So maybe we'll continue on with the next talk. Thank you, Delphine. Yeah. Right. The next presenter is Maria McGuinness.
Hi, everyone. Thanks for having me here. So, firstly, I'm from Ireland, from the University College Cork, PhD researcher. So all these analyses are still ongoing, just so you know. So the title of my talk today is The Irish Seasonal Coastal Current, the Source of Mugae Atlantica in Ireland. So Delphine teed my talk up quite nicely just there. So this is Mugae Atlantica. On the left, we have the polygastric stage, and on the right, we have the eudoxid stage. And in Ireland, we would consider this a harmful jellyfish. In this conference, we've heard a lot about the interactions between jellyfish and fisheries, but in Ireland, it's more so jellyfish and aquaculture. So these jellyfish are so small, kind of max 7 millimeters. They can drift into the cages of salmon farms, sting the salmon, sting their gills, reduce their immunity, reduce respiration, and this can ultimately lead to mortalities. And as my colleague Hannah spoke on Wednesday, abundances of Mugae in recent years in Ireland have been very high. So last September, October, we were getting 3,000 individuals per meter cubed. And a harmful event, the threshold is about 200 per meter cubed to be considered harmful. So these are some really high abundances. The season for Mugae in Ireland is around the end of July to the end of October, early November, so they're not present year round. So where are they coming from if they're only a transient member of the plankton in Ireland? Well, there is a known resident population of Mugia in the Western English Channel, indicated by the grey star on the map there. And work by my supervisor, Damien Haberlin, published in 2016, observed similar abundances with a bit of a time lag between the L4 dataset located in the Western English Channel and in Bantry Bay in southwest Ireland. And he suggested that there was a potential connection between these sites. But if so, what is this connection? Well, there's a well-described seasonal coastal current in the area that actually connects these two regions. So it's depicted by the orange color on the map here. As you can see, it flows from the west coast of France to the Western English Channel, across to southwest Ireland, and then up north, connecting with the coastal current in Scotland. And extensive research has been done on this coastal current concerning phytoplankton. So briefly, how does this coastal current form? Well, it forms quite close to the coast, and it forms only in the summer months in Ireland when the offshore Celtic Sea becomes thermally stratified. So basically, the sun heats up the water column. There's a lack of mixing in these offshore areas, and this creates a thermocline. So there's warm, buoyant water on top, a rapid change in temperature at the thermocline, and a cold, dense pool of water beneath. And this is a bottom density front which approaches the coastline. Then when we get to the inshore waters, they're a lot more mixed due to the action of the waves. It dissipates all this thermal energy. So when we get the mixed inshore waters meeting the stratified offshore waters, we get a bit of a surface front occurring. And there's kind of nowhere for the water to move. So what ends up happening is we get a perpendicular flow, which is our coastal current, perpendicular to this bottom density front. And due to the Coriolis effect and us being in the northern hemisphere, it is westward flowing. So it flows parallel to the coast. It's quite narrow as currents go, 5 to 10 kilometers, relatively fast flowing, especially compared with the surrounding water. So we're talking 20 to 40 centimeters per second. And what's more, large quantities of phytoplankton have been seen to be moved in this current around the southwest coast of Ireland. So our hypothesis is, if this current can transport phytoplankton, why not jellyfish? To investigate this, we were lucky enough to go out on a 7-day research cruise around the southwest coast of Ireland on one of our national vessels. And basically, we wanted to characterize the parameters of the coastal current, the structure of the water column in the area, and take zooplankton samples in the current itself and then in the surrounding water bodies, just to make sure that what we were seeing was clear differences. We chose Southwest Ireland for a number of reasons. Mainly, that's where we're based. Secondly, as I said, there's high numbers of mugia in that region in recent years. There's also a lot of salmon aquaculture in that region, and a lot of the research into the coastal current has also been concentrated in that region. So we used CTD methods on board to characterize the physical structure of the water column. So that's just pictures of the CTD being deployed into the water column. And we had 14 stations along a 30-kilometer distance, starting inshore at around 30-40 meters depth, moving progressively offshore. Each station was spaced 2 kilometers apart, so we got a really nice picture of the water column. And then what we did was all of these measurements that we got of temperature, salinity, density, we loaded into Ocean Data Viewer, and we built up a physical picture of the water column. And this determined where we did our plankton sampling. We used multi-nets. This multi-net had 5 nets, so we could take 5 separate depth bins, which allowed us to assess not only the horizontal distribution of zooplankton along the transect, but also the vertical distribution within the water column itself. The mesh size was 300 microns, and we collected 76 zooplankton samples across 3 transects, with an average of 77 meter cubed of water filtered per sample. And so we took samples in the current itself, inshore and offshore. Our prediction was that we would get higher abundances of mugia in the coastal current waters when we compared it with the mixed inshore and the stratified offshore. So this map shows our Transect 1 CTD data. I only have time to go into one transect, but you can talk to me if you want to know about the other ones. So the orange dots are the CTD stations, and as you can see, we started quite inshore and moved progressively offshore. And this is a section plot of temperature for our Transect 1. So on the x-axis, we have section distance. On the left of the graph is our most inshore station, starting at 0, and as we move right across the graph, we're moving more offshore. On the y-axis, we have depth, and it's reversed, so the top of the graph is the surface, and as you go down, it gets steeper, and it's colored by temperature. So red is 17, 18 degrees, green, yellow is 14 to 15, light blue is 13, and then purple, dark blue is 10 to 12. So if we look at the left-hand side of the graph, we're getting a gradual change in temperature as we go deeper. This indicates we're getting a gradual, this indicates, sorry, our inshore mixed waters. Compared with the offshore part of the graph, on the right-hand side, there's a clear band of red, warm water, a rapid change in color, so rapid change in temperature, or our thermocline, and then a large band of blue-purple cold water beneath. So that's our bottom density front. This structure is quite typical of the Celtic Sea in the summer. This plot now shows something different. It's geostrophic velocity, which is the directional flow of water, so how fast it's flowing and in what direction. Again, section distance is on the bottom, inshore is on the left, offshore is on the right, depth is on the y-axis and it's reversed, and the color indicates the flows. If you want to know more about the specifics of geostrophics, you can talk to me, but just bear with me. It's the eastern component. Basically, it means if it's a positive value, it's flowing eastwards. If it's negative, it's flowing westwards. So the orange-red are positive, so it's our eastward flowing water, and our green, blue, purple is westward, is negative, so it's westward flowing. So that core of blue in the center of the graph is strongly westward flowing water, and that's what we identified as our coastal current. And these little icons of a multi-net just show where we took our samples, inshore, within the boundaries of the current itself, and offshore. And I just want to point out as well that that band of blue is really only in the top 30 meters. So below that, we're getting eastward flowing water again. So for our results, first of all, we were delighted that we got Mugia in the first place because Damien attempted this a number of years ago and found no Mugia. And what's more is we found Mugia in the coastal currents, that top 30 meters. So this was really exciting for us. And what was even more exciting was that there was lower abundances in the depth strata below the current, and it was essentially absent from the offshore waters. Now, we did find slightly higher abundances of Mugia inshore compared with the coastal current, but I'll come back to that in just a second. And just to show you that this isn't the distribution of all the gelatinous zooplankton, I want to show you the distribution of a typically oceanic species, Aglantha digitale. The distribution is essentially flipped. It's opposite. So we're getting most of the Aglantha below the current and in the offshore waters, with barely any in the current and inshore. So it just shows that the offshore water is really connected with that below the current section, and the current is kind of connected with the inshore waters. So back to our hypothesis that Mugia atlantica is transported by the coastal current. I'm inclined to accept this. We found Mugia in the current, and we did find it across our other transects as well. But to our prediction that we would get higher abundances in the current compared with the inshore and the offshore, not quite. Yes, there was more than the offshore, but there was higher in the inshore compared with the current. But when we think about it, the kind of impetus for doing this research was because we were seeing such high numbers inshore. So of course we were going to find some Mugia inshore. And like a lot of current systems, this current isn't, a hard and fast boundary where nothing gets in and nothing gets out. Likely there's loads of small eddies that spin off at various points of the current, seeding and transient in situ population. So that's what we think is probably happening. And just briefly, we've also done some community analysis. So I didn't just assess Mugia and Aglantha, we looked at the gelatinous zooplankton community in total. And this shows, I'll just explain. The circles are below the current, the triangles are within the current, the pluses are the inshore, and the X's are the offshore. It's probably kind of hard to see on the screen, sorry. We can see a bit of clustering between the inshore and the current samples, and clustering between below the current and the offshore samples. So I'm pretty confident that there is some partitioning of the gelatinous zooplankton community here. This is still preliminary, so we're not quite sure if these clusters are significant yet, but it's probably a species-specific response for the likes of Mugia atlantica and Aglantha digitale. For the Physonex siphonophores, we kind of saw a very different picture. But anyway, in our conclusions, Mugia was found within the current. While, of course, we can't say it's coming from the Western English Channel at this point, which we would probably have to do molecular genetics to say that. We can say that we're pretty confident it's not coming from an offshore source. So thank you so much for listening to me, and if you have any questions, let me know.
Thank you, Maria. Thank you for the time. Got time for questions?
Thanks for the interesting presentation. I know your graphs were only showing the polygastric stage, but did you see any difference in relative abundances of the eudoxids and polygastric near the coast and in the current?
So the abundances overall were roughly similar. We did see more eudoxids in general, abundance-wise, than the polygastrics. Similar patterns as well. The only difference is where, say, polygastrics were more inclined to be in that surface, 0 to 15. The eudoxids were more inclined to be in the 15 to 30. But in terms of the different water bodies, the distribution was similar.
Sorry, about sizes also, did you see, were they smaller in the current, the seed population, and larger when they were on the coast, or anything like that?
To be honest, we didn't measure them. For me, it was a bit of a learning curve with all the oceanographics plus the zooplankton, so that is something I would like to look at in the future. But no, I'm sorry, I don't have that information.
Maria, there's a question here.
Sure. So that particular populations and species, it will prevailing during that particular seasons. Otherwise, it is through well established populations in that permanent coastal structures. It is there year round. So whether that particular species is prevailing during that particular seasonal current, otherwise after that also it is well established that particular populations in that around in and around that salmon farms.
So Muggy is really only present in Ireland in that late summer, autumn. So it's blooming then. Like we have time series for the last 4 years in Bantry Bay and also in another location in Southwest Ireland. And we don't see Muggia from January to June, really. So it's not always around the salmon farm. That's why we were looking at the current, really, because it's a transient population, wondering where it's coming from. And you know, the numbers that say Hannah was seeing in Bantry Bay last year, they multiplied too rapidly for it to be just in situ growth. It had to be seeded from somewhere else. Yeah, I hope that answers your question.
Thanks, Maria. So the next presenter will be Martin Vodopicek, and is going to talk about the modeling and modeling the dispersal of Pelagia noctilica in the Mediterranean Sea. Welcome, Martin.
Hi, and thanks. Yeah, the family name is Vodopivets. It's a bit hard to pronounce. I completely understand
tried.
that. Yeah, sure.
Martin, are you competing for the student award?
No.
No.
Thank you. So I'll be talking about Pelagia noctiluca in the Mediterranean Sea. And if your ears hurt when I say Pelagia, I got really confused about the pronunciation a few days ago. So I went to the fountain of wisdom called Google and checked how it's properly pronounced. And actually, Pelagia would be how the ancient Romans would pronounce it. So this is classic Latin pronunciation. While the modern Latin, called Church Latin, would pronounce it as Italians do, which means like Pelagia. But I will stick with my Slavic and ancient Latin pronunciation. So I'll be talking about Pelagia. My co-authors are Davor Lucic and Alenka Malej, who is sitting there in the audience. And let me start. I know that most of you know Pelagia very well, but anyway, for the others, it has a very painful sting. And I almost hear the Australians laughing now because with their comb jellies, but for us Europeans, this is very venomous. It has no polyp face, so it's completely pelagic, and it performs these pronounced vertical migrations. It dives several 100-meter deep over during the day, and then rises towards the surface during the night. Now, our story begins in the northern Adriatic Sea. This is the square you can see there in the map. This is also where our station is located. And this area is actually subject to very sporadic Pelagia blooms. You can see that on the timeline on the top of the image, where we had like a decade of very frequent blooms beginning at the end of the 70s, but then we had almost a 20-year gap afterwards, and then again some sporadic occurrences after 2004. Now, of course, we're interested what is the main mechanism causing this, and me coming from physical oceanography, of course, the first thing I would think about is currents. For that purpose, we used a product from the Copernicus Marine Service called Mediterranean Sea Physics Reanalysis, which gives us currents from 1987 onwards. Sadly, you can see that 1987 is the exact year when the major bloom ended. So sadly, we cannot model that period. We can focus only on the later years. We coupled these currents with an open drift, open source particle tracking tool. This is a program code which seeds fictional particles into an area and then advecs them according to the currents from the other model. So we wrote a special module for open drift, which seeded 10,000 particles in the Northern Adriatic on the years when Pelagia was observed. And then we ran the simulation backwards in time for 2.5 years. And the particles performed vertical migrations, as described before, so diving deep during the day and then rising towards the surface during the night. The minimum depth was 500 meters, or at least 5 meters above the ocean floor. And there is a result of such a simulation which shows these tracks in time. The next one. So these are the results. This is the density of particles for all the simulations summed together. And of course, you can see them going down through the Adriatic Sea, but then you can see two paths after that. It diverges. One goes to the Western Mediterranean Sea, which is, of course, a very plausible source of Pelagia. There are very frequent sightings there. Maybe you saw, I hope you saw the poster yesterday. Sonia was presenting with sightings of Pelagia, which shows very frequent sightings in Balearics and that area. So this is a good kind of result. But on the other hand, there is also another path coming from the Aegean Sea, which is marked with the letter A. And actually, we don't know much about Pelagia in Aegean Sea. Maybe some of you do. We don't. This opens three possibilities. One is that there is a more or less permanent Pelagia population in the Aegean, and it seeds the jellyfish towards the Adriatic. Or the other one is that the Aegean is just a stepping stone, so maybe the jellyfish are coming to the Aegean from another area, maybe Western Mediterranean, and then up to the Adriatic Sea. And the third one is that this path actually exists, but there is no jellyfish in the Aegean Sea, so they don't come from that area, actually. But then we decided to make things a little bit more complicated, and we seeded the particles from the Western Mediterranean Sea. And we seeded 3,600 particles, ran the simulation for 2 years. You can see them dispersing through the Mediterranean Sea. And then we took a step further and decided to introduce food-dependent mortality and reproduction and growth to make it a proper individual-based model. Again, we turned to Copernicus Marine Service and used the biogeochemistry analysis, which is available from 1999 onwards. Now, what we did was, let me rephrase that. So we decided that each of the particles will represent a certain number of individuals, or maybe a certain biomass. Actually, at this point, this model is very abstract, and there is no difference between that. So it represents a certain number, let's say 10,000 individuals, and this number changes according to food availability. Now, how to do that? We use that equation, which is written up there. I know some people are afraid of equations. Please don't be. This one is quite straightforward. So I'll go. If you can see the curve there, this is the tangents hyperbolicals that was used in our calculations. It is a function that saturates at a certain value. That's why we chose this function. Our basic idea is that actually after a certain threshold, the food availability doesn't limit the growth anymore. And then, so the PP in the equation is primary production. Dn is actually the change of number of particles in time. Individuals. And the k are 4 parameters of this equation, which determine its stretching in vertical and horizontal direction, and its translation left, right, up, and down. So we ran 2.2 million simulations with this equation. Why? Because we have no idea about the value of these parameters. So at each simulation, we just changed the parameters slightly and reran our 2-year run. And after the run, we decided that we want to keep the initial population quite stable, which means that the particles that have left the initial area had to be replaced with growth of number of individuals of the particles that stayed in the area. And in the end, the difference should be less than 20%. So we kept only the simulations that did this. So from 2.2 million simulations, we were left with 13,000 simulations, but they're placed in this 4-dimensional space. We have 4 parameters which are changed, and we are looking for aggregations in this 4-dimensional space, which would indicate that we are onto something, that these parameters actually have some meaning. So sadly, humans are not made to think in 4 dimensions. But luckily, we have artificial intelligence, so we use the k-means clustering to search for clusters in this 4-dimensional space and return the centroid values, which would be the values of k, and use this to perform multi-year runs. Now, I'm good with time. Okay, but even so, there are 3 plots on the right side that show the shape of our curve depending on these parameters. So these are some of the clusters that were chosen by the machine, by the algorithm. And the top one, as you can see, saturates very quickly. So actually dependence on food concentration is very low. And actually it's always positive, so there is no mortality. And so discarded configurations like that, this is completely unrealistic. The bottom one is almost linear, so there is no saturation, and we want saturation, so we discarded the configurations like that as well, and kept only those that were similar to the middle one, where you have mortality below certain primary production value, and where you have saturation above certain threshold. But sadly, even among these, most are unstable. So the population goes extinct in 4 years or explodes, and all of a sudden we have millions and millions of particles running around, which is computationally unfeasible. But here we go. Some of them actually look quite well. This is one of these. This is the 6th year of simulation. The scale, the color scale is logarithmic. And you can see that kind of a very strong population established itself between the Balearics and, for example, Sardinia. And this is something that we would expect to happen. And then there is export of particles towards the Eastern Mediterranean, but their numbers are starting to drop quite rapidly. They're turning blue and green. And you can also see some particles entering the Adriatic Sea, which makes us living there very happy. So this is more or less preliminary work. It's actually started as a side project, but I kind of got in love with it. It's really nice to see patterns, developing from very basic principles, but in the end, mimicking something that is really close to what you see in nature. So, as said, we have a very small number of very basic assumptions to form our model. There are many other things that maybe should be taken into account or not. We'll see. We'll try some of those. For example, we have loggerhead turtle abundance on the right, a paper that was published last year, and I just actually found out about it a few days ago. And that, for example, would enable us to introduce spatially variable mortality. So this is work in progress. There are things to be done. But yeah, I must admit I'm quite happy with the results. So I acknowledge the funding from our agency, and I thank Tinkara as always for her support in this work. Thank you.
Thank you, Martin. It's one of our babies in Marseille. And actually, I'm quite surprised to see so many in the Gulf of Lyon, because there's only a certain time when they actually can get into the Gulf. It's when we get the Mistral. Actually, it's after the Mistral. The current is pushed on top of the slope. And
I know, yes, I heard about that. I think this is the paper by Berlinet probably, right? That did exactly export of particles again towards the coast. Well, I have to emphasize it is just a very simplistic model of a very complex biological process. So, of course, there must be differences. But even like to form, something that seems plausible, I think is quite a success.
Thank you. Any question?
Hello. Nice presentation. I also thought of the idea of the reverse particle dispersal during my class in numerical ocean modeling. But my question is, have you considered using a nested model within hotspot areas wherein you find of interest? And a follow-up to that was, what is the size of your grids?
So I'll go in reverse again. The grid is 4 to 5 kilometers. This is a European forecasting model, but it is also used for reanalysis. So we use this one. This is NEMO code. Well, yeah, I mean, it could be done. Why not? But we are certainly not at that stage yet to be interested in such fine scale dynamics to need a nested model. I mean, 4 to 5 kilometers for now, let's say, should be enough. Yeah.
Hi. It was a nice presentation. Thank you.
Thank you.
So my question would be whether can we change the size and shape of that particle which was introduced into the currents so that we can understand which direction or how fast it is moving along with the currents.
So I'm not sure that you are asking about the shape of the particle?
Yes, you released around 3,600 particles.
I released the particles, but they are completely passive. So they are transparent. They have they go with current speeds. So they have no. How would I?
Is it possible to set the size of particle in that?
The size?
Yes.
To set
size
it.
Or shape, yeah.
I never thought about it. I mean, you can. Depends on what you're interested in. So the particle has no hydrodynamic properties. It's just it goes with the speed that the current at that location dictates. So it's like actually a small parcel of water. But it depends. For example, when we were looking at other similar simulations in recruitment, we considered it bigger since each of these particles is actually super individual, so representing a large group of individuals. So in that sense, it could be bigger, yes. But in the hydrodynamic sense, it's actually infinitesimally small.
I have a last question. Have you also considered the swimming behavior of the Pelagia into the model?
No, in this sense, not. I knew this question was coming, of course. No. As said, well, this is a long discussion. Of course, they swim. It's quite obvious. But you would have to show that it's energetically feasible and how would one say, beneficial to swim in such a horizontal direction. And I know nothing about it, and I haven't heard anything about it. So they might swim, but I can't put it into a model until someone tells me how fast they swim and why they swim. So until that point, they are completely passive.
I'm just a quick comment. I'm not a Skyphozon person, but just to give you a hint, when you poke at Pelagia, they would just dive deep right away, or they would just swim away from the cue that you're delivering. So maybe you could think of like contact between your like sample points or whatsoever. Then whenever they interact with each other, they, I guess, also swim away from each other.
Okay, bear in mind that this is kilometers of solution, so they are quite far apart. Okay, high density would maybe mean, but as you mentioned, contact. That's why we don't let them go deeper than 5 meters above the bottom, because they dislike contact, so they stay above the bottom, and this is just as far as we go in that sense.
A quick one from Rebecca. We still have one presentation to go.
Thanks. That was really fun. Pelagia is a fascinating animal. Did the migration make a difference?
Sorry?
The vertical migration?
Yes.
Does it make a difference? If you
I
took
didn't
it out?
try actually. I started with vertical migration from the start, so I didn't try what would happen if they didn't do the vertical migration. It did make a difference when we introduced food-dependent growth and mortality. It was actually quite a problem because in the beginning they favorized shallow areas, which is completely wrong, right? Because they couldn't dive, they were all the time in the food area, just feeding and growing, and all of a sudden they exploded in nearshore areas, which is completely unrealistic. So we had to introduce, for example, there is additional mortality, there is a punishment for being in shallow areas so that they are not penalized by diving deep. But regarding the transport, there must be a difference. You know, of course, the currents at the surface are usually much stronger than in the deep. But no, I didn't do any comparison, actually.
Thank you.
Thank you, Martin.
Thank you.
So the last talk of this morning session is going to be given remotely. Hi, Doris. Can you hear us?
Can you hear me?
Yeah, we can hear you. So Doris is going to speak about antoathecate jellyfish and their polyps in Swedish waters, an integrative taxonomy approach. So the floor is yours, Doris.
Thank you. I'm very grateful for the opportunity to give my full presentations. It's exciting to be part of this symposium, of course, with all the exciting talks. So sorry for any repetition from yesterday, but I'll expand some more. So I have a project called Hydrons, or Hydrozoa in Sweden. And yes, I'm a PhD student at the University of Gothenburg and would like to be considered. But we focus on antoathecate diversity in Sweden. Oh. So Hydrozoa taxonomy actually used to be an active field in Sweden. During the 1700s, Linnaeus and Forssell described several taxa. These are just a few examples that I show you here. And the field continued to be active during the 1800s with several monographs and other contributions. But during this whole time, the focus was usually only on the benthic diversity, so missing the pelagic stages and thereby also missing a lot of the diversity, potentially discriminating against antoathecates, as many of them have very small and similar looking polyps, like several of the hydroctinids, for example. And after the 1800s, this field kind of came to a halt in Sweden. During the last century, the only targeted survey that's been done was a 1 week long workshop led by Dale Calder in 2010. But then, of course, they've been covered to some extent by more general faunistic surveys as well. For example, a huge one in the 1920s and 30s led by Leonard Jägerskiöld. And then homage to that one, led by the Swedish Taxonomy Initiative, a 4-year survey. But again, they completely excluded the pelagic stages, which is also showing in the numbers for antoathecates that they've been underrepresented. In the 4-year survey, for example, they only saw 13 species. So currently, this leaves us at 73 species ever recorded in Sweden, but over 20% of these haven't been seen ever since they were first recorded or even described. And with no one working on Hydrozoa in Sweden, our national records have become outdated with old synonymies accumulating and misidentifications hanging around, no one to validate new records. And we know from work with DNA barcoding and integrated taxonomy done outside of Sweden that morphology doesn't necessarily tell the whole story. Cryptic speciation is common in Hydrozoa. And integrative taxonomy has also proven useful to link previously unpaired life stages, which could come in handy here because several of our species we don't know the full life cycle for, even for very common species here like Sauridiusartia gemifera, where we haven't seen the polyp. So currently, Hydrons, sorry, maybe I missed, no. Hydrons is the only group working on hydrozoa in Sweden, and we can do this thanks to funding from the Swedish Taxonomy Initiative. They were born in 2002 after a commission from the Swedish Parliament to actually describe all multicellular species here and distribute information about them. But the project's also dependent on the expertise from my external supervisors, Louise Martel and Aino Hosia. You heard Louise tell you about worm-filled jellies yesterday, and you should probably go check out Aino's poster on gelatinous zooplankton in Norway. They've been studying hydrozoans in Norway in their projects Hypno and Norhydro for many years. And through hydrons, we in Sweden can kind of tap into their expertise and build on the finds they have from their here. And this is really essential because even though Thomas Dahlgren, my main supervisor here in Sweden, is a knowledgeable taxonomist, he doesn't specialize in hydrozoa. And apart from us, the main group, we also have collaborations. To the far left in the picture here, you see one example that you met this morning, Johan Soto Angel and his project NOAA. Here's from a sampling effort this summer. So there's quite a few parts to this project. A central task, of course, is to provide an updated national species list with robustly confirmed species. So we're collecting fresh material from diverse habitats to create a geotag reference collection, along with barcodes that will be freely available, CO1, 16S, and ITS. If you look in bold now, you'll see that there's very few barcodes from Sweden, and they have some issues possibly with misidentification. And combining these data, the morphometrics, the biogeography, the genetic distances, we will explore the diversity and evaluate dubious species and describe new species, etc. But also, aside from this integrated taxonomy part of the project, we're interested in how well eDNA is at gauging the diversity compared to traditional methods. So we'll look at this through data to metabarcoding. To find, to maximize the diversity we find, we concentrate on the marine areas. So mainly the northwest coast of Sweden, where we have the highest salinity and the highest diversity. And around in this area, we sample both shallow localities by hand and go out on ships to sample deeper localities by dredging and doing grabs and taking plankton nets. And to see how the eDNA compares, we take water samples at multiple depths to see which part of the diversity is represented where. Can we, for example, see the benthic community in the water column or even at the surface? So this is where we are now. We're just 10 months into the project, so it's early day, but I can give you a brief overview of the results. We've finished our first sampling season, which has included 2 week-long stays at Kristineberg Center, one of the 2 marine stations run by the University of Gothenburg. And I spent a few additional weeks at the other station teaching and maybe sneaking some samples from the courses there. And a little by chance, I got to tag along as the Swedish Meteorological and Hydrological Institute did one of their monthly samplings on board research vessel Svea, going down along the west coast and into the Baltic and back, taking plankton along the way. So yeah, the black spots in the map here is where we have samples from this far. And along the way, we've had a few new records for Sweden, including Velella Velella, or By the Wind Sailors, that actually beached around both the marine stations this fall. So that was quite exciting to finally have them. But our nets and dredges don't select for Antarctic, so we have other curious finds as well. 2 that we've seen most this summer are actually among these new finds, the Narcomedusa solmaris and Mugia atlantica, or Mugia atlantica, that we've heard a lot about in several talks now. Interesting to hear from Maria that they didn't find them in the offshore water. Otherwise, the current that comes in here would have been a good source. But yeah, and we've had one instance of Helgicida cari that's also new for Sweden. But back to the Antarcticates. We have at least 25 species this far, but all the material is in process, so there's potentially more. But the ones we have include several that are very rarely reported. One example is Eudendrum cnidophorus, which is described from here, but not seen in the field since 1907. We have sequenced this, and this far it does nestle within Eudendrum, but it doesn't match a barcoded species. But we need to look at more markers because there are some that could be confounding that aren't, you've only looked at CO1. And two very different examples are Hybocodon prolifer and Pachycordyle. Both of these are quite common species. We've seen them a lot, but there's more or less no records in Sweden, likely just because no one's been looking. And we also have data supporting cryptic speciation. So these are a few that we will look closer at. And first out is the genus Euphisa, where we will build on results from Hypno and Nordhydro in Norway together with NOAA for looking at the species that are present in our region. And Euphisa aurata is very common in the plankton in both our countries. But barcoding suggests that it's actually two species under this name. And this is supported further by morphological differences that strictly follow the genetic differences. And besides these clusters, there's two more species in Sweden, Euphisa tentaculata and Euphisa flammea. And now only Euphisa aurata has a described polyp stage, and since that's now a species complex, it's a bit unclear to which one that to which medusa that polyp belongs. But we have molecularly confirmed polyps for 3 of the 4 species in the region. And together with Johan and Project NOAA and others, we're looking at the family Tubularidae, including the hydroid Tubularia indivisa that you see down in the right corner here. This might also be a species cluster. There's molecular data suggesting this, but however that pans out, we will do species fact sheets for the species present here. If you look in the Swedish database now, you'll see that there's no pictures, no descriptions, so there's really low incitement for people to report them if they see them in the field. They won't maybe know what they find. And finally, there's Cordylophora caspia, an invasive species that's probably a complex. If you look in bold 9 now, you'll see that it might be up to 4 species under this one name just lumped together. And currently we don't know which ones of those we have in Sweden, but we have them over a huge salinity gradient. So we're curious to see. Right now, the only record we maybe have is a potential first lake record from Sweden, but we need to confirm that molecularly first. But the plan is to go sampling next year along the East Coast to see if we find any difference in the, any molecular evidence for differences along the salinity gradient, or if it's all the same actually, to be able to understand how they disperse and potentially manage them. It's, I think, a reasonable first step to see who is here and who spreads where. And that's all I had for now. Thank you very much for listening and for this opportunity. And I can take any questions.
Thank you, Doris. We have time for one quick question. I've been reminded about the time. No question for Doris? Okay. Thank you very much, Doris, for joining us online. And best of luck for your coming cruises. Okay, thank you very much for that morning session. So lunch is waiting, I'm sorry. We need to make a few. It's not here. I think it's in the other session.
So it's time to honor our chair and co-chairs. I invite Mr. Toggle to honor them. Co-chairs. Thank you very much.
Okay, some stuff happened. All right, so we're a little bit behind. We want to get back together as quickly as possible. I apologize. It's going to be a short lunch because we need to finish promptly at 3:30 or something like this, so that the director of CMFRI can come make some address to us. He'll have to leave properly after that. So can I ask everybody to come back at 2:15? I think that's right. Yeah, so 2:15, so 35-minute lunch. All right, sorry about that. Thanks all. All right, good afternoon, folks. So we'll get started in just a moment. A couple of announcements first. So we're trying to just get or keep the schedules a little bit straight this afternoon. So we'll start at 2:30. That should mean that we'll finish at 3:30. I'm chairing this session, so I apologize, but I'm gonna keep everything running like clockwork. Director Gopalakrishnan of ICAR's Central Marine Research Institute and Kochi will make a short address to us at 3:30. And then we're going to head outside for a group photograph. And then we'll have tea break. And then it'll be posters. And then we'll reassemble here for a short set of appreciations in memoriams. The awards ceremony will occur then. And then I think Bijou will close the meeting and we'll head out for dinner or something like that, if that sounds about right. Right? Okay. All right. So with that then, it's my great pleasure to welcome to the stage Dougal Lindsay and KV Jayachandran as our co-chairs, and Ramesh Chitragada as our rapporteur today. I'm chair for the session, and so I'd like to start by inviting Michael Brown to come start his talk.
Right. Good afternoon, everyone. So I've always been told to start a talk with a joke. At the 6th International Jelly Blooms Symposium, my first ever conference, I tried to start my talk with a joke, and it was met with deafening silence and a disappointed look from my supervisor. So there won't be any jokes this year. So I'm going to talk about morphoplasticity and environment, the lines between, where the lines between taxonomy and ecology blur. So while most of us will inherently understand the term morphoplasticity, Google apparently doesn't. If you try Google morphoplasticity, you'll get a bunch of different answers, so let's just break it down. So coined from the term morphology, which originates from the Greek morphe, referring to general aspects of, or in biological terms, it refers to the general aspects of biological form and the arrangement of parts of a plant or animal. Plasticity in a biological context refers to the adaptability of an organism to changes in its environment. So morphoplasticity is talking about the changes in an organism's form based on its environment. Why do we care? I was very happy when Alan put up a section of the ICZN code earlier in his talk. This is a section relating to holotypes and the fixation of names based on a name-bearing type. Ignore the part on the right-hand side. We'll get to that in a bit. So the idea of a holotype is very interesting in that in theory, it's what we go back to when we don't know. It's if we find something and we don't know what to compare, or if we've got data that disagrees, we go back to the holotype and we compare to that. And the idea of morphological description, so when I first started, was obsessed with genetic work. I was obsessed with computational biology, and I still am. But Andre actually made a comment on one of the first manuscripts I submitted, saying, well, what's the usefulness of the species description if you're not going to provide us with morphological characteristics that people can actually use in field? So I went back, did some work, came up with the species description there that actually worked. And it agrees very much with the philosophy that I have about if we're going to do science and we're going to put all this time in and get this funding, the science needs to be useful. And so it needs to be able to be applied by a wider range of people than just us in a lab. The comment on the right-hand side is actually, it came from my PhD review this year from one of the examiners, and I was slightly frustrated by it at first. I was actually a little offended because it seems to break down what we do quite simplistically. And I had a moment of, how dare they simplify our work so easily? And then you go and look at what we do fairly frequently statistically. So just to give you an idea, if we talk about holotypes and why we have problems in this area, thanks, Mike, for letting me use this next example. But if you were a young scientist and you arrived in Palau and you were looking at Mystigius, those two things for most people don't look the same. If one of those was your holotype, you may have a serious problem trying to describe the second as the same thing. So, and then on the converse, we have, well, not on the converse, actually agreeing. If you look at Aurelia, so we can distinguish Aurelia morphologically a lot of the time, hopefully most of the time. And if you want to see more of that, a great paper is Scarano's 2017 paper where she describes Relicta. She has some very clear photos and descriptions of the differences between these. But what's very interesting is if you have a look over here at the top right-hand side of the cap analyses, those blue triangles and the green squares, those are the same species. Those are both Aurelia from different populations, and they exhibit the same, almost more morphological variability sometimes than some of the other species that we examined at the same time. Not to say they couldn't be distinguished. They still fall, both groupings fall outside of the other species in the area or in the graph, but they display a huge amount of variability. So if we look at species delimitations, we know at the moment we have nearly 70 species concepts. That's not particularly helpful or useful for us a lot of the time. There's no unified species definition, and Alan and I were talking about this the other day. Part of the reason is that some species hypotheses and species concepts may be in complete opposition to others. And then I was reading some discussions on morphospecies. I went back to some of Mayer's work, and morphospecies were never meant to be, it was never meant to be a species concept itself. It's representative, it's diagnostic of a species. It's not meant to be a species concept in its own. And that comes back to what a lot of us are pushing about integrated taxonomy and integrating multiple lines of evidence. So when we talk about species, we often look at what we have with us at the time. And that can become quite confusing when you're doing species descriptions because species diverge over time. We know that this isn't a set in stone result that we're seeing at the moment. Things grow. This is probably one of my favorite pictures in science, the tree of life, looking at divergences of organisms. This is Darwin's original draw of the tree. And so we know these things happen. You look at, in the jellyfish context, we've got huge divergences in Aurelia over large geographical positions, and they are quite distinct at local level. We know that our environments affect species, so we've got Darwin's drawings of finches. I'm on a bit of a Darwin trip at the moment. But we know that these adapt and evolve for ecological niches. I don't know if Darwin would be particularly happy with me using his discussions in the concepts of species. He seems quite judgmental of the species concept. And so then we look at, all right, case of the West African cichlid. So in the space of time that humans and chimps diverged from our most recent common ancestor, there have been more than 700 divergences in West African cichlids. These fall within 3 major lakes in Africa. And what's quite interesting is genetically, we see interbreeding between a bunch of these populations. So we're getting niche occupation. We are getting specialized species evolving, but we have seen fairly recent interbreeding between populations. And then we look at work that one of my colleagues did, a very brilliant colleague of mine, we look closely to home for me and Chrysora. So we have 3 or potentially 4 species along the west coast of Africa, but I find these very interesting because the ranges for Chrysora fulgida at the top and Chrysora galensis at the bottom are actually very, very close. In fact, at the extreme ends of their ranges, they overlap. Yes, we know that we get huge variation in color morphs and structure within these species. But if you look at those 2 species, they are fairly distinguishable. Now, the map probably belies how close these populations are, but I can, within the space of half an hour, dive in the home range of Igalensis and dive within the home range of Fulgida. In fact, I cover those ranges on my drive to the lab quite in the mornings. So then you ask, well, how diverse are those environments? So anecdotally, if I dive in probably closer to the home range of Igalensis here in False Bay, I can dive in water of about 18 degrees. And if I cross over the Cape Peninsula to the start of the Benguela upwelling region, which is where we find Fulgida, on the same day, I can have 8-degree water. So Agalensis is found in an area that we sometimes term the Goldilocks zone. So we have, as Delphine pointed out, we have the Agulhas current running down the coast here. It's a warm water current. We have the Benguela upwelling system, which runs up the west coast of Africa here, which is a very cold but nutrient-dense current. And what we have happening here in False Bay is eddies of warm water splitting off that then mix with this nutrient-dense water, and we get what's called Goldilocks zone. But that's a slight digression. These species sit in fairly different ecological areas, which may be, and I've discussed this with Verena, may be part of the reason that we are seeing a divergence within the species. So with all this being said, where do we go from here? And this is probably the part of the talk that's going to make some people uncomfortable. My argument is that, that depending on what you want to do, that answer differs. So one of my favorite lines from a taxonomic paper comes from a paper that Mike and Lisa wrote a while ago, and it basically said that taxonomy is an expression of the human need to classify things. And so when we talk about species concepts, we really need to look at the use of those species. So if you're a conservationist and all you're looking to do is conserve an organism in your area, the morphology, if it's being defined by the area, is probably good enough to go on when you're identifying a species. If you want to look at whether an organism has invaded an area as an invasive species, you need to delve a fair amount deeper. If you're an ecologist, you need to make sure that what you are identifying is what you say it is, and so you need to put a lot more effort into your species descriptions. If we go one step further, we look at taxonomy, which is where I based my original philosophies on. And there we have to be certain. Mike spoke about making the boxes and then making sure we fill them with the correct things. We need to do this properly, and that's where the strengths and the development of integrated taxonomy is very useful. In that case, I am looking at developing a machine learning model that will distinguish these species without the introduction of human on a morphological pattern, on a morphological level, so that we can then run genetics on a priori groups without having to make those decisions as humans. Our decision making is often fallible. If you're an evolutionary biologist, I think you should be telling me how to do this. It's your job to discern these sort of things, and we should be borrowing information from other fields like evolutionary biology when we make these decisions. And with that, thank you for listening to my talk.
Great, thanks, Michael. So we have about 3 minutes for questions. All right, so tell me a little bit about your AI thing. So that seems to me like it's going backwards.
So the idea is not to make decisions based on it. The idea is that so if we go back to, well, the slideshow is gone, but if we look at the CAP analysis we do, we need to group those. We often group species based on, so we can use genetics, but we often group species based on what we know. So we'll say, yeah, these look similar. We sample them from the same locality. The idea is to go, okay, cool. I take all this morphological data, use an unsupervised machine learning model to group the patterns, and then you look at the genetics of those patterns and you decide what it's actually showing you. Is it population level? Is it species level? Yeah, so that's the idea.
So you think that AI will not have the foibles that humans have? Or because that implies that there's actually a signal that is incontrovertible in the morphological data, right?
Yeah, so it at least implies that we are able to split and group based on morphology, which isn't always the case. But it's a nice way of just removing one step of us putting an opinion in before we have enough information.
All right, thanks. Additional questions?
It was a nice talk, Mike. Thanks. Thinking about the morphoplasticity, we also usually use one life stage to identify the species, for example. How would that affect this use of AI to use the morphology, in the sense that would you need to wait? For example, Joanne presented a very nice case that we have usually in hydrozoa, in which the morphological plasticity may be restricted to either the jelly or the polyp, and then you cannot identify the other one. But I guess it would have to account a little bit into that, or I don't know. It's more, I have no idea how it could be incorporated. But
No,
what do you think?
I mean, you're absolutely right. I guess the answer is, and we were discussing this with Lisa the other night over dinner, is when you do these comparisons, you always need to compare like with like. So I can't go and describe the Medusa stage of an Aurelia and then say, well, it differs from the polyp stage of something else without comparing like with like. And so we would have to do the same in this case.
Okay, all right. Thanks, Mike.
Thanks, Mike.
Okay, so next on my list is Galen Verhagen. Surprise. Who's going to tell us a little bit about drivers behind the diversity and distribution of a widespread midwater narcomedusae.
Hello, good afternoon. I hope you're all having a great symposium. I'm very excited to talk to you about deep sea jellyfish and the results of my previous postdoc at Jamstack with Google. They're 3 of the 5 co-authors of the results I will present today here in person, which is really nice after a long pandemic. So there's Dougal, Mayhill, and Alan Collins. So although this is a jellyfish conference, we barely talked about the midwater. I was really happy to see Atola this morning, but actually the midwater is the largest biome ecosystem on this planet, and it's also dominated by gelatinous zooplankton. It provides many ecosystem services like fisheries or the carbon palm. But the reason why we didn't see so much deep sea talks is not surprising, because there is a lot of difficulties that comes with sampling these enormous ecosystems. So we're not only speaking in terms of volumes, but also huge pressures. As a result, it's also very expensive to sample. And in terms of analysis, you really have to think it 3 dimension. So can we therefore do more than just descriptive research? Can we do more than just say, look, we saw this nice jellyfish, it was in there? The answer is no, so thank you for your attention. No. No, there is many ways, and today I will talk about ecological niche modeling, but if you're interested in more genetics techniques, come to my talk. So what's ecological niche modeling? So this was a technique developed mostly first to study terrestrial environment. And what it is you collect some presence data, so latitude and longitude data of your species. You combine it with environmental maps that you're interested in. And then the model will calculate the ecological niche of your species, so which temperatures they prefer, and then we can project it again on the maps to find locations where there's a high probability to find your species. Now, about 10 years already, since thanks to Bastian Bentlagen, we are able to model also in 3 dimension in the oceans, which is unfortunately not yet much applied. So I'm here to say, please model in 3 dimension. And this is possible thanks to maps on environmental conditions that are now available in 3 dimensions. I use the one from World Ocean Atlas, but there are much many out there. So you basically decomposed in these different layers, but you still model in one big model, and then you redecompose afterwards your maps. And then in terms of presence data, you need also data in 3D, so latitude, longitude, and depth. Today, I will present a case study about the Narcomedisa genus Solmissus. Why Solmissus? Because it's widespread, but since it's a Narcomedusae, it is also feeding on other gelatin zooplankton. Therefore, it has an important trophic role, as it's a top-down regulators of the midwater gelatinous web. Okay, so what happens when you start your postdoc on deep-sea jellyfish in January 2020? This soon becomes your office, and now you have to find data for your postdoc. Where do you go? So luckily, there are some biodiversity databases out there, such as OBIS or GBIF. If you're lucky, there will be information on depth, which you need for your model, but none of them will come with a picture, so you cannot quality check at all. When it comes to deep sea imagery and videography, luckily there's much more databases out there, such as Jamstack or MBAR's PhantomNet, SeaTube, and more now of this expensive deep sea expedition will broadcast live their RV videos. So it makes it much more accessible to everyone to get this data. So my first step was to check which kind of data to use in my models. So I compared, on the left you can see a map using data from OBIS, with just the green points were the ones I had depth information that I used. And they are much more widespread than the ones I got from the deep sea databases that I could vet it, so which means I personally checked every image to make sure it's a Solmissus. And there, over 90% of the data comes from North Pacific, because institutes like Jamstack and MBARI are studying the midwater there, but other locations will focus mostly on the benthic and forget there is a whole ecosystem on top. So I modeled then the genus, and so on the left for OBIS, on the right for the Veta data, and in 3D, so we went from 200, 400. Warmer colors means higher probabilities to find Solmissus. And what you may have noticed is, so the colors or the distribution that was projected reflects the data that we put in, but how did these models perform? This can also be calculated thanks to a statistic called the continuous Boyce index, which goes to -1 to +1, where +1 is your perfect model. Will always find your jellyfish wherever it is, and like around 0, it's not better than random, where you just have thrown some points on the map would be equally good. And as a rule of thumb, if you have a CBI that's higher than 0.7, you have a highly performing model. And so for the ones with OBIS, we had an average performing model, but we had a very good score for the vetted data set. So here we can clearly see garbage in reflects garbage out, even though, well, I hope we'll have more data from other locations as well for the vetted data set in the future. Okay, a bit more about the genus Solmissus. Oh, the time is going fast. Okay, so let's see. Say there were 3 accepted species for now. Before we started, included Solmissus incisa was a widely distributed species, and it was suspected to be a cryptic species. And with preliminary data in 16S, we confirmed this. And one of these cryptic species is likely Somysus plicae, which has more than 28 tentacles. And when we look at where these are distributed, so on the right were the 28 and plus species, and on the left, we could see there was a division along the depth gradient with ones with less than 28 tentacles around 400 meters, and the other one around 800 meters. And this was also reflected in our model. So the way that we looked in 3D, we were able to also see there was some different niches, although there is some overlap in 2 dimensions. Now, very important when we are interested in climate change is looking at what are the environmental factors that are limiting the distribution of deep sea jellyfish. And here all our models, whether on genus level or on morphotype level, found that dissolved oxygen was the most important limiting factor, which you may find surprises. But when we looked at really low concentrations is where you found the most of our jellyfish. And because I had checked all these pictures, I could also work on the reproduction of Narcomedusae, which doesn't have a polypentic stage. It will produce jellyfish that will be produced by these oocytes. And we found there was no seasonality. We found that polyps with these visible oocytes all year round, and reproducing jellyfish were found in slightly warmer waters. So that bears the question, will these deep sea jellyfish be a climate change winner as with slightly warmer temperature or very low dissolved oxygen? They're doing very well. So what can we conclude? I think I cannot stress enough the importance of online databases when it comes to do deep sea research when dealing with such a huge ecosystem. But please include some images or videos that we can check the quality of your data. Then we found that Somysus indicta is likely a complex of cryptic species, and that there is some differentiation along the 3D axis, and that Somysus was reproducing all year round. And likely with the upcoming climate change, it will be favorable for Somysus, at least just for environmental parameters. We didn't look at any biotic interactions. So thank you for your talk, and if you want more details, you can check the papers or just ask me. Many thanks.
Okay, thanks, Caroline. I think it actually turns out that we have lots of time.
Oh, we do?
Because I don't know whether that reads properly or not on the background. Okay.
It says exactly 12 minutes. So.
Yeah. Okay.
Okay.
Yeah. See, the bit of the 4 doesn't work on this side.
Ah.
So it was 8, not
You
6.
tell me that now.
I didn't know. All right. Okay. So questions for Gallian.
Hi, wonderful presentation. I just would like to know, you say you have mentioned that it reproduces throughout the year. So throughout the year.
Yes.
So what will be the maximum number of density that we see in the midwater for this particular genus?
The maximum number of?
Yeah, density.
I didn't understand.
Individuals. Number of individuals that we see in the midwaters.
That's hard to say. On a world map now, it's like roughly 2,000 data points, but it's also vary from dives where we can see. We had some Falcor dives where we maybe observe 5 when we go 1 gradient. Sometimes it's 1, but my data was mostly based on RV. So and when maybe other ways of getting data may get more information. So I really worked with presence data because it's really hard to quantify because of that. Yeah.
Okay, thank you.
No.
Being on the author list, I don't know if this is allowable or not. So you stressed the importance of databases for occurrences that include images. Do you have any suggestions where people who may have images and occurrences could put their occurrences so that
people like you could do modeling on them? Where would you suggest they?
Which is not necessarily deep sea. That's a good question. Do you have an idea in mind? Or I know the greater public, I know you like iNaturalist, which is just for the greater public, but for scientific data, it would be nice to have like one big database. For genetics, we have bolts that now requires pictures for genetic data. And if we're able to do that as well for biodiversity data, would be great. Yeah.
iNaturalist, you cannot put a depth value in. It would have to be as
Ah.
text, so not so useful.
We should
But
email them.
since we know how important genetics are, if you put it on like barcode of lifey kind of places with an image of the
Yeah.
actual animal.
So bolt is a really good one for the genetic there too, yes.
Yeah. I'll just make one comment. So there's also a database called Geome, I think Geome. And so
Okay.
they're trying to work out how to curate all of this, like genetic and non-genetic data. So that might be worth a look to.
Good to know.
Yeah, Geome.
Why all the co-authors asking me questions here? Are you discovering the paper?
You were encouraging us to think in 3D, and I wanted to know what you're going to model next. Like, do you have taxa that you're interested in tackling next?
Yes, so my current postdoc is now dealing with Antarctic jellyfish, so more polar. And this time I have money, so I will do some genetics and eDNA as well. And then hopefully I'll be able to model in the Sulfur Ocean as well, which is, I like hard to reach ecosystems apparently. But then, yes.
Okay, so thanks, Geralian. Fabulous talk. We don't have time for another question. Sorry. All right. Okay, so our next speaker is Dougal Lindsay.
I'll use my timer on the phone then. So, in Japan, we like bathing in hot mineral baths. And today I'll be talking about how that relates to jellyfish. List of my co-authors here, many people who are in the room here, and it's a collaborative project where we really need everybody to have brought all of this together. So exactly what would happen as the global ocean warms? People say it might be more jellies. Some people say, no, not really. We don't know yet. But all of these predictions have been based on lots of data. There were 37 data sets went into the figure in that paper by Condon et al. On the right. For the deep sea, this is distributions and species occurrences in 3D, just as Gerlin was saying, and there's not very much away from the coastlines and not very much in most of the midwater. The figure on the right is a poster child, Aegina citrea, 4 tentacles, the only Narcomedusa with 4 tentacles. Therefore, that was what they used for their distributional model to see how things will change by 2050. And that single species actually included 7 different cryptic species in different families as well. The paper's at the barcode there, the QR code. And so a lot of the data on the distributions was actually wrong. Probably less than 10% of those points were actually Aegina citrea. All right, so what do we do about that kind of stuff? This is the proposed mesopelagic ecoregions of the world's oceans. So ideally, we'd want to go to each of these ecoregions and see how they're different. The actual ROV-based gelatinous fauna diversity data that we have for more than 2 dives are these little smiley faces. And you can see that we don't cover very many of the bioregions that we would really want to look at to model worldwide, because these distribution data modeling gets highly skewed by having too many data points in a very well-surveyed area compared to non-well-surveyed areas. So the 2 places where most of the information come from is the Monterey Bay, where Ambari have been
doing ROV dives for decades and off Japan. And here's a comparison of what the 0 to 1,000 meters looks like. And you'll see with Monterey Bay, there's lots of siphonophores in the shallower layers, and the Medusae are a bit deeper. Whereas you see with the Sagami Bay, we had the Medusae were shallow and the siphonophores were deep. So we're basing our what we know about the mesopelagic around the world as we see it with ROVs based on these two places, which are both very different to each other. I know et al. Did work in the Mid-Atlantic Ridge data, and they have species occurrences list, but with nets, it's very hard to get relative abundances because some animals are broken up and don't survive, whereas others are. So you really need to be working with ROVs as much as you can. This is bathypelagic from 1,000 to 3,500 meters depth in Monterey Bay on the left. The green is a unsegmented polychaete worm, Piobius, in very, very large numbers. On the right is the Clarion-Clipperton zone, where we've recently been doing environmental impact assessments for deep sea mining. The pie graphs, the red lines there are denoting the gelatinous fauna, which are not worms. The worms are in pink. And these depths, 1,500 to 2,000, 2,000 to 500, to 500 and below. So you see they're very different, again, between Monterey Bay and the Clarion-Clipperton zone, which are the only two places where we have abyssopelagic gelatinous fauna information. So what do we actually know? It's not very much. Is there anything we can do about it? How can we model future scenarios when the data is so limited? So our idea was to use submerged calderas as proxies for a global warming ocean. And off Japan, we have several volcanic undersea mountain ranges. And the first place we looked at was the Kurose Hall, which has 110 meters is the shallowest point. It's 820 meters at the floor. And the other place is the Smith Caldera. And we did dives inside and outside, so at the same latitude, longitude, pretty much, we were able to compare the fauna there. The Kurose Hall here, as you can see, the temperature outside is 4 degrees down at 800 meters because we have the cold Oyashio-derived waters is coming in from the north, whereas the Kurose Hall, the rim, stops these cold waters from coming in and it's geothermally heated from below. There's no vent, but it's warmer at 11 degrees at the seafloor. This was in the year 2000. And what we found was inside the caldera, there was a very low diversity compared to outside, but there were many more animals there than there were on the outside. And the most common animal inside was this one animal, Illyria bruni, used to be Forsteria bruni. And on the left there, the bar chart, you can see that it occurred mostly around the seafloor. And there was not a single animal seen on the outside. This was actually originally described from waters east of India. And this was the second record ever inside a caldera, with none outside off Japan. So we revisited the Kurose Hall 20 years later just to see how things had changed, if the jellyfish was still there. And we found, no, we couldn't find this Illyria bruni in there anymore, and instead the whole caldera was full of Corleotis sloni, the viper fish. In one video frame, you could probably count 30 or 40, and it's the highest concentration ever seen on the planet. Again, the caldera has warmed up in that time from 11 degrees at the bottom to 18 degrees now. And we're going back there to see what's happened in February, March, to go and see if it's even warmer, and if the viperfish are there or the jellyfish have made a comeback, or if it's going to erupt and wipe out Tokyo. So, and this is the Smith caldera. The Smith caldera is also warmer inside, only 10 degrees versus 5 degrees outside. And the lip of the caldera is deeper than the Kurose hole. It's at 400 meters or so. This is a comparison between the Kurose hole and Smith's caldera. And we found that the diversity was highest on the outside of both calderas. Next highest diversity was inside the vent caldera, and then the lowest diversity was inside the non-vent caldera. Whereas the population density was the opposite, with the inside the non-vent caldera had the most animals, and then inside the vent caldera and outside the calderas, not so many. Interestingly enough, inside the Smith's caldera, we also found large numbers of Olearia bruni, associated at the same depth as the vent plume, possibly feeding on vent larvae or something like that, which was pretty cool. And of the ctenophores, there were many, many lobates. On the outside, mostly in shallow waters, and on the inside of the caldera, very large proportion of lobates down in the deeper areas. The lobate morphotypes that were abundant inside the caldera, there was an undescribed family of lobate there. And the warm inside caldera community was very different to the cold outside community. Shameless plug here. This is a new subfamily, new species genus subfamily of ulmarid medusae, which was just published on Monday of this week. And this is also only found in the Smith's caldera. So far, we've seen 3 individuals, all in the Smith's caldera. We've done hundreds of hours of dives in other places, and we've never seen it anywhere else. So keep your eyes out and tell me if you see anything that looks like this. The barcode will take you to the press release. And it was very strange because St. Georgia, this Medusa, was closest genetically to different subfamilies in the ulmarids of jellies that don't have tentacles on their bell rim. St. Georgia also does not have tentacles on its bell rim. They're actually under the subumbrella on the surface. We probably should have erected a new family, but we didn't have good enough genetic data to erect multiple families instead of putting everything in the Ulmaridae, so we've just placed it there for now as a subfamily. Okay, so what are the other target areas that we can go to? We've done what we can around Japan. So we look at our biogeographic region area, and we see some interesting places with warm water temperatures, the Mediterranean and the Red Sea, of course, but also the Sulu Sea, which is a sea which is surrounded by Malaysia, Indonesia, and the Philippines. It also has very shallow rims. Inside this sea, 10 degrees Celsius down at 5,000 meters, and it has less saline but warm waters down in the deep areas. The Hakuhomaru, which is the University of Tokyo ship, went there and sampled with an INS net, no ROV, unfortunately. But in the Celebes Sea, the station in the Celebes Sea had lots of classified siphonophores, the rocket ship siphonophores, that are big enough that you can also see with an ROV, so it's a good target to be looking for. But they're very common and presumably important in the deep sea outside these kinds of enclosed basins. We didn't catch any in the Sulu Sea, so a whole family of siphonophores was missing. There were lots of Anthomedusae in the Sulu Sea where we didn't see so many in the Celebes Sea as well. So again, very different, even though they're very close to each other in terms of geography. So it wasn't just the classifieds that were absent. There was only 1 out of 5 species of the multistriate Lensia siphonophores. Deep sea Lensias were missing, Gilia reticulata missing, siphonophore Frillagalma, Botryonema brucei was missing, but there were some deep sea cnidarians that were there. So it's not that deep sea cnidarians can't live there, it's just that taxonomically, some taxonomic groups are unable to live there in the deep sea when it's warm. Is it because there's physiological barriers or not? We're not really sure. But in the case of the classifieds, we have some information from the Mediterranean as well. And we did some surveys there with the group from Barcelona, ICM SEEK, in 2013, the fish jelly cruises. And we did find one classified there. It's the basal group for the classifieds. The genus Kephis was there, but no others. And so we have a list of animals that are missing in the Mediterranean as well. We look at this on a global scale, and we see in the warm waters, whether they're hot warm water, whether they're high salinity warm waters or whether they're low salinity warm waters, there's various taxa that are missing. The one hole we have in the data there was the Red Sea. So we teamed up with KAUST and took their ROV out to do some surveys in the Red Sea. We found Salmonella bitentaculata, which is found in the Arctic, the Antarctic, Southeast Asia, absolutely everywhere in the Red Sea as well. So there's probably more than one species hidden there. And we've got a proposal in to go back with a big oceanographic vessel and an ROV from Jamstec. So keep your eyes on that space. So what else is it that we can do here? Monterey Bay has an amazing data set. The bar graph on the left is the Clausifiidae that they have in their data set. And on the right is Botryonema. These are the two taxonomic groups that are missing in the warm, deep waters. Unfortunately, for the small cryptic taxa, they're very difficult to see with ROVs and even more difficult to identify to species level. So it's a total number of 13 versus hundreds and hundreds for those other larger animals. So we really do need more data, which will have to be caught with nets or shadowgraph cameras, perhaps, to get some of this distribution data in other areas for the smaller animals. But in the case of Lencia havoc, in Sagami Bay at least, it's a very different distribution to that found at Mbari. And as we've heard, we really do need to get genetic data on some of these animals because sometimes you can't tell them apart morphologically, but they may be different species. And it looks like we have cryptic species for some of these small animals as well. Unfortunately, no sequences for any of the Monterey stuff where most of the distribution data comes from. But I'd really like to get this genetic information. The next areas we'd like to look at is the outflows from the Red Sea and the outflows from the Mediterranean Sea. So if anybody's going there on a cruise, can I come along? Thank you very much. If there are any questions.
Right, wonderful. Thanks, Dougal. I think we're got the 15 minutes in there. Yeah, yeah. So catch Dougal later if you have any questions about really interesting set of observations. All right, so our final speaker for this session is A.B. Kumar.
Hello, good afternoon. Good afternoon, everybody. And probably to end the session, as I wanted to just tell you a small story about citizen scientist involvement in documenting biodiversity in India. Throughout the world and including in this JBS7, there were several presentations, including posters on involving citizen scientists in documenting jellyfish diversity across the world, monitoring, ecological interactions, and as well as even in genetic studies. And let me start with the 10 commandments in citizen science programs throughout the world, and whether this presentation fits into that. And one is that, we involve them in all these scientific endeavors. And second, it should have a genuine science outcome. And third, the professional scientists should involve the citizen scientists, and there should be mutual benefits. And the fourth, they participate in multiple stages in the whole process of research methodology, right from framing the hypothesis to finalizing the results and conclusions, and whether they receive feedback from the scientific community, and that's the fifth one. And the sixth one is the citizen science is considered as a research approach like any other. The whole process involved the research process. And seventh one is, the data and metadata is made available publicly in the form of a research paper later. And the eighth one is citizen scientists are acknowledged in project results and publications, and that's another component. And citizen science programs are evaluated for their scientific output, data quality, participant experience, and wider social and policy impact. And the final commandment, which is again, all these commandments were proposed by the European, the Citizen Science Association. And there are whether some legal or ethical issues are involved in this. And we have seen in the last few days there are large number of successful projects across the world involving many citizen scientists groups, starting with the Jellyfish to many other. I am not naming all. And all these things are involved. And when you look at these successful citizen science programs in documenting jellyfish diversity, we would understand that that is highly skewed. And there are only two stronger representation from Asia, and one is primarily by the Israeli scientists, and the second from the Hong Kong group. And there is no much representation elsewhere. I just wanted to tell you why it is important, and especially in Indian context. And I start this story when we involved a citizen scientist, and his name is Umid. Umid, are you here? Yeah, Umid is here. And I wanted to also to acknowledge him because most of the photographs you see in the photography exhibition is taken by Umid. And actually, I don't know him earlier, and then we were actually involved him in installing a hydrophone to locate the movement of whales along the Kerala coast. And he was the person who was helping us. And then when we started launching this program JBS7, I asked him to give some good photographs, which we can use it in the website as well as in the brochures. And then he gave the photographs, and then we started identifying them using he dive across the world, in fact, and then some of the dives in India provided some good quality photographs, including the amphibious photographs which you see there, but that's not from India. And the first one is the dive from Andamans, and Andamans from the Andaman Islands, as you can see there on the map there. He photographed this Aurelia species, and when we started analyzing along with the Rias, and we found that the structurally this species is slightly different. And because in India we had Aurelia aurita and Aurelia solida reported earlier, but this animal has some strikingly different characters, particularly in its lappets, and they have more than 8 lappets, more than usual lappets, and that led us to some molecular work which is now going on. And probably the species which bloom, as you can see in the beautiful photograph by Umi, is that this could be a new species which is seasonally bloom in Andaman waters. And this is another species. Again, the photographs, the quality of the photographs is so high. Messagius papua, and this is from the Pak Bay in Tamil Nadu. And also some of the a series of photographs. I'm not showing all the photographs. This series of photographs also tells us several stories regarding the ecology of this species. Particularly, it's the movement patterns and in the seagrass meadows in the region. And again, some of the very interesting things which probably we have to work on later. So all these photographs actually provide us some of the topics to work on in future. And this is Lobonima smithii, and this is again the species which occur in the coral reefs ecosystem in the Pak Bay between India and Sri Lanka. And this is one of the largest species, and you look at the because we are impressed by the quality of the photograph, and the species is Ropilima hispidum, and then if you look at the in detail, we could study the ecology as well. You know how the carangid, especially the Alepus jabada, and how they are associated, their number, and the way in which they associate with this organism, etc. So that is another interesting feature. And when Catacylus was photographed by Umid from Andamans in India, and this is again it could be the Catacylus mosaicus, and this is a new record to Indian waters, again from the citizen science photograph. And next beautiful photograph is Crambion mastigo fora, and this is again from Andamans. Again, no record earlier from Andamans. Andaman is again a coral islands represented by more than 360 islands in Indian region. And this is Saina Nosaki, again by another fellow diver, Jonah, shot from Tamil Nadu coast, from Trivandrum coast, very close to that. And yesterday I've seen some of the social media postings by Stamper, where he also recorded the same species from some of the dive sites in Trivandrum. And some of the photographs which is exhibited here will also provide us some of the very interesting associations here. And this is Sainia Nosaki with the closely associated brittle star. Ophionemis marmorata. And this is a photo by Daniela, a fellow of Umid. And there is a specific reason why I showed this photograph here. And many of the species which are documented as Sainia nosaki from India are not Sainia nosaki. And this is the Sainia nosaki which she photographed from Maldives. Is it Maldives, Daniel? Yeah. It's from Maldives. That means, this is the species which is not present in India, but it is present in the Maldives. And so the involvement of the scuba divers in documenting the photographs as well as cataloging biodiversity along the Indian coast is very important. And I come with some of the suggestions, and primarily we need to involve the practicing, to help the practicing taxonomists, who are very few in a larger country like India, especially if you look at the jellyfish documentation in India. In still 1950s, there were stronger records of documentation, and only in the last one decade, the people again started working on it. And in many of the hydrozoan groups, there are no experts in India as well. So this is a time where we need to involve the citizen scientists in documenting diversity primarily, particularly in a context when the scientific funding agencies, and also I'll tell you that in India funding agencies are primarily the government agencies. Normally throughout the world this happens. The funding agencies, if you apply for a space science project, you will get it. In nanoscience, nanotechnology, you will get it, but for the basic sciences, it's hard to get money for running the projects or surveys. And though this is the only way in the post-COVID world we can involve citizen scientists, including as parataxonomists, to collect data and help the taxonomy community here to document the data strongly and build a stronger database over a period of time. And also, there are few departments, including the marine science departments, which work parallelly, and there are a lot of duplication of work. And I recommend again that this kind of duplication of work can be avoided by strategically planning marine exploratory surveys. And there is a stronger need to develop a group of youth primarily trained and in or skilled in taxonomic exercises, and that could be the aim of the workshop which is coming after this conference. And also we need a stronger the local field and survey manuals in this group. And also we cannot work in all the areas together. We need to prioritize the hotspots, especially in the knowledge gap areas. And finally, there is an example of involving the citizen scientists in documenting biodiversity in India. This could be one of the best models as far as India is concerned, and that is eBird. And when eBird was launched in India in 2014, and a lot of passionate birdwatchers joined it, free of cost. And it is now after 5 years when they initially published the data, and currently there are more than 1,300 species covered, and more than 1 million of checklists available from each and every part of India, a larger country like India, and more than 200,000 photographs and the calls recorded by various citizen scientists across the world, and more than 10 million observations in all. So this is a typical or classical example how we can involve citizen scientists in documenting biodiversity, but that is not happening in the marine environment as far as India is concerned. And my appeal is that it can happen in the marine side as well. And some of the international communities and groups which are already documenting it can collaborate with the Indian partners so that the documentation process can be can involve large number of scientists as well. And the reason is very simple, because we have a lot of technically trained, especially in IT and other sectors, and most of the youngsters in India are trained in IT because they get job there, and then they are also passionate in biology. So once we involve them and assign them with the different duties, including in cutting edge research involving jellyfish, and we can have a stronger database to present in JBase 8. Thank you.
Okay, we have a couple of minutes of questions for Bijou.
Sir, it's a very wonderful presentation, sir. I just would like to know, some of the photographs seems to be taken, I think, after sunlight it goes off. Is it right?
This is the aesthetic part of that, and probably Umeed will answer to that. Umeed is a photographer there. You can stand up and then yeah.
In the latest was probably at about 3:30 or 4 o'clock in the afternoon, not later than that. So it's just what I've done with the settings and the light.
Okay. Yeah. Thank you. Nice photographs.
Thank you.
Okay, an additional question?
Sir, success of the eBird is they were able to approach the correct audience. Birders were there. Now we have such a big fishing community. I think we should involve them, and that would give us better data sets.
Agree. I fully agree. And also, especially in the fishery sector, especially if you really wanted to collect the bycatch data, actual fisheries data.
And they have good knowledge on local names. Maybe they might not identify scientific names, but we are not utilizing the potential that we have.
Yeah. We have, in fact, CMFRA, and we have documented the traditional knowledge of the fishers in particularly with regard to the jellyfish as well.
Thank you.
I think we will. Question the time. Perfect. Thanks. Okay. Thank you, Bijou.
Thank you.
All right. So that's the end of our talks for this afternoon. Do you have any? Okay, so we have a break. Oh, yeah, okay. Okay, so I think the announcements that I have are that our Illustrious speaker from CMFRI is in another meeting with Delhi at the moment, so will be with us in a half hour. So we're going to go do the group photograph first, and then take tea break, and then we'll come back here and pick up with the special talk. A couple of announcements before we do that is if you kindly volunteer to help evaluate posters this afternoon, let's meet here after Dr. Gopalakrithnan's talk. And if you're presenting a eulogy or an appreciation this evening, please upload your presentation with our IT friends at the right. Okay. All right. So I think exit. Sorry.
We have two mementos to bring. I request Professor Padmanabhan to give the mementos to the co-chairs. Lindsay, please. Professor Jayachandran, sir.
Okay. So now break for a photograph. Okay, all right. Sorry, hang on a minute. Where are we going? The steps out here? Yeah, okay. All right, so if you can please head just outside the dining hall towards kind of like the stepsy area, and we'll get organized over there. Photograph, then break for tea break, then we're back in here. Yeah, so back in here around 4 o'clock. Okay, thanks.
let Grammarly rewrite it. Grammarly makes your message clear.
ladies and gentlemen, esteemed colleagues and distinguished jellyfish researchers. I'm scared it's not a session, Srinath. Just 5 minutes. I'll spend only 5 minutes, followed by a release of a book. Then about me, myself, you can call me Gopal, Gopal. Gopal, I'm Gopal. Full name is Gopala Krishna. It's a long name. I'm the director of ICAR, Indian Council of Agricultural Research, Central Marine Fisheries Research Institute, CMFRI. So one of the collaborators in organizing this JBS, Jellyfish Bloom International Symposium 7 2023. You can see the logo, that fish logo is, it says, I'm the director of the institute. The headquarters is located headquarters of CMFRI is located in Kochi, almost 220 kilometers north of this place, a coastal city in Kerala itself, the state itself. And we have stations all along the Indian coast. That is from northwest and southwest, southeast, then northeast, and the island ecosystems. So that is covering almost 8,118 kilometers long Indian coastline. So the strength is like I'll tell you, this is the largest fisheries institute in the country, and we have a station here as well. It's very close to this venue. We have a station. It's in Vizhinjam campus, where the head is also with us. And the mandate of the institute is monitoring and surveillance, like we assessing the monitoring and assessing the marine fishery resources of the Indian exclusive economic zone, and also studying the climatic and anthropogenic changes, and developing sustainable marine fisheries practices. So as a policy advice, see, we generate based on the biology and regular stock assessment and landing estimates, we generate policy documents, and which are to Government of India, that's the federal government, and to the coastal state governments. So second is to augment fish production. Like Indian fish production is somewhere around the potential is somewhere around 5.31 million metric tons from the seas around India, the harvest fisheries. So out of that, we harvest slightly less than 4 million metric tons per year. So this is not adequate for the ever-growing population of India. So we have to augment the fish production. So the second mandate of the institute is to carry out the mariculture research activities coupled with the biotechnological, genetic aspects and nutritional and the disease control aspects. We have to do the selective breeding and other things to augment fish production, and also to then document the marine biodiversity of the country. That is the third mandate. Then the fourth mandate is act as a geospatial geospatial repository of information. This is this is the largest, not only the largest, but also the oldest fisheries institute also. This institute was established by the British in 1947, prior to India's independence, on 3rd February 1947. And so we are 77-year-old. So this institute is 77-year-old. So we have a very strong database of all the commercially important species of more than, I should say more than 1,200 species, biology, stock, all distribution, all data of different species, plus the species available in Indian waters. And the scientists, the staff strength is, I'll tell you, it is 167 scientists. 167 scientists plus director, and PhD scholars will be around 150. So 122, why I said around 150, 122 PhD scholars are there in headquarters in Kochi itself, and rest are distributed in the different regional stations. So this is a mandate of the institute, and actually the workshop was jointly with Dr. Biju. It was planned, supposed to be in Kochi. But the last minute, Professor Mike knows the difficulty. Last minute, we were thrusted upon with a major event of 16th organizing a 16th Agricultural Science Congress, just last month, and the total number of participants were more than 2,000. So I was the organizing secretary. So I had this venue was to be changed, but we extended all the support. So just now, because the parliament is also going on, so I am supposed to be there in my office. I am not supposed to travel because the parliament session starred questions, all member of parliament questions will be there. We are not supposed to travel when the parliament is on, when it's working. So from Delhi, always we are on hotline when the session is going on. So I am just leaving after this. So as we draw, this is about the institute. So you all are welcome to visit CMFRI when just visit. If time permits, at least visit I tell my head of at least this the city center to take you to. It's an excellent hatchery facilities, breeding center. We have a small, well established center is there in the city, hardly 10 minutes away from the venue. So kindly visit if time permits. So as we draw the curtains on this enlightening and inspiring Jellyfish Bloom Symposium 7, JBS 7 2023, held in this picturesque and heritage city of Thiruvananthapuram, Kerala, it is both an honor and privilege to stand before you. I understand that over the past few days, we have embarked on a remarkable journey through the complex and fascinating world of jellyfishes, exploring their interactions with humans and their role in our ecosystems and endless possibilities they present to science and society. This symposium has helped us to expand our understanding of multifaceted relationship of jellyfishes and human beings, highlighting both the opportunities and challenges. JBS 7 2023 also delved into bio inspiration and the venom regarding jellyfish venom, associations and ecology, biodiversity, and other intriguing aspects of jellyfish research. We also had a special session on jellyfish fisheries, trade, status, trends, and impact on livelihood today morning. But unfortunately, I missed most of you because it was a panel session. So I wish you all had attended that session as well. So on a broader scale, this symposium has been a melting pot of ideas, fostering collaborations and inspiring new research directions. We delved into topics ranging from jellyfish fisheries and trade to the potential impacts of climate change on jellyfish populations. The discussions were not just limited to the biological aspects but extended to the socio-economic and environmental implications as well, and the need for intensifying our responsibility in towards preserving the delicate balance of our marine ecosystem. So, as I conclude, I am reminded of the profound words of Rachel Carson. The more clearly we can focus our attention on the wonders and realities of the universe about us, the less taste we shall have for distraction. So just I quote her words. So looking ahead, the path forward calls for the continued collaboration, innovative research, and an inclusive approach that considers the intricate relationship between humans and the marine environment. Let us carry forward the momentum gained from this symposium, striving to make the difference in the world of jellyfish research and beyond. In closing, I extend my heartfelt gratitude to the organizing committee, speakers, participants, and everyone involved in making this symposium a resounding success. Thank you once again, and I look forward to crossing the paths in future endeavors as we continue to unravel the mysteries of the jelly world. Thank you so much. And this is followed by a book release jointly by the University of Kerala, and it's a draft this thing, and the PDFs will be. It's a very colorful book. Just the title is Jellyfishes of Indian Seas: Ecosystem, Economy, and Management. There are 12 chapters. So the draft one is it's being released today. May I request Dr. Biju, all of you kindly just come and just we'll have this release. So PDF, I'll assure that the PDFs will be sent to, I'll send to Dr. Biju. Dr. Biju will send the PDFs, soft copies of this book to everyone once there are 12 chapters in this, and once this is final, we'll be just sending maybe within 2 weeks, maximum 2 weeks. Thank you so much. Thank you for the patient listening. Thank you so much.
Thank you, Dr. A. Gopalakrishnan, for his insightful brief and address about our JBS 7 symposium and the book release also. So, as he said, this book will be a draft and we will be editing and publishing it soon. So now I think there is a poster session and you all can come back by 6:30. Reassemble here by 6:30. Okay.
Thank you. And one more announcement, and that is those who have missed visiting the craft village, it will be open only up to 7 o'clock. So don't miss it. If you have a chance, you visit the craft village as well. Okay. And those who are presenting the posters, you can be there. The evaluation will start now.
Good evening, all, and a warm welcome to all of you for the valedictory session of the 7th International Jellyfish Boom Symposium. So now it's time to draw the curtain for 7 JBS. Over the last 4 days, we have had deep insights regarding jellyfish fisheries, taxonomy, genetics, biodiversity, bioinspiration, and so on. I request Padma Shri M.C. Dattan, Professor Mike Dawson, and Dr. Srinath, scientist CMFRI, to be on the dais. Thank you. Now I request Dr. Srinath, scientist CMFRI, to welcome the gathering.
Respected guests, esteemed colleagues, and fellow jellyfish researchers, a warm welcome to you all on this momentous occasion of the valedictory function of JBS 7, Jellyfish Bloom Symposium 7. It is indeed a privilege to stand before you today amidst this gathering of experts, researchers, and passionate individuals who share a common fascination with this enigmatic world of jellyfish. As we gather here today, we bring to a close a week of enriching discussions, thought-provoking presentations, and fruitful collaborations that have delved into depths of jellyfish physiology, ecology, diversity, and social implications. Over the past few days, we have embarked on a journey through the mesmerizing realm of these gelatinous creatures, exploring their intricate life cycles, their diverse roles in marine ecosystems, and their growing influence on our human world. Jellyfish Bloom Symposium are recurrent get-together of the global jellyfish research community, which aims to bring all the jellyfish researchers all over the world to come together and deliberate on the development of the research in this field. This symposium, in particular, the jellyfish, the 7th edition of JBS, was a collaborative effort which was initiated by the International Advisory Board and implemented by Kerala University and ICR CMFRA, and it has successfully brought together 172 delegates from 30 countries, fostering a global exchange of knowledge and ideas. As we are about to conclude this symposium with this valedictory function, I wish to give a warm welcome to Padma Shri Madhavan Chandradattan, former director of Vikram Sarabhai Space Center and scientific advisor to the Chief Minister of Kerala. Dr. Chandradattan is a recipient of many honors and awards, including the Performance Excellence Award of 2009 from ISRO, the Outstanding Chemical Engineer Award, again in 2009 from Institution of Engineers, India, and the Individual Service Award in 2006, again from ISRO. He was also the head of the Launch Authorization Board of the Chandrayaan-1 project, which has finally yielded to Chandrayaan-3, which took India to the moon very recently. It was in the news. The government of India honored him in 2014 by awarding him Padma Shri, the 4th highest civilian award, for his contributions to the field of science and technology. In May 2016, the government of Kerala appointed Dr. M.C. Dattan as a scientific advisor to the Chief Minister. Sir, I welcome you to this event. I also extend a warm welcome to Professor Mike Dawson, University of California, who has been integral for planning and the conduct of this event as a member of International Advisory Board. Welcome, Mike. I must also welcome, though he is not in the stage, Dr. Biju Kumar, Head of the Department of Aquatic Sciences and Fisheries, University of Kerala, who has been instrumental as the organizing secretary of the symposium. Welcome, sir. Once again, I welcome all of you, all of the each and every members of this gathering. Welcome to this event. Thank you.
Thank you, Dr. Srikanth. Now I request Padma Shri MC Dattan to address the gathering.
Good evening to all. Most respected scientists, researchers, especially the organizers of Kerala University and CMFRI, who has taken a great responsibility of organizing and arranging such a wonderful 4 days program with a lot of international participants in the field of work. As already explained about my background, I am not a biologist, but I love jellyfish. I am from a seashore village nearby here called Varkala. So born and brought up by seeing the sea and also the fishermen getting the different types of fish, love the fishes, eat well fishes. And it was my very favorite item, the jellyfish. I'm talking about maybe around more than 65 years back in a childhood. Those days, the edible jellyfish, very rarely people purchase and process as a food item. People have a concept that this is some very bad creature in the sea which should not be eaten. But later, after some time, when in the market the price has gone so high, and people understood these are all getting exported, they understood it is a very useful, costly, nutritious food item. Then the market value has gone. That is the background. As explained, I am basically a rocket scientist for more than 43 years. And after that, I joined with the Chief Minister of Kerala and helping him while looking after all the scientific activities of the state. I started reading the life science after coming with him. And if somebody asks me now, which branch of science is most important, without any doubt, I will tell life science. Because all other branches of science and technology is for sustaining the life in this planet. No doubt about it. And nowadays, with all these kind of climate change, global warming, etc., every scientific activity is totally oriented towards sustenance of life. With that, not only this government has already started a park, life science park in Thomakel. A lot of institutions have started functioning there, like Virology Institute, now the Center of Excellence in Microbiome, and Center of Excellence in Nutraceutical. And, on the veterinary science research, lot of things are going to come there. That is the interest of the government on development and nourishing the life science part. On the particular subject of the jellyfish, when I discussed with some of the friends who are all retired professors, zoology professors, it's a very controversial name, the jellyfish. And one of the professor told it is not a fish. I discussed with Biju Kumar. Biju Kumar says that he is correct. It is not a fish. And one more professor, when I discussed, he told it doesn't have any organs. But it doesn't have any organs means fantastic performance it is having, then without organ. As again I told, Vijuma told, yeah, in a way it is correct. But what is an organ? What is an organ and how we can define? We say, we have organs, sensing organs and action organs. And added to that, there are internal organs which maintains the engine, the body. So like that, if you look, it's a very unique creature by the Creator. Unique in the sense, so lovely, so beautiful, so soft, colorful, excellent movements, mobility and motion. And if what not, so if the scientists have an attraction to go more and more details about this particular creature in the ocean, in the marine science, there is nothing wrong about it. I also got the book of abstract from Biju last day, and I am going through the complete number of papers. I think most of the area with respect to jellyfish has been covered by the researchers. It will be a very beautiful document, for others who come forward for a reference. The most interesting, other than the edible part of it, the most interesting thing which I found was this creature, the jellyfish, is an indicator of the marine ecosystem. Indicator, or you can call it as an instrument for studying what is going on in the ocean or ecosystem. It has got full of sensors. When I say sensor, we have in our department, in rocketry, we have a sensor development division itself. Because the rocket, what is going, you will not believe, thousands of sensors are embedded on the body of the rocket, and we continuously monitor the health of the rocket. So that sensors play a very major role. And this creature, the jellyfish, what are sensors it is having? It has got a GPS. And like that, the way by which it's a propulsive device. Propulsive device means the mobility. How fast it moves, turn, and sense the directions. In rockets and satellites, we have an oscilloscope. We call oscilloscope is position sensor. Position sensor not with respect to the global positioning, with respect to its own position with respect to the pitch, roll, kind of things. Twisting, turning. The rocket, when it moves, it should not deviate this way, that way. Not only going to other places, even from this original predicted trajectory, it should not move. So that oscilloscope is a oscillograph is a very, very powerful instrument we make here in our central line. Similarly, if you, for studying the ocean system and the changes what happened to the marine system, this jellyfish can be used as a small specimen, and you don't go to complete ocean, etc. You go to some selected area, some selected jellyfish, and you go on, the scientist can go on absorbing its nature, its movements, changes, and the color changes and all other things, that will give an indication about what are the changes going on in the marine system. Changes in the sense, maybe warming, the acidity, the biodiversity, or the climate change effects. Even more than that, It also does some very wonderful things like, binding or absorbing the microplastics, nanoplastics, microplastics, um, things which, we dump to the sea by different methods. This is like in water purification, we use coagulant for, coagulating the fine particles and etc. Here, this also done. So if you look into that also there in some papers are all there on this particular aspect of it. And then the biomedical advantage. As a biomedicines, what are things it comes, including the polysaturated fatty acid and omega-3 fatty acids, whatever is there, how it affects the cholesterol, reducing the LDL and increasing the HDL, what are aspects it does. So if you tell there are one after another, there are many things will come. And out of all these things, surprising to me was this creature has got a effect on withstanding the microgravity. That is the one which the space scientists make use of it for doing experiment in the space station. Generally, when the gravity changes, all kind of creatures, has to adjust with that, but difficult. But this jellyfish has got an excellent mechanism, well built inside mechanism, which can overcome that effect. And I read that it is a special kind of crystals developed in the body with the calcium sulfate deposits. This is also very, very, very interesting. And if you look at like this, it's a very unique creature and very loving shape. And the people, who are all the artificial intelligence people who are going to design the underwater robots, for the underwater robots, for design of it, certain fundamental inputs can be derived from its movements, its capability to dive, capability to turn around and to move, and its nature is elastic nature, slippery nature, color changing nature, and the propulsive forces, mobility from up to down and sideways, etc. How fast it moves and sensing the positioning system. I think with the look at all these things, etc., I found among the natural creations, this is one creature which gives lot of stimulant to the scientists for going deeper and deeper into so many aspects from which a marine scientist can do wonderful thing using this creature as a indicator or an instrument, scientific instruments for doing further things. And while going through the book of abstract is an excellent excellent resource of knowledge generated through more than 150 scientists who has contributed for that from various parts of the globe. And also it contains the species of the jellyfish from different parts, different seashores. I could go through it, with respect to, its characteristics getting different from one sea marine region to another marine region. I didn't know that, almost 1,000 species are available. Out of that, only about a dozen number is edible. All other things are not edible and, but still exist in different forms. And this from my getting involved with Dr. Biju Kumar for this function only, I could gather this much additional knowledge other than what I know in the scientific field. I thank the organizers so much, congratulate them for conducting such a wonderful gathering with a lot of planning and execution, providing excellent logistic support to all the people for coming to our state. And fortunately, the rain has not troubled much on these last 4 days. And so I thank all of you and congratulate you for attending this seminar here in our hometown here. And wish that all you will be benefited excellently well in your field of research for doing more and more useful things for the society. Thank you.
Thank you, sir. I request Professor Mike to hand over a memento to Sri M.C. Dattan as a token of appreciation. Thank you. Next, I would like to invite Professor Mike to take over the stage for in memoriam and honor lectures.
All right. So this is something that is new. We're trying for the first time at the meetings. We think it's important to honor people who've contributed to the discipline. So I'm filling in for Dror Angel, who led organization of this portion of the meeting. Dror sends his apologies from Israel that he can't be with us tonight. Okay. So kind of rather than an after-dinner speaker, we decided that at this meeting, it was time to try to formalize what has been done ad hoc at a couple of occasional prior meetings. And that is to honor those who've contributed a lot to the field. Some of the folk that we're going to honor were present at the first meeting at the Gulf Shores of Alabama in 2000. Some sadly have recently passed away. Most still with us, but unfortunately not able to join us in Kerala. We're delighted that two honorees are here with us, and we'll thank them in a short while. So a major emphasis for this section was to bring to the fore key contributions of senior researchers whose work provides not only important historical context for the work that we do now, but in many cases also remains useful. So science can be a little bit faddish, right? Following new technologies, exploring those is important, and the need to cite recent work to demonstrate relevance of the work we're doing. But in some insights, they're still important, can fall from favor solely because they're older, right? Not because they're no longer relevant, and not because they don't actually contribute to the future of our science. I have a note here, which is like kind of meant to be a joke, that you can kind of think of them as the bell bottoms or the mullets of science, right? They were in the past, you think they're not fashionable anymore, but in the future, they're going to be great again. All right. So, where are we there? All right, so much older work still offers current insights and is still able to contribute to better science if we only know where to look. And that gets harder and harder as more science gets published and we have to wade through volumes of work, right? And so bridging that gap is in a way a responsibility of those of us who have a foot in both past and the future of the discipline, so we can help identify contributions that will stand the test of time. And so this is a bit of what we want to do. So in this context, we'll have 10 short presentations on our colleagues past and present. We're going to start with 2 in memoriams for Veronica Fuentes and Dale Calder. And then we'll have 8 short appreciations for our colleagues who are still with us. I think rough estimate, the running time should be about 50 minutes. We'll try to get through this promptly. And so with that, I think I'll invite the first person to come up. We're going to do this in alphabetical order by the honorees. And so I think that makes Dale Calder the first, and so that'll be Alan Collins. Yeah.
Can they advance them for me, or shall I advance? I'm sorry, I'm going to have to read from a script. Actually, it's here in front of me. I'd like to, oh no. Calder. I think someone put my my research talk in here instead, so that's not right. Sorry. I turned it in, I swear. It's actually, it's right here. Right here. We could just have better do Veronica. Or maybe it's gonna be. Thanks. So I would prefer to just speak it, but this is text that was excised, that was put together by Antonio Carlos Marquez from Brazil, and Henry Chung from Canada, and James Carlton from the United States. So we begin this moment honoring the life and contributions of Dale Calder, his legacy of excellence as an eminent Cnidarian taxonomist and wonderful human being, with an essential disclaimer. Those who knew Dale recognized the challenge of saying something worthy of his extraordinary story and the outstanding quality of his contributions. To those who didn't have the privilege of knowing him, we regret our inability to fully convey the extent of his influence as a preeminent taxonomist of hydroids, a leader in his field, and one of the most remarkable individuals to grace the scientific community. So Dale Ralph Calder was born in 1941 in New Brunswick, Canada, and grew up on Deer Island in the Bay of Fundy. His formative education was undertaken at the United Baptist Bible Training School in Moncton, New Brunswick, and in the fall of 1960, Calder commenced his tertiary education at Acadia University in Wolfville, Nova Scotia, attaining his Bachelor of Science and Honors in Biology in 1964. Being mentored by Professor John Sherman Bleakney, a naturalist who researched diverse vertebrate and invertebrate groups across varied habitats, including caves and infralittoral zones, pioneering the use of scuba diving in such studies. Bleakney guided Calder in the biospeleology of Nova Scotia's caves and influenced his research on the marine fauna of the Bay of Fundy. Reflecting on his educational journey over 5 decades later, Calder honored Bleakney by naming the species Eudendrium bleakneyi from the Bay of Fundy in 2017, describing him as an esteemed mentor who contributed greatly to the content of this work. In 1964, Calder relocated to the USA to pursue a master's degree in marine science at the College of William and Mary in Virginia. His thesis delved into the ecology of marine invertebrate fouling communities among the species hydroids, which marked the commencement of his distinguished journey into medusozoan research. Continuing under the supervision of his master's advisor, M.L. Bremer, Calder acquired a PhD in marine science in 1968, and his dissertation centered on the taxonomy of hydroids in the southern Chesapeake Bay, yielding a catalog of 55 species, including 43 hydroids and 32 medusae, and encompassed biogeographic and ecological analyses, phenological insights, and life history. During this time, he made contact with a lot of the prominent Cnidarian researchers of that time, Crowell, Cramp, Peterson, Millard, and Rees, and Calder's work soon elevated him to the esteemed ranks of these Cnidariologists. Calder returned to Canada for a postdoctoral tenure in Ottawa, Ontario, continuing to publish on freshwater and brackish hydrozoan species now known to be invasive. This included his report of the brackish Limnomedusa Meiotis inexpectata in the journal Nature. And in 1969, Calder returned to the United States for more stable employment, holding simultaneous positions with the Virginia Institute of Marine Sciences and the William and Mary School of Marine Science. And during this period in the early 70s, he conducted his initial formal research on Scyphozoa, particularly focusing on the study of nematocysts and life cycles, all while continuing to publish taxonomic and faunistic surveys of planktonic and benthic hydrozoa and scyphozoa. In 1973, Calder moved further south to Charleston, South Carolina, where he served as associate marine scientist for South Carolina's Wildlife and Marine Resources Department, and he took on the role of assistant professor in the biology department at the College of Charleston. His research interests continued to split mainly between taxonomy, faunistics, and the ecology of hydroids, as well as studies on life cycles and cnidomes of Scyphozoa. Calder returned to Canada in 1981 as associate curator in invertebrate zoology at the Royal Ontario Museum, and later becoming a senior curator. And from December 1983 onwards, he also served as associate professor at the University of Toronto. In 2002, Dale concluded his affiliation with the University of Toronto, and a year later became curator emeritus at the Royal Ontario Museum after taking early retirement in December 2003 for family reasons. Dale's first spouse, Elizabeth May Secord, passed away in 2004, and he remarried again in 2008 to Ray Ding. And post-retirement, he and his family developed a routine of traveling south to Florida each northern winter. Now, Dale produced 137 publications throughout his career, comprising over 4,700 pages. And you can see here how Calder worked in many different regions in the world. And while he focused on taxonomy, faunistics, and nomenclature, he also made contributions in ecology, biogeography, fisheries and environmental impacts, biographies and the history of science, life cycles, nematocysts, and even the biology of cave-dwelling organisms. And likewise, he emphasized hydrozoa, but he also studied scyphozoans and cubozoans, broadly mirroring their relative diversities, and he also found time to study other organisms as well. These word clouds reflect the large number of researchers with whom Calder worked and the wide variety of topics that he addressed in his research. And over the years, he collaborated with 99 co-authors, primarily from the United States and Canada, but also from Brazil, England, Japan, Mexico, the Netherlands, Spain, and many others. One of his significant contributions is represented by Calder and Peters' 1975, in which he utilized cnidome data from Chiropsalmus quadrimanus, the four-handed box jellyfish, to discuss the systematic status of cubomedusae, concluding that the cnidome, together with differences in morphology and life history, supports the conclusion that cubomedusae are outside the mainstream of Scyphozoan phylogeny and should be separated from the remaining orders of the class. He was essentially concluding that the new class Cubozoa that was erected by Werner in 1973 should be recognized for all the sea wasps. As noted above, his contributions were diverse, including life cycle and cnidome studies of Scyphozoans, his mentioned work on cubozoans. Calder's faunistic works were based on extensive and meticulous studies of multiple collections for Bermuda, the North Mid-Atlantic Ridge, the Hawaiian Archipelago, the Bay of Fundy, Florida, and many other places. These works reached the highest level of quality and became seminal references for students in the field. Beyond his taxonomic contributions, Calder also produced significant collaborations in ecology and biogeography, which brought many new insights into the group. Overall, Calder authored names for 55 taxa, the Scyphozoan subclass Coronamedusae, and then within Hydrozoa, 2 families, 9 genera, and 42 species. 4 species have been named after Dale to honor his immense contributions to the field, and he gave back to science, the science that he loved, by meticulously crafting historical biographies for 13 different cnidariologists. And just a small sampling of some of his most impactful work, but what lists like this can never capture are the other ways that he influenced the field. Dale was a true gentleman, always gracious, patient, and generous with students, colleagues, and others with whom he interacted during his career. For example, Dale's personal qualities made him one of the finest reviewers in the field of cnidarian taxonomy and one of the inaugural editors of the journal Zootaxa, where he served from 2005 to 2013. And in these roles, where vanity and unethical behavior are all too common, Calder set an exceptional example of how to conduct collaborative and healthy science. His dedication and consideration for his colleagues was unparalleled. He consistently elevated the quality of submitted articles, assisted in taxonomic, nomenclatural, and linguistic aspects, and tirelessly reviewed extensive manuscripts. He frequently went beyond his already rigorous evaluation of manuscripts, particularly those of students of younger authors, mainly from non-Anglophone countries, offering gentle guidance and encouragement which invariably resulted in much improved publications. And even as his health declined and made it difficult for him to complete his own projects, he never refused to review a manuscript or to provide advice. His kind-hearted Herculean efforts stimulated Cnidarian study and raised the field's standard through his example, knowledge, and integrity. And these distinguished qualities also enabled him to mentor a succession of young scholars from 1970 until approximately 2000. And during this period, Dale played a supervisory role for approximately 18 graduate students, and he was particularly sought after by the University of Toronto for serving as members on committees. And now, Dale passed away on June 4th, 2022, at the age of 81 in Toronto. And here on the left is Dale with Henry Chung and the late Anita Brinkman Voss, not long before she passed away. And on the right, he's seen with his wife Ray and daughter Danina. He's also survived by his two daughters Nancy and Andrea, with his first wife Elizabeth. Always characterized by his reserved and modest demeanor, Dale is revered as an educator, a gentleman, and an accomplished scholar. Thank you.
Okay, so next up will be an in memoriam for Veronica Fuentes by Macarena Marambio.
Well, Veronica, she was a great person. I know that everybody who knew her knows this. She died young, but she did a lot. So she was born in Argentina, in Cordoba. She never forget her country. She was always with the Argentina in her heart. In 1975, she studied biology there, but then she went to Germany to make her PhD. But she was still half Argentina, half in Germany. And her both passions were the Antarctic and jellyfish. And then in 2007, she came to Barcelona, and she had the great challenge of create a new group in the ICM. And she made her postdoc, but she wasn't able to finish because she had to retire because of the cancer. And she participated in a lot of research projects, national and international. She was part of different Antarctic oceanographic campaigns, also Mediterranean campaigns. She participated in more than 80 national and international presentation, conference, symposiums. She published 75 papers in collaboration and by her own. And she guided 10 PhD theses during her short career. And actually, I was asked to talk about her literature, her papers, her published papers. But I realized that Vero was much more than her papers. She was, for the ones that we grew with her, she was more than that. She was a friend, she was a teacher, she was like a mother with all the people she knew and the students she had. So when she arrived to Barcelona, the first challenge was to set up the aquarium area. We had nothing there. And they needed the aquarium suitable for jellyfish. And these are the pictures of the first things they got at the beginning, the first years. And the aquaria some years later were really improved with the help of her husband, Alejandro. A lot of experiments were carried out with ephira, adults, jellyfish, polyp flats, ecophysiology. Those were years with a lot of work in the Barcelona group. And meanwhile, national and European projects came, the Vectors project, Medusa project, the Cubomet, Met Jelly Risk, iMet Jelly, the Fish Jelly, Reclimate. And the life cycles of the two most important jellyfish were closed. The life cycle of Pelagia and Rhizostoma. They were closed at the Barcelona lab. And these have been our jellies all these years, since she arrived there until nowadays. Well, actually, nowadays we don't have the aquarium area, but we are recovering it. And at the same time, she started the creation of the Jellyfish Observation Network, the training to the rescue services, the administrations. She began with this, the dissemination activities at the beaches and everywhere. We went everywhere because she believed that dissemination, expanding knowledge of jellyfish was a way of getting to people. So she was involved everywhere. Actually, in these pictures, in these photos, you see a domo that they created with Alejandro and that we had 2 hours and a half to construct in the beach and spend the whole day there with the aquarium and the jellyfish. And we went to the whole Catalan coast during years. And she was always involved with all these activities. And with all this, the papers on different topics regarding gelatinous animals were published. Ecophysiology, all kind of experiments, also interactions between jellyfish and fish, between jellyfish and aquaculture. And she also conducted valuable research on the first blooms of Nemiopsis lady, and also Charybdea marsupialis in the Mediterranean with the Cubomet project. She also conducted a lot of works with contributing to the knowledge of Rhizostoma and also of Pelagia. And these are some of the papers because it was hard to look for them all, but you can find the literature. I think it's better to remember Vero by looking at the photos. These are some pictures in the Antarctica. It was one of her loved things. But then she also had the campaigns in the Mediterranean. And she spent a lot of time with Jennifer, that it was like a mother to her and for us also. And of course, the group in Barcelona, the Jellyfish group from Barcelona. We spent a lot of good times with her. I think we will never forget what we learned of, not of science and not of jellyfish, although she was really passionate, but of life, because I think she taught us very, very important things. And we will never forget the jellyfish bloom that was organized in Barcelona. And actually, I think it's the ones that were there, I think that maybe it was her last effort, because after that, she had to leave science and try to recover. And I'm sure that if she was alive, she will be here really, really happy, because actually she spent some of her last time here in Kerala learning about Ayurvedic medicine for trying to heal herself. She did everything, but life is life. So here I leave you some of the comments that I got from the people that knew her. And you can see that it's passion it's friendship, and it's all you can remember about her. We will remember her every day, and we'll try to do our best in Barcelona now that she's gone. Tomorrow will be 2 years since she left us. So thanks, Vero, for everything.
Thanks, Macarena. Third in memoriam, sorry, is for Phil Pugh, Masech Manco.
Hello again. So this is something very last minute. So the presentation is rather short, but it was brought to my attention by Luis that but the in memoriam section didn't include Phil Pugh, and he made quite an extensive contribution to a very narrow field of jellyfish research. But I feel that we owe it to him to at least mention a few words about him. So those who have had the chance to meet him might remember him as the guy who was just dripping his hands into jars of formalin because he wouldn't want to damage his precious siphonophores. So he was the most meticulous taxonomist ever. He's well remembered as the guy who was picking each and every tiny piece of siphonophores from those ROV jars, spending hours and hours in front of those jars. But this is not the only thing that he did. So Phil started his career in Southampton by looking at the phytoplankton. So actually his first steps in career were pertaining to phytoplankton. And only when he finished his postdoc in London, he discovered the magnificent world of siphonophores. So he was invited to join Mike Thurston, a ostracodologist, for a cruise, and then fell in love with siphonophores. So he cruised everywhere in the world, notably with Rich Harbinson, then Steve Haddock, and basically all the people who had the access to ROVs, looking for siphonophores and trying to ID as many species as possible. And so he did contribute quite a lot. He published more than 100 papers, mostly on taxonomy of siphonophores. And he participated in over 50 research expeditions and came with tons of species description based on those research expeditions. So when Phil started his work with siphonophores, we knew only about 130 species of siphonophores. And when he finished, so the current count of siphonophores is 194, and he described 45 of them. So 25% of siphonophore diversity, we owe it to Phil Pugh. So that's quite a lot. And you can see the red bars of this plot are actually the ones authored by Phil or Phil and his co-authors. But it's not only species descriptions. Phil contributed a lot of, he resolved a lot of enigmatic taxa, sorted out the convoluted taxonomy of siphonophores. And these are some examples of the species he described. But he also co-authored with George Mackey, who we also mentioned already today, and Jenny Purcell, a very important resource for all of us working with siphonophores, a siphonophore biology book that features all the aspects of siphonophore ecology, development, and even taxonomy. He was particularly interested in siphonophore life cycles, although he never actually worked on siphonophore culture. He had a soft spot for the life cycles of siphonophores, so he went through all the ancient literature, picking out the fascinating details of how siphonophore develop. And what he tended to do was to produce those magnificent reviews that were lengthy, I admit that. But you can be sure that if he published a review, that there was no mention of any omissions. His Rhodalite review includes records pre 1800s. He would copy ancient drawings, redraw them. Like there are literally the best resource existing. But he also trained the new generation of siphonophorologists, if that word exists. He made significant contributions to many careers. I'm sure you heard of Casey Dunn. His career wouldn't be where it is as it now without Phil Pugh. I also had the pleasure to work with Phil. He had this funny tradition of inviting the people who entered the world of siphonophore taxonomy to his allotment in UK. And this was his testing grants. So you would spend the whole day working on weird tasks that he would assign. Like I was building a fence around his tomato plot. And I was questioned about siphonophore morphology. But that was fun. I really enjoyed these moments. But yeah, so the last slide I have is the species that was named after him by Dougal and his colleagues, including Veronica, who was also the co-author of this species, and the species is named after Phil. And so this would be all, but as another last-minute information, Jan pointed my attention towards another prominent hydrozoan researcher who passed away 2 years ago, Jen Watson, who passed away at 94, and she has herself described nearly 200 species of hydrozoans. She was a prominent Australian researcher, so it would be a pity not to mention her name on a meeting like that. So I think we owe them tons of gratitudes for doing the hardest work ever, sorting out the mysterious world of the taxonomy of hydrozoans. So that's all. Thank you very much.
Okay, so we'll switch over to in appreciations now, and the first is for Rick Brodeur. This is by Tom Doyle and Mary Beth Decker, and it's going to be a video that they recorded for us.
Hello, I'm Tom Doyle from Ireland.
And hello, I'm Mary Beth Decker from the United States. And we're thrilled to be here to celebrate and recognize Dr. Rick Broder's significant contributions to jellyfish bloom ecology.
Rick grew up in Massachusetts, a couple of hours from the ocean. He didn't spend much time at the coast, but he had an early interest in submarines. While at university, he got an internship at the Woods Hole Oceanographic Institution, where he spent 6 weeks at sea as a 19-year-old. After this cruise, he knew he wanted to study fisheries science, and the rest is history. Rick did his graduate work at Oregon and Washington, where he studied trophic ecology and pelagic fish.
As a postdoc for Canadian Fisheries, he wrote a famous paper about long-term variability in zooplankton biomass in the Pacific. Here, Rick was studying large-scale regime shifts before the term was coined. Rick started as a research fisheries biologist with NOAA Fisheries in 1991. He spent many months at sea studying Bering Sea and California current ecosystems.
Rick's research interests can be broken down into 3 areas: the early life history and recruitment of fishes, the trophic ecology of fish and food web studies, and the distribution and ecology of gelatinous zooplankton, which came later. Rick has been incredibly productive, as indicated by his H-index of 70 and over 280 publications. A word cloud of his papers highlights his main research areas. But while only 20% of his papers are on jellyfish, can you imagine what he would have done if he only worked on jellyfish? But the appeal of Rick's research is its interdisciplinary nature, and it's only someone with such broad interests that could have written his jellyfish papers.
One of Rick's important early contributions to jellyfish science documented an increase in jellyfish caught in Bering Sea research trawls. Rick published this paper when the jellyfish community and the general public were becoming aware of apparent rises in jellyfish populations around the world. Rick kept an eye on these trends, and when he noticed this big drop in jellyfish biomass, he brought together a team of oceanographers, jellyfish ecologists, and modelers to explore possible drivers of these changes. He showed that many factors drive jellyfish fluctuations and that warming may not necessarily lead to more jellyfish in all systems. Rick has also looked extensively at food habits and the distribution of fish and jellyfish. This paper showed that jellyfish diets and also their distributions overlap with those of forage fish. Rick shows us here that jellyfish blooms could negatively impact important fish species.
Rick is a very active member of the Pisces Intergovernmental Organization, which coordinates marine research across the North Pacific. Rick was a member of numerous working groups, but notably, he co-chaired the Jellyfish Working Group with Shinichi Ui. And never one to stand still, Rick has been at the forefront of ecosystem-wide investigations for over 4 decades. This is clearly illustrated in Rick's 2019 paper about a huge increase in pyrosomes off the California current ecosystem during the largest heat wave, or marine heat wave on record. Rick's work documented a dramatic shift from the typical crustacean plankton community to one that was almost entirely dominated by pyrosomes.
Rick is well known in both the fish and jellyfish communities for being an outstanding mentor. He is especially supportive of young scientists, including the two of us back in the day. So many are very grateful to Rick for all he has done to move their careers forward.
And like all happy endings, Rick did get to go down to the bottom of the ocean in a yellow submarine. Thank you, Rick, for all that you've contributed to the Jellyfish Bloom community.
Okay, great, thanks. So next up is Bill Hamner, who I was going to do the appreciation for. Okay. Right, so just organize my stuff. Right, so Bill Hamner shaped the way that we study and know plankton. He took sampling off ships and boats and away from the coast and into the blue water of the Pelagic Ocean. He started this work with his wife and colleague Peggy Hamner and a suite of graduate and undergraduate students and colleagues a little over 50 years ago. So it's timely to appreciate and highlight his long-standing and outstanding influence on the discipline and to bring these to the forefront again. And the main lessons are careful observation and innovation for new generations, as we would argue that they gain renewed perspective on their importance for a holistic picture of plankton. So this is kind of like a graphical schematic of Bill's biography. Bill's unique perspective on plankton came in part from his early work as an ornithologist. So about the first decade was mostly on birds, and he published 4 or 5 times in Science and Nature, including his first paper. And so what he really brought to the discipline of plankton biology was an outstanding perspective that he learned with birds and ethology. So Bill brought the practice of ethology into the seas. Its incarnation was blue water diving, which he started in Bimini in the early 1970s, was featured in a suite of publications, including publicly accessible ones, including in the National Geographic. The classic papers for blue water diving are his publications in 1974 and 1975. And then he applied that technology in a variety of contexts: Australia, Antarctica, the tropics, and it led also obviously to a lot of work in submersibles in the modern day. For this work, he received a prestigious award, the Nogi, from the Academy of Underwater Arts and Sciences, which was awarded to both Bill and Peggy. So Bill's major innovations were blue water diving as a technology, but also its use in better observing living plankton. So it's enhanced understanding of diversity of plankton, He also contributed to the establishment of our understanding of the coupled nature of biological and physical determinants of the plankton. He brought ethology into the plankton, as I mentioned, and with that, he was able to document unexpected behaviors that really were not expected of plankton at the time, right? Plankton is drifter, but plankton do many things other than drift oftentimes. And I think it really there's this phrase from a different discipline about a feeling for the organism, right? So that's what Bill brought to plankton. It's like a perspective from the organism about what it is like to live in the plankton. So we chose essentially this paper as representative of Bill's research, and because it really integrated, it really integrated the breadth of Bill's interests. It's captured in this subset of illustrations drawn from it. The one looking at the relationship between U and Reynolds number is actually from a lay of 1977, but it reflected on its diversity of influences. And it captures the importance of the biophysical relationship in determining the diversity of life forms in the oceans and plankton, right? Looking at homology, homoplasy, why taxa that are very different evolutionary relationships have similar form, adaptations to living in the plankton lead to these strange and wonderful forms. And particularly this idea about the physical environment very much shaping the life in it leads to one of these large problems that we currently deal with, right, which is the problem of cryptic species that bedevils biodiversity studies in a pre-molecular age, and that we're only just now sorting through and trying to understand the functional traits that as a consequence of that. One of the great privileges of writing an appreciation is that you can actually ask the person about their work, right? And so we asked Bill what he thought was his most underrated paper, and so this was his response. What he wrote about it was that horizontal foraging occurs in this gelatinous zooplankton, as in this example from the ctenophore leucothea. Even these plankton can exploit thin horizontal layers of prey by moving slowly horizontally with lobes and mouth forward and without roll for periods up to about 40 minutes. So this organism has a functional top and a functional bottom and a left and a right and a front and a back when leucothea is feeding. So they're functionally bilateral at this point, something that is not what we might initially predict for what we typically think of as a largely radial symmetric metameric plankton, but rather would associate with something like cephalized larval fish. And he also comments that blue water diving technology has been used oftentimes to collect organisms undamaged to bring them into the lab to study, but we should be trying to use it more and more for observations of the animals actually in situ, so that if we collect them for other additional analyses, we know what they were doing at the time that we sampled them. Right, so Bill's key bibliography. Bill published about 100 papers. We've selected 5 must-read ones from across the breadth of the institutions at which he works. All of his papers are worth reading, I would say. There's much in there to be learned about the foundations of our field, what it is to refocus those thoughts into current and future endeavors. Bill's work is also a great example that you can't oftentimes measure somebody's contribution simply by the number of publications or the number of citations that they have. Bill's work really shaped our field and also shaped the way that people think about the plankton. And the last couple of citations on here were his publications in National Geographic. And then, as others said, they leave a great legacy. So this is a list of Bill's graduate students. Okay. One of these is Larry Maiden, who we'll hear an appreciation of next. It was recorded by Kelly Sutherland.
Larry Maiden grew up in the San Francisco Bay Area in California and did his undergraduate work at UC Berkeley, graduating in 1969. He then joined Bill Hammer's group at UC Davis. It was there that he met his future wife, Kate, while teaching as a teaching assistant for a field course at Bodega Marine Lab. Together with Bill and his group, Larry made blue water dives from a 14-foot Boston whaler in the Gulf Stream from Bimini, a set of islands in the Bahamas. And that was from 1971 to 1972. The following summer, the group conducted blue water research in the Gulf of California, in Baja, working from a Zodiac. During these blue water dives, Larry made the first in situ observations of salps, describing their morphology, swimming, and feeding behavior. Beginning in 1974 as a postdoc at the Woods Hole Oceanographic Institution with the late Richard Harbison, Larry translated blue water diving and the study of gelatinous zooplankton to ocean-going research vessels, where he was able to combine classic oceanographic sampling with underwater observation and collection. He went on to secure a permanent scientist position at WHOI in 1975, where he remained for the rest of his career. Over his career, he studied and published work across many zooplankton taxa, including ctenophores, medusae, siphonophores, and salps. In addition to observations underwater, he did lab experiments with field-collected salps and other gelatinous organisms. His meticulous observations of salp feeding rates, metabolic rates, and particle selection are still frequently used to inform food web studies and biogeochemical models. He served as chief scientist on a number of NSF-funded research cruises, where together with colleagues, he reported on plankton ecology, especially the dynamics of salp swarms, their ecological impact, and their vertical migration. He helped describe new gelatinous species, especially from the deep sea. Collectively, Larry's collaborative research spanned scales from the organismal to the oceanographic, and often entailed instrument development to enhance sampling and exploration. Perhaps even more impactful than his research output were the number of students, postdocs, and colleagues whose careers he touched. Larry introduced countless scientists to blue water diving and opened their eyes to the value of observing fragile oceanic and deep sea plankton face to face. He was generous with his time and resources. His quiet acts sometimes helped reconstruct research directions and careers. Though it may not be apparent in the prose of published research papers, to all that worked with him, it was evident that Larry is an adventurer and an explorer. He dove with plankton worldwide via scuba and submersible, including the North Atlantic, South Atlantic, North Pacific, South Pacific, Indian Ocean, Red Sea, Celtic Sea, and the Southern Ocean. Each research expedition was underpinned by hypothesis-driven research questions, but there was always a component of exploration and willingness to follow his own curiosity and that of his students and colleagues. His curiosity about organisms, their diversity, and their individual roles and interactions added a strong natural history bent to his papers. He also created beautiful photographs of jellies. Any narrative about Larry must also include his dry sense of humor and calm in the face of ridiculous or less than optimal situations. His calm, unflappability, dry wit, whatever you want to call it, served Larry equally well in dicey field conditions and leadership roles. During the latter half of his career and after ascending the ranks to become a senior scientist in 1996, Larry served WHOI in a number of leadership roles, including chair of the biology department, director of the Ocean Life Institute, and finally, deputy director and vice president for research. These roles broadened his interest to include conservation, ocean resources, and policy. Larry's remarkable calm, insight, and perspective, traits that made him a valuable colleague and mentor, also made him a valued and steady leader. When he retired from WHOI in May 2019, Larry had been at the institution for 43 years, more than half of its history. During those years, he shaped the scientific ethos of a major oceanographic institution, and more significantly, the fields of organismal biology and oceanography.
Okay, next up is Martin is going to honor Alenka. And so maybe we can ask Alenka to come up too. Okay, that's perfectly fine.
This will not be the right one, I guess. In the meantime, I'm very honored to be invited to speak about Alenka. And I hope I get the presentation prepared. Alenka. So I'll start with a few facts. Alenka graduated in 1972 from biology at University of Ljubljana, and in the same year she got an employment at very recently formed marine biology station. Actually, the station was formed in 1969, so it was just a very young institution at the time. She completed her master's in 1977, also at University of Ljubljana, under mentorship of Jože Stirn. In 1984, she finished her PhD at University of Zagreb under mentorship of Adam Benovic. She was the head of our station from early 90s for almost 20 years until 2009. And she became a professor at University of Ljubljana in 2003. Alenka is a member of many international organizations. And after her retirement, she started a new career as an international judge in First Lego League, a job she really enjoys, working with children and their first research steps. She was also instrumental, actually, because in 1991, Slovenia became independent for the first time in history. And Alenka was instrumental in getting our young country in many international organizations and getting recognized internationally. She always liked field work, so she was on numerous national and international expeditions. She cruised the Adriatic several times with the Andrija Mehorovičić expedition, but she was also in UK, Argentina, Israel, France, Turkey, and I'm sure I forgot something, but maybe she'll add later. But her research actually didn't start with jellyfish. It started with crustaceans, and her paper on crustaceans of East Atlantic was actually the first widely cited paper of then very young group of marine biology station. So she put our group on the world map. But then in the late 70s, the blooms of Pelagia started, and Alenka started working with jellyfish, and this remained her favorite subject ever since. So I'd like to give special attention to her paper from 1993, where she did stable isotope analysis on Pelagia noctiluca and its prey. It's probably one of the first, if not the first paper to use stable isotopes on analysis on jellyfish. And she has shown that Pelagia, even though in it was in our shallow coastal waters, fed primarily on net zooplankton and not, for example, on microzooplankton or DOM or POM. She also did the biochemical analysis. I think it's obvious I don't know what I'm speaking about because I'm a physicist, I hope, but Alenka I'm sure she can explain more about the paper than I did. And anyway, but what she has shown is the enrichment with C13 in jellyfish compared to its prey, and the lower ratio of CP and CN compared to its prey again. But I'll actually finish on a much more personal note. I'm very, very proud to be the last of Alenka's many PhD students. I couldn't have wished for a better PhD advisor, actually. She introduced me to the wonderful world of jellyfish while I was coming from a completely different field. And above all, she understood that while raising a scientist, you have to allow him to explore some of his own ideas and fail as well on his own, and help him when he fails. And even more, even after retirement, Alenka is still the highest instance of wisdom in our group. And me and my colleagues turn to her often with questions when applying for projects, writing papers, or reaching seemingly dead ends in our research. And I actually bow deeply to Alenka to be willing to help us even and advise us even after retirement. Thank you.
So next up is Kylie. He's going to give an appreciation for Jenny Purcell.
It was a bit of a spoiler, but yeah, I love this photo of Jenny. So if you do a search on the Web of Science and look up the word jellies or jellyfish, and if you do a search on the researchers who've studied jellyfish, then Jenny is the one who tops the list, along with Jo Burnett. So she has been the most productive and prolific publisher on the topic of jellyfish of anyone ever, and that's quite an achievement. So Jenny actually grew up in landlocked Missouri, so she was far from the sea, so it's perhaps a little bit surprising that she became a marine biologist. However, her father really cultivated a love in the natural environment. So for her first degree, she went to Stanford University, and during that time, she spent time at Hopkins Marine Lab, and her first ever publication was actually on anemones back in 1977. So not jellies, but still the Cnidaria. So she followed that with a PhD at the University of California, Santa Barbara, where she worked with Alice Aldridge, who's another giant in our field. And during her PhD, she studied siphonophores. So that's where her career on jellyfish really started to take off, if you can call siphonophores jellyfish. After that, she did a couple of postdocs. So first of all, she went to Woods Hole Oceanographic Institute, where she worked alongside Larry Maiden. So again, doing some of this fantastic blue water diving, which I think we all look very longingly at as scientists these days. She then followed another postdoc with George Mackey, who we have actually learnt just passed away just recently, and that was at the University of Victoria in Canada. So it was during these postdocs that Jenny really formulated her ideas about jellyfish as being predators and competitors of larval fish, which is one of the main legacies of her research. Jenny then took up a tenure-track position at the University of Maryland, where she progressed to full professor. And during that period, she won numerous National Science Foundation grants, as well as grants from the USC grant program. This period, I think, was quite challenging for Jenny. She didn't really enjoy the challenging academic atmosphere, and she elected to actually move back onto soft money, and she spent several years working at Western Washington University. She then began a phase of her career that she referred to as being an itinerant scientist. So for about a decade, she spent time working in labs all around the world. So some of the labs that she worked in were with James Lowe in Taiwan. She spent about a year in China working with Sun Song's group. She also spent time working with Tom Doyle in Ireland. And, when you talk to people, like Tom, Tom was just saying that the time that he spent with her, she was incredibly generous with her time. She was full of enthusiasm for research. And he said that she was most at home when she was at sea. So I think if you think about Jenny, her legacy in terms of research is unparalleled. She's made significant changes to the way we think about things, particularly how changing ocean conditions can influence jellyfish populations. But I think there's two legacies other than just the science that she's produced. First is that she forged a career as a woman in marine science during a time that was a lot more challenging than what we face females in marine science now. And she's always been very passionate about supporting junior researchers, particularly young female researchers. But I think perhaps her greatest legacy is this. So Jenny, along with Monty Graham, was the person who initiated the first ever International Jellyfish Bloom Symposium. And this is at Gulf Shores, Alabama in the year 2000. And I attended as a very newly minted graduated PhD student. And you might see there's a few other faces there that you can recognise, including Mike Dawson. Kathy Lucas was there as well, and Kathy is actually in Australia at the moment. She's on sabbatical and visiting our lab. She couldn't come here, but she says hi to everyone. And the other day we were just talking about this conference and the fact that we were both early career researchers and completely in awe of the senior researchers that we were meeting this facility or at this conference. So if you're a junior researcher here today, then I know exactly how you felt. And don't be worried if you're feeling like all being surrounded by some senior people, because yeah, we were all in your position once as well. So this was 2000, right? So we're now in 2023, 23 years later, and we're at the seventh International Jellyfish Bloom Symposium. So I think we need to give a big thank you to Jenny for initiating this event, and we hope that it's going to continue for many more years. Thank you.
Alan Collins for Peter Schuer, I think.
Excuse me. Oh, you got it. Cool. All right. So again, I'm going to read shortened text and use the PowerPoint that was put together by Antonio Carlos Marquez team. And for this, we're honoring Peter Schuer, who I think can be argued is the most influential hydrozoan systematist alive. He retired this past year during 2022. And Peter's still actually a pretty young man. He was born in 1958 in Switzerland. And his scientific trajectory might seem somewhat surprising. He started in science as a lab technician in biochemistry and microbiology in Basel and Bern. And then he went on to study microbial biology at the University of Bern, first completing a diploma thesis and then a PhD focusing on DNA recombination in yeast, which he completed in 1988. He found his passion in zoology, taxonomy, and morphology during his first postdoctoral position in Austria with Professor Reinhard Rieger at the University of Innsbruck. And upon returning to Switzerland after 2 years, he finally turned to Nigeria, and after 3 years, journeyed to New Zealand and spent 3 years studying the life cycle and systematics of New Zealand marine hydrozoans, leading to one of his seminal monographs. After this, he returned to the University of Basel, continuing his studies on hydrozoans, but also working on their developmental genes. And after a brief pause in his studies, he secured a tenured position as a research officer at the Museum d'Histoire Naturelle in Geneva. Here he had the resources and time to really excel as a leading expert in hydrozoan biodiversity, integrating morphology, molecular data, and life cycles. And his discipline, dedication, and meticulousness are evident in all of his contributions to hydrozoan society science, and they're characterized by their very high quality, reliability, and thoroughness. Now, over the years, Peter published dozens of papers, most often as first or last senior author, significantly advancing understanding of Cnidaria through several groundbreaking studies. He published the first formal cladistic analysis for the higher taxa of Cnidaria in 1993, and I was personally lucky enough to collaborate with him during a period when he worked on a series of phylogenetic analyses on Medusozoa, Hydroidolina, Leptothecata, Capitata. And Peter's prolific publication record encompasses detailed revisions of various taxa, notably extensive series on European hydroids, European anthoathecate spanning several years, and in-depth synopses of the faunas of Britain, New Zealand, Iceland, Greenland, Kei Island, Okinawa Islands. And so far, Peter has named 51 hydrozoan taxa, 4 families, 8 genera and 39 species, and he also coined the name Endonidozoa, which unites the two endoparasitic groups of Cnidaria, Myxozoa and Polypodium, and he's had a genus and species named in his honor. While he did not formally mentor master's or PhD students, he significantly contributed to many researchers' careers, my own, many others. And beyond his published work, his reviews and editing are known for their thoroughness and helpfulness. And one of his hugest contributions to science has been curation of the online database of hydrozoan names. This is part of the World Register of Marine Science, WORMS. And this serves both the hydrozoan community, but also the larger community of marine biodiversity scientists who require access to reliable names and taxonomic assessments of species validity. And so this is from his 1993 work, Inferring the Phylogenetic Relationships among the major groups of Cnidaria. This was a very inspiring work for myself. To much more recent work, collaborating with citizen scientists and a blackwater diver extraordinaire, Richard Collins, not related to me, describing Gulf Stream hydromedusae, including new species descriptions and placing them within the hydrozoan tree of life. He's got a lot of work still coming, and so here we celebrate the work of Peter Schuchert, what he's accomplished, and congratulate him on his retirement in 2022. Thankful that he remains active, editing for WORMS and continuing his research free from bureaucratic constraints. Collaborating with Peter Schuchert has been an honor, and we extend our gratitude for his dedication, professionalism, and his immense contributions to hydrozoan research. Thank you.
Okay, thanks, Alan. And then last but not least, in honor of Shinichi Ue by Dougal Lindsay. Shin, where are you? Would you like to come up to stage?
So either turn around and see the man in person, or it is Shinichi Ue, who he was born in Yamaguchi Prefecture in Japan, which is that little red, I don't know if you can see the red spot on the map there, down right at the south of Honshu, the main island. It's a very famous place for pufferfish. And I'm sure from a very young age, he was living his life dangerously, eating fugu. He got his PhD further north at Tohoku University, which is where Japan is bent there in a backwards L shape, just up there. And he retired from Hiroshima University, where he spent a lot of his academic career. I was surprised actually how long ago he retired, gave his final lecture. But back at the 4th International Jelly Bloom Symposium that we had in Hiroshima, you might, many of you I guess were there and have fond memories of the great symposium itself, but eating things, many weird and wonderful things, and drinking out in the downtown Hiroshima. I have some memories and some nights I'm not quite sure I remember completely. But probably we know him, we at least know him most for his work on jellyfish blooms, and especially Nemopolyma nomurai, the giant jellyfish that used to visit Japan quite often in very large numbers until people managed to get funding to study it, and then it miraculously disappeared and pops up from time to time still in fairly large numbers. And so this is Shin's most cited paper, over 1,200 citations for this paper, which is pretty impressive. For jellyfish blooms, but
This is actually the paper that I have used the most, cited the most myself. It's kind of like a data paper, even before data papers became a thing. One thing about Shin's work is he always is, they're very data rich and meticulous, and lots of information on rates and carbon contents and all of this important functional stuff, which especially myself, I do mostly taxonomy, and a lot of researchers in Japan are skewed in the jellyfish studies to taxonomy. But this is before we all said, well, we have to be concentrating more on functionality rather than just what is where. It's like the nitty-gritty, the actual numbers that go into the models so that you can actually do stuff with the data you have. And from a very long time ago, Ue Sensei has been writing those kinds of papers. He actually started, I think, copepod eggs in mud, I think was the first scientific contributions, and lots of stuff on egg production and copepods at the start of his career. His first Scyphozoan paper wasn't until 1994. And then he's also written on larvaceans. I know Connie is a big fan of his larvation work. And his jellyfish bloom work only really started in 2001. And then after 2005, he came over to the dark side. And no more copepods. All jellyfish papers for quite some time. And recently he's been working on ketognaths as well. So copepods, larvaceans, jellyfish rates, carbon contents, etc. Ketognaths, a really, really wide variety of things that Ue Sensei has published on over the years. A very wide breadth to be able to place observations in a wider context. He's also been very active in dissemination and telling the public about jellies and plankton in general. It's a thing in Japan to dress up in animal costumes. And Ue Sensei also dressed as a jellyfish here, not to miss out. It's also worked tirelessly at the Plankton Society of Japan, mentoring all of the younger researchers and running the society for many years as well. So he's actually there in person, so make sure you take the opportunity, if you haven't already, to go up and say hello and take advantage of his knowledge.
Okay, I think we just have 3 shortish things remaining. Suran?
Thank you, Jellyfish community, for honoring the leaders of excellence, those who leave lasting memories. Their legacy will live forever. Thank you, Jellyfish community, for their fond remembrance. Now I invite Professor K. Padmakumar, co-convener JBS 7, for declaration of awards.
Okay, so these are student awards for talks and for posters. Thank you, everybody who took part in the evaluations.
So it's a pleasure declaring the results of the evaluation of both oral presentations and poster presentations. So we have very excellent oral presentations as well as poster presentation. So the judges find it difficult to decide among which is the best, but so finally we have to decide. So we had a scoring, and based upon the scoring, this was decided to award 3 awards for the oral presentation. So we have the first winner is Scott Morrissey for the presentation on use of eDNA to test hypothesis on the ecology of Cairnux flickeri. Biju also joined. The second winner is Radish Kumar K. Is he there? So the title of the presentation is Spatio-temporal variation of elevation in jellyfish catch of bagnets in Maharashtra, India. If he is not there, someone from he hails from Central Marine Fisheries Research Institute. So one of the representatives can, or his colleagues can come and receive the certificate. The third prize goes to Jessica Strickland. The presentation entitled Convergent Oceanographic Slicks as Potential Habitats for Jellyfishes. So the next set of award on posters. So the winner is Sarasa Nagatsuke. The title is on taxonomic position of Aurelia in the seas around Ogasawara Islands. Please. The next is Lara Marstella. Tolerance of Cassiopeia Andromeda to warming scenarios of the Mediterranean Sea. Next is Surya S. An update on the jellyfish swarming dynamics along the southern coast of Kerala. Congratulations to all the winners. There will be next time. So let's go for that. Thank you.
Thank you. So before moving on to JBS 8 bits, I request Professor Mike to announce the publication of JBS 7 papers in journals.
Okay, so this will be a brief announcement. Stefano Pereno is working on this, but he wasn't able to join us today, this week. So we're still in negotiation, but he's been working really hard, looking at a variety of publication venues that are quality and affordable to everybody. Right? And so we'll send you an announcement on that, probably, I hope, in the next week. One of the publication venues will be the Journal of the Indian Marine Biological Association. Right, so that's an open access regional journal, which I think some of us are eager to support. Okay, so we'll send more information on that in a short while.
Thank you, Mike. Now we will have JBS 8 bits to announce the next meeting.
Okay, so we've had several really interesting discussions over the past few days about potential venues. And so we have a short presentation now on the destination for Jellyfish Blooms 8.
Hello, everybody in Kerala, in India. My name is Tom Doyle, and I'm based at UCC, or University College Cork, here in Ireland. And Mike has asked me to present a few slides to say why Ireland may be or might be the next host for the JBS 8. I'll just share my slides. Okay, so why Ireland? Well, when you think of Ireland, you probably think of a couple of things, and pints of Guinness are certainly at the top of it. So yes, if you come to Ireland, there'll be lots of pints of Guinness. There'll be lots of music. We'll take you to some local bars so you experience and get to hear Irish music. There's lots of castles. In particular, this castle here is Blarney Castle. If you, it's very near our university. You climb to the top of it. You kiss a stone called the Blarney Stone, and then you have the gift of the gab. So you know, we're also famous for Irish dancing. You probably won't get to experience river dance, but we'll certainly make sure there's some Irish dancing available. It's famous for its literature and Irish poets. So W.B. Yeats would be probably the most famous, and Seamus Heaney more recently. And then it's famous for being the land of saints and scholars, so St. Patrick's Day. But then I suppose in terms of music, U2 would probably be the biggest band that I'm sure you're all familiar with. But I don't think we can guarantee that U2 will play at the Symposium. It's considered the Green Isle. It's lots of green fields. It's green all year round, and it's beautiful mountains. It's full of sheep. It's got some lovely beaches and a lot of archaeological heritage going back thousands of years. Some of these stone ruins are over 2,000-3,000 years old, and these beehive huts are famous from the Star Wars movie. But of course, you guys are probably more interested in the sea, hugely productive seas, because it's temperate waters with a big shelf, very broad, extensive shelf. So lots of megafauna, including humpback whales, fin whales, resident populations of bottlenose dolphins, lots of seabirds, including puffins, and of course there's lots of elasmobranchs, including basking sharks that come in very close to shore. So these are things you'd probably get to visit. And then there's a great diversity of gelatinous zooplankton. from the lion's mane jellyfish to siphonophores such as apolemia, maybe 25, 30 species of hydromedusae that we know of. And of course, we do have lots of pelagia, which caused the massive fish kill from many years ago. There's a history of marine science in Ireland, so you may not have heard of John Von Thompson, but he invented a plankton net. He lived here in Cork Harbour, and he also discovered that the zoea larvae of metamorphose into crabs. But our hero would be Maud de Lapp, and she was a pioneer in gelatinous zooplankton studies. And over 120 years ago, she was the first person to culture the compass jellyfish, or Chrysiora hysocella. So that's not very far from where the conference would be. And then there's been a significant body of research carried out in Irish and UK waters over the last 30 years. But I guess we'd all go back to Frederick Russell's The Medusae of the British Isles, and I'm sure everybody's very familiar with that book. But in terms of research, you have to think of the work by Cathy Lucas on Aurelia and Southampton Water, and then the work of Chris Lynham and Andrew Briley, with Steve Hay on all the looking at time series in the North Sea and in the Irish Sea. Then the work of Graham Hayes, putting tags on jellyfish, looking at their vertical movements. My own work with Emily Baxter, and now with Maria and Hannah, who are there with you, looking at how different jellies interact with salmon aquaculture. But there's work from Philip Lamb and Martin Taylor looking at metabarcoding to look at predators of jellyfish. And then the work of Houghton and Chris Harrod and Fleming looking at the trophic ecology of jellyfish using stable isotopes. So there's a great body of work on gelatinous zooplankton that's happened in certainly the last 20 years in Irish and UK waters. So where is Cork City? Where would the venue would be? Well, it would be at University College Cork, which is based in Cork City. And Cork is an island, or Cork is based in Ireland, and it's on the fringe of Europe. And over on the right of the panel here, you can see Ireland is in red, and then Cork, County Cork, is in red here. And we zoom into that, you can see Cork Cities here. It's based on the River Lee just as it comes into Cork Harbour, which is one of the largest harbours in the world. And then this is this on the bottom left is University College Cork. It's about 170 years old. It's a bit like Hogwarts, some of these old buildings. And then the streets in Cork are very accessible. It's very easy to get around. It's a small town, or small city, has a population of about 125,000 people, and 25,000 of those are students. So it's a young population. So who would Team Ireland be? And well, it would be Team Ireland and UK, because Cathy Lucas said she would help run this event with me. So, but of course it wouldn't just be me in Ireland. It would be my colleague Davian Harberlin and Hannah Brownlow and Maria McGuinness, who you've met and who are there with you there today, would help run this event. But I would also lean on some good colleagues, John Houghton and Chris Lynham, who are very close by. They don't know anything about this bid, so so don't tell them. And yes, so look, there'll be a lot of us that would help to run and make this a successful event. But that's it. Thanks for considering Ireland, and enjoy the remaining time in India. And take care. Bye bye.
And we actually had like an embarrassment of good offers from that. So thanks for everybody for considering that. And we'll see some of the alternatives crop up in the future, I'm sure. Okay. That's it. Do you want to come up, make closing remarks? Okay, so with that, I think we're, oh, Sawarna's going to come. He says.
I'll ask some participants. So I request a few participants, preferably representing each continent, for feedback on the symposium. Please come forward and give us a feedback.
Hello, everyone. I think that in the name of all the audience, we are all happy with the results of the symposium. We'll have part of the attendees will continue with the workshop, and I think it was a prolific. We had prolific discussions. We met new people. We met old people, old friends, old colleagues. So it was a very good time for us. We met a lot of people from India and from Asia, so it was really, really good to know these people. Some we had contact only by email, and now we know the person, so it's very good. And I think that I can extend my gratitude to all the organizers in name of all the audience. So I ask you to applaud all the people from India. Thank you.
I think for an awful lot of us, this was actually our first ever trip to India. And so it's been a fantastic opportunity to learn about the culture, but also about the really rich research that's being done on jellyfish within this region. And I'm hoping that we're going to lead to a whole lot of new collaborations with our Indian colleagues, because I think there's so much work that can be done within this area, and it's been so understudied, and I think there's an enormous amount of potential for a lot of, I guess, collaborative research to come from this. So thank you very much to our Indian hosts. It's been a fantastic opportunity, and hopefully many of us will be returning to India sometime in the future. Thank you.
Okay, thank you. So I request Professor Biju Kumar to propose vote of thanks.
It's almost we surpassed all the time for the dinner, so I'm not going to extend. I have no paper with me, but I have a lot of love and thanks to all of you for being with us. I'm sure that there may be some hardships in between, and in fact, I remember the day when I raised my hand in Cape Town, and Mike was the person who gave me support to raise my hand. And, I don't know what was in my mind at that particular period of time. And then I thought, anyway, it is a challenge to do it in India. And there are specific reasons, and primary reason which I thought is that there are stronger knowledge gaps. And India is developing a fishery, but sustainability is a larger question. And India should host this seminar primarily. And also many of our friends have not been to India. And this is a place to visit, to learn, to work together, because we have a larger culture of hosting people. India never invaded another country so far. And that was a culture. We have a culture of living with the other groups and other communities across the world. We welcomed, in fact, many people from outside, the so-called invasive people as well in the recorded history. And then when we took up, the strength was definitely the International Advisory Committee. Right from the day the first meeting started, they were with us, guiding us in framing the themes, the policies, the methodologies, the timelines, etc. And then we initially start thinking about, along with the CMFRE, to do it at Cochin, which was more accessible in a way in terms of flight connectivity. And then they were imposed with another larger meeting, and they could not even spare scientists, because it was a very larger conference of 2,000 people, and almost all the scientists were engaged in that. And then this forced us to bring this conference to Thiruvananthapuram, Trivandrum. And it was a challenge. And then we thought of even postponing it for some time. And then we thought ultimately, anyway, we decided that, we'll hold it in this part of the year. We'll go ahead. So we went ahead, and the strength was all of you. You all came, and probably the largest number of abstracts received for this kind of a conference. And definitely, I'm sure that with your support, we have succeeded in organizing it. And the second strength, which actually supported me in organizing this program, is my colleagues from the University of Kerala, from CMFRE. All of us, work together, and the strong team of my research students in the university, they were volunteering. We were actually, there was no event management except for the travel, and all these things were done by the student community, all the services. I thank all of them, and including my own professors and senior professors, including Dr. Padmakumar, Professor Jayachandran, all were, despite the fact that they were seniors, retired, and they came for this success of this meeting. And I thank every one of them, every one of you for the success of this meeting. And you have a dinner getting ready. I'm not naming anybody specifically. Dinner ready. And this time we will have the live things, the parota, which is made live. You can see how it is made, and the live sweets, everything. And then you try the live juice also. That's something special. Probably you might not have tried many of the Western friends. Thank you all. Good night to all. And those who are coming for the workshop, then you please contact Lisa for the next 3 days. What is our plan, etc. Wish you all a happy journey back. Thank you. Good night to all. Namaskar.
Yeah, so then just thanks also to Bijou and the Indian local hosts again, please. Wonderful meeting.